Good morning. Welcome to Individual Therapies on the Rise. My name is Garrett Homry. I'm Research Director for Regulatory Policy at Duke Margolis Institute for Health. Very excited about today's workshop, convened by Duke Margolis and our partners at the Rare Disease Innovation Hub at the Food and Drug Administration. Duke Margolis is convening this workshop under a cooperative agreement with the FDA, but it is not a federal advisory committee. We will not be voting, making recommendations, or other recommendations today. We have an ambitious day ahead. Before we jump into that, friendly reminder, statement of independence. Duke Margolis is an independent academic organization. We do not take partisan positions, etc., but we do welcome folks to share their mind throughout the day. We will be delving into a large amount of incredibly exciting but also complicated topics. It mirrors our process over the last several months planning this meeting. Lots of work, very exciting, but also complicated on a range of issues. The primary job we were tasked with was from the FDA when we took this on a few months ago was investigating non-clinical and clinical data that would inform submissions to the FDA for individualized therapies, often called N of ones, probably more appropriately N of few. As part of the process over the last several months, the Duke Margolis team interviewed 34 stakeholders. I bring that number up because it's about double the amount that we normally do in preparation for these meetings. The input from those interviews and our recurring working meetings with the Rare Disease Innovation Hub and more extended teams within the FDA are what inform today's agenda, including participants, topics, etc. Although today's primary focus is intended to be a deep dive into the non-clinical, clinical trial design and data aggregation, integration related questions associated with the development and administration of individualized therapies. And today we are speaking specifically about gene editing and individualized antisense oligonucleotides. Despite that being the primary focus, I know we'll hear a lot about the following also. Risk-benefit calculation that is unique in these very small populations with severely debilitating or life-threatening diseases. CMC-related issues, what FDA approval looks like for therapies that don't neatly fit into the traditional NDA or BLA approval paradigms. A recently published New England Journal of Medicine article from the FDA Commissioner and CBER Director will also certainly be part of the conversation. And something that's a bit atypical for our Duke Margolis FDA convenings, issues of cost, reimbursement, scalability, those will be recurring themes also. I do the list to reiterate that it is ambitious for a single day. As I was just talking with some of our colleagues, if I had my druthers, today's big day would be followed by a 30-person roll up your sleeves workshop tomorrow on CMC issues. Day after that, same thing on platforms. Day after that, talks. Give us time. Only so much we can do in a single day. Quick walkthrough of the agenda before handing off to my wonderful colleague, Amy Comstock-Crick. We'll begin today with opening remarks and presentations by the FDA. We'll then hear a panel on the current landscape for individualized therapies. After a short break, we'll resume with presentations and a discussion on non-clinical considerations. Following lunch, pick up the conversation with clinical considerations, which will also include presentations and a subsequent discussion. Afterward, a session on generating evidence from clinical experience and integrating existing data. And then we'll wrap up the day with a reflection of the day's discussions, consider opportunities for what's next. Attendees, excuse me. Attendees, whether in person or virtual, are encouraged to contribute to the meeting through the Slido Q&A function, which will be passed along. And those questions are passed along to the moderator in the ever-handy iPad that we all carry around. We in the FDA want to make sure that these discussions are engaging and interactive. So please don't hesitate to submit questions. We'll also have mics available for our in-person attendance. So we'll be running those. Please raise your hand. Quick reminder also that the meeting materials are available on the Duke Margolis event website already. Here's that QR code for Slido. You can go ahead and scan that now or submit your questions during the discussion. We'll also have it pulled up kind of throughout the day. time permitting. We really do intend to bring some audience questions into our panel discussions. Also, please join the conversation online via LinkedIn as well, where you can mention Duke Margolis when sharing content related to this workshop. Now, it's my pleasure to introduce our speaker for opening remarks, Amy Comstock-Rick, Director of Strategic Coalitions for the U.S. Food and Drug Administration, Rare Disease Innovation Hub. Travel luggage.
Are you going to do some reading up here? Thank you, Garrett. And as Garrett said, I'm Amy Comstock-Rick, the director of the Rare Disease Innovation Hub, and it is really with great enthusiasm that I welcome you to Individualized Therapies on the Rise. As many of you know, this is the Rare Disease Innovation Hub's second Rare Disease Innovation Science Exploration, hence RISE workshop. These workshops are a series of scientific workshops sponsored by the Rare Disease Innovation Hub and led, as you've just heard, by the wonderful team at Duke Margolis. RISE workshops are multi-stakeholder education and engagement opportunities for drug developers, FDA reviewers and staff, and patient organizations to engage and collectively educate about novel approaches and emerging issues in rare disease therapy development, with a strong emphasis on issues common to multiple diseases or a class of diseases. The first RISE workshop was held on September 3rd in this room, and virtually, of course, was discussing clinical trial control options, external and internal, particularly in very small disease populations. The agenda for that program, which I encourage you to look at if you haven't, is available on the Duke Margolis website along with the recording. One guiding principle of the Rare Disease Innovation Hub is to closely involve the rare disease community in ongoing conversations about innovative approaches in regulatory science, including this workshop series. I cannot really emphasize that enough. A real value and goal of the Hub is to find ways to further engage the expertise and knowledge of the rare disease community, drug developers, and patients to inform FDA policy and process, separate from individual product review. Many of you with whom I've met always hear me say we do not do individual product review, and that gives us the freedom and the opportunity to discuss issues and concerns and suggestions coming from drug sponsors and patients, separate from any individual product. This has been our first year of the Hub, as you know, and I hope that you will hear more about our engagement with the community in the coming year. But in that vein, in June of this year, the Hub opened a Federal Register docket to receive proposals for future rare disease workshops. The Federal Register docket opened and has received many proposals, some of which helped guide today's program, as well as led us to the topic for the next RISE workshop. I encourage all of you, if you are so inclined, to submit a RISE workshop proposal. The docket that is now open will close on December 31st, but I assure you that a new docket will open shortly into the new year and will remain open for all of 2026. I will tell you to mark your calendars. The next RISE workshop is March 30th, 2026, and the topic will be announced in the next few weeks. Honestly, we're very close, but it doesn't have a title yet, so we can't announce it without a title. Turning to today's program, I think all of us can imagine a day when individualized medicine transforms medical care from a generalized approach to a precision-based approach, where treatments are as unique as the individuals they treat. While that day may not be here yet, we are getting closer all the time and at lightning speed. It's FDA's mission and responsibility is to adapt to and facilitate this transformative scientific advancements while ensuring scientific and ethical standards are being maintained. Today's workshop is a part of that ongoing process. You will hear from FDA speakers on its latest thinking in this area, and in turn, FDA reviewers and policymakers will hear from many of you about the opportunities, advancements, questions, and concerns in this field. With that, I have a few people to thank who have worked very hard to put this exciting workshop together. The team from Duke Margolis Institute for Health, of course, Garrett Hemray, who you just heard from, Christine Podsiad, Mia Williams, and Brian Cantor. The outstanding, committed, and loyal team from the FDA working group who put this together, led by Teresa Baracchio and Anna Quilis. There are many more in the group. I thank all of you, but it's a long list. And of course, the Rare Disease Innovation Hub core team, Pippa Friedman, Kathleen Davies, Stephanie Amacaro, Julie Valancourt, Sandy Retzky, and Nancy Guan. With that, I am going to turn to our first presentations of FDA speakers who will lay the foundation for the day. We will hear in this order from Teresa Baracchio, Director of the Office of Neuroscience in CDER at FDA, Captain Bart Rogers, Clinical Pharmacologist, Office of Clinical Pharmacology, Division of Translational and Precision Medicine, CDER, and Brian Stoltz, Branch Chief of Gene Therapy, Office of Therapeutic Products, CBER. So with that, I will welcome and turn the podium over to Teresa Baracchio.
Hi, thank you. It's really a great pleasure to be here today. And I'm going to try to start us off by setting the stage of what our experience with individualized therapies at the agency has been so far, to set the stage for the discussions that will follow. So therapies targeted to specific disease-causing genetic variants have been in development for years, but it has only been recently that we've been able to realize this goal for individual patients. And we are honored to have many participants here at the meeting today who have advanced the science to make this a reality. And a few of you may get a shout out in my talk here. As regulators, our first mandate is to ensure that therapies are developed with acceptable risks. We take into consideration the severity of the condition, and we need to ensure adequate safeguards in clinical studies and in the post-approval setting. Our second mandate is to evaluate and ensure that therapies are efficacious for their intended use and that those benefits are sufficient to justify the potential risk of the product. Individualized therapies for patients with rare genetic conditions present unique challenges for these goals. There may be an insufficient number of patients to run adequately controlled trials that provide the strongest evidence for the benefit and characterization of risk of a product. And the small number of patients limits the potential for commercialization and investment, which leads to fewer resources available than would typically be required for a commercial development program in a more prevalent condition. The agency's initial and most extensive experience with individualized therapies has been with antisense oligonucleotides, or ASOs. More recently, gene editing therapies have taken the spotlight. Although there is a potential for other therapies to target unique genetic variants or specific pathologies, ASOs and gene editing therapies currently offer the greatest potential for development and commercialization for very small disease populations. And these products are the focus of our meeting today. Although CDER regulates ASOs and CBER regulates gene editing therapies, the centers are working together on our approaches to individualized therapies. However, there are unique aspects of ASOs and gene editing therapies and the individual development programs that may necessitate different regulatory approaches. Any differences in approaches between the centers should not be interpreted as a lack of alignment, but rather a reflection of the unique aspects of the products and programs that may require different regulatory considerations. I will now provide a brief overview of the agency's experience with individualized therapies. My discussion of specific investigational new drug applications, or INDs, will be limited to information that is in the public domain. In 2018, the agency allowed the first IND to proceed for an individualized ASO for a patient named Mila. At the age of 6, Mila was diagnosed with a form of Batten disease, neuronal ceroid lipofuscinosis 7, a rapidly progressive neurodegenerative illness that leads to early death in childhood. Given the rapid decline of Mila, the agency worked closely with the investigator, Dr. Timothy Yu, to allow initiation of treatment with an ASO given the name Milasin that was targeted to a unique unique pathogenic variant in the gene MFSD8, also known as CLN7. The IND was allowed to proceed with less non-clinical and manufacturing data than FDA would typically require for first-in-human studies. After initially showing some benefits from treatment, the disease eventually progressed, and Mila died in 2021 at the age of 10. I would like to acknowledge that both Julia Vitarello, Mila's mom, and Timothy Yu are here in the audience today, and we appreciate your participation in this workshop. In 2019, the agency received an IND from Dr. Neil Schneider for an antisense oligonucleotide targeting FUS pre-mRNA called jasifusin that was designed specifically for a young woman named JC with a genetic form of ALS. JC received the treatment very late in the course of her disease when substantial neurodegeneration had already occurred. She died from ALS in 2020 at the age of 26. Additional IND submissions for individualized ASOs followed. And based on these experiences, CDER saw a need to develop guidance for sponsor investigators for the development of individualized ASO therapies with the intent to streamline development for individual patients. In 2021, CDER published a series of 4 guidances for individualized ASO therapies that provide regulatory advice on administrative aspects of IND submissions, product quality and non-clinical data needed to support an IND submission, and clinical considerations for initiating and continuing treatment in patients. Since then, CDER has worked with investigators to determine how best to implement these guidances in the development of additional individualized ASOs. With the publication of a case report in the New England Journal of Medicine in May of this year, attention has focused on the development of a customized lipid nanoparticle delivered base editing therapy for baby KJ. Baby KJ has carboxyl phosphate synthetase 1 deficiency, an extremely rare inborn error of metabolism affecting the urea cycle, which has a high rate of mortality. In this case, a large multidisciplinary team came together to develop the product. The pathologic variant was identified, a product was developed, non-clinical studies were conducted in mice and non-human primates, mouse models and in vitro assays were created, and these informed proof of concept as well as potential for benefit in informed dose selection and evaluated off-target effects. Within 6 months, dosing was initiated. Improvements in clinical outcomes were seen very quickly, which included the ability to increase dietary protein intake, reduce other treatments, and importantly, it was observed that viral infections did not trigger a hyperammonemic crisis, which would otherwise be expected. Additionally, reduction in blood ammonia levels were reported. It was also reported at the time of that publication that baby KJ has been growing and his neurologic status has been stable, which is unexpected at this age with this condition. However, long-term outcomes and durability of the treatment effects still need to be characterized. We anticipate rapid growth in the development of gene editing therapies following this initial breakthrough with baby KJ. And I'll now discuss some of the issues that we have encountered with the individualized ASO programs that may be relevant to the future development of both individualized ASOs and gene editing therapies. Since 2019, CDER has had over 40 INDs submitted for individualized ASO therapies, primarily for diseases with neurologic manifestations that require an intrathecal route of administration. We have generally followed the CDER individualized ASO guidance documents for these INDs. However, the guidances were written with a few assumptions based on our limited experience at the time that we were writing them. As there appeared to be no clear potential for commercial development, the guidance documents were written from the perspective that investigators would be largely academic. However, there has been interest from both commercial sponsors and nonprofit organizations in developing these therapies. The guidance has specified that individual ASO drug products should be from a well-characterized chemical class for which there is substantial clinical and non-clinical experience that is either publicly available or to which the sponsor has a right of reference, and that has largely been our experience. The guidance is focused on minimal manufacturing and non-clinical requirements to initiate and continue dosing, assuming that INDs would be for unique genetic variants that impact only 1 or 2 patients. However, we typically receive INDs for conditions that impact more than 2 patients, although still a very small number. Notably, the guidances did not address the potential for future marketing authorization or approval. So, what is an individualized therapy? Individualized therapy is a treatment targeting a unique disease-causing genetic variant. Commonly, the term unique has been viewed as a treatment for a single patient or an N of 1 treatment. However, as I just noted, it has been CDER's experience with individualized ASOs that there is typically more than a single patient who may be amenable to treatment. The number of patients to be treated will impact benefit risk considerations and requirements for development. However, it can be difficult to prospectively define the number of patients that may ultimately be treated with the therapy at the time of an initial IND submission. An IND may be initiated for the treatment of a single patient with an extremely rare disease-causing genetic variant who is available for treatment at that time and at that investigator's clinical site. But there may be additional patients who may also be amenable to treatment with the ASO. In some cases, additional patients may be known at the time the IND is submitted, but not available for treatment at that time, or they may not be able to be treated due to other factors, such as a severe stage of disease that's not unlikely to benefit from treatment, medical comorbidities, geographic location, or other factors. More patients may also be identified after a treatment is developed. Once a treatment is developed, the population eligible for treatment may expand due to increased awareness and utilization of genetic screening in patients with the condition. The J-Diffusin program targeting the FUS variant in ALS illustrates the potential for expanding populations. Initially, only 1 patient was identified for treatment. However, additional patients were quickly identified once J-Diffusin was developed. J-Diffusin had the potential to treat a larger, although still very rare, population of patients with ALS due to FUS variants. However, this bigger population would potentially be enough to allow the conduct of a controlled clinical trial, which provides the strongest evidence for the effectiveness of a product. There is now an ongoing clinical trial for an FUS-targeted ASO, and additional data was needed to support the use of the ASO in this larger population. We recognize the urgency to treat each individual patient, but we also must keep in mind that obtaining the highest quality data that we can on the benefits and risks of a product will ultimately ensure the greatest access to patients. Factors that we may consider as part of our benefit risk assessment include, but are not limited to, the number of patients to be treated and the clinical context of the disease, including the rapidity or severity of the disease. It has been challenging for us to set specific requirements on the size of a population needed to establish certain requirements for development because the clinical context for these patients is important. However, in general, as the size of the population to be treated increases, additional data may be needed to characterize the benefit and risk of therapy prior to initiation of clinical use under an IND or potentially to support a future marketing application. A therapy for patients with more slowly progressive disease and longer life expectancy may require more or different data than a disease that is rapidly fatal. I will now discuss some of the development considerations for individualized therapies. Product quality is a critical aspect of therapeutic development. As ASOs can be readily manufactured, manufacturing and product quality of ASOs have generally not been a major limitation to initiating individualized therapies, especially because the ASO guidance documents specify that ASO drug products should be from a well-characterized chemical class. Given their complexity, Both in vivo and ex vivo gene editing products require more manufacturing information than ASOs to initiate clinical treatment or to support a marketing application. However, benefit risk considerations are similar for ASOs and gene editing therapies. The agency recognizes that the development of non-clinical data to support initiation of first-in-human dosing adds to costs and timeline for development. However, given the novelty of many of these products, there is a need to provide proof of concept and assess risk prior to initiation of clinical treatment. Notably, as many of the patients with genetic diseases that will be treated will be children, and these therapies would be considered to have a substantial potential for risk, there is a legal and ethical need to offer the prospect of direct benefit for treatment of pediatric patients under the requirements in 21 CFR Part 50, Subpart D, which is titled Additional Safeguards for Children in Clinical Investigations. The overall goals of the non-clinical studies are to provide proof of concept, identify a maximum safe clinical dose, establish feasibility and reasonable safety of the proposed clinical route of administration, and identify potential toxicities and physiologic parameters that help guide clinical monitoring and risk mitigation plans. A frequent issue that we've encountered in CEDAR with the ASOs is that non-clinical studies may not be designed to provide adequate data to cover the maximum doses that may be used clinically. The studies may not test doses that are high enough or provide data to document that higher doses cannot be achieved to establish a maximum feasible or tolerated dose. This highlights the need for early engagement with the agency when planning individualized therapy programs. As previously noted, benefit risk considerations such as the severity and rapidity of the condition and number of patients that will be treated will factor into the data that will be needed to support clinical use of the product. However, once there is clinical experience with a product, that may also potentially be leveraged to support dosing in additional patients. Initiating treatment in humans requires careful planning to ensure that safeguards are in place and that quality data is collected to support continued treatment, repeat dosing, or expansion of treatment to other patients. Appropriate regulations must be followed, including obtaining informed consent that addresses not only the risks that are identified, but that also considers the uncertainties around risk and benefit, given the more limited information that may be available from manufacturing and non-clinical studies. Institutional review boards need to be notified and provide approval. As I previously noted, if children are to be included in the protocol, the requirements for Subpart D must be met. The individual's clinical and genetic diagnosis need to be adequately characterized with understanding of the role of the gene variants targeted by the product in the pathogenesis of the patient's disease. Identifying the appropriate dose is challenging for both ASOs and gene editing therapies. Although non-clinical data inform initial and maximum doses, for ASOs, intrapatient dose escalation is often used where dosing is initiated and escalated to higher dose based on pharmacodynamic effects and potentially to clinical response. Dose escalation may be used to a more limited extent in vivo gene editing products. Critical to this is the ability to develop pharmacodynamic biomarkers that are relevant to the therapy and disease that can assess response to therapies and can be informative both for dose optimization and supporting the efficacy of the product. There are substantial challenges with assessing benefit and risk in individuals for these very small populations. Although randomized controlled trials may not be feasible, it is still critical to collect high-quality data. Protocols need to have a pre-specified plan for systematic and rigorous collection of data to assess an individual's clinical response for both efficacy and safety. This data is needed to support that the benefit or risk assessment remains favorable to justify continued administration in the case of ASOs or repeat dosing for either ASOs or gene editing therapies or expansion of dosing to additional patients. Such data may also potentially provide evidence to support a future marketing application. As in the case of J-Suffusin, if there are a sufficient number of patients available to support it, a controlled trial should be considered, as it will provide the strongest evidence to support the effectiveness of the product to ensure future access. The agency is open to the use of master protocols, which may include platform trials or umbrella trials to assess multiple therapies for a condition. However, the optimal approach may differ based on the disease and the therapy to be used. The experience with baby KJ presents an ideal scenario for the development of an individualized gene-targeted therapy. These features were outlined earlier this month in the agency's publication on a plausible mechanism pathway in the New England Journal of Medicine, which describes the identification of a specific molecular or cellular abnormality linked to a causal pathway of the disease, development of a medical product that targets the underlying or proximate biological alterations, well-characterized natural history of the disease in the untreated population, confirmation of target engagement based on animal models, in vitro or in vivo assays, and improvement in clinical outcomes or course. There are many genetic diseases that are amenable to this approach, such as the urea cycle disorders. However, there are also many therapies for genetic diseases that will not meet these criteria, particularly in diseases with known genetic susceptibility, but for which the pathophysiology of the disease is incompletely understood. Although there were substantial benefits seen with nusinersen in SMA, and this raised high expectations for the field of individualized ASOs, unfortunately, it's been CDER's experience with individualized ASOs that we have not generally seen the dramatic benefits observed in the treatment of baby KJ. Benefits may be observed, but it's often not improvements, perhaps because of the nature of the diseases that are being treated, which require assessment of slowing of progression rather than improvement in symptoms. These diseases may be more complex. There may be uncertainty regarding the pathogenicity of the disease target, and initiation of treatment may not happen until a later stage of the disease. And in many of these diseases, the natural history is not well established enough to serve as a robust comparator to assess changes in disease trajectories. These considerations will impact the regulatory approach to therapeutic development. So I know this may be a disappointment to some, although the recent New England Journal article outlined some considerations regarding the plausible mechanism pathway. We are still currently working out the details of how and when this new pathway will be implemented. So I cannot offer many details on how this will work out. I'm going to end this presentation by defining a few commonly used terms that may be interpreted variably by different stakeholders. We believe it will help to have a more productive discussion if we can establish a common vocabulary on these terms. So the first word I'm going to put up here, is platform, which is used many different ways in drug development. Platform technology is a well understood and reproducible technology applied to the development of a product or series of related products that consists of the same or highly similar features across development programs. This term can apply to ASOs or gene editing therapies. If you add the word designation on here, this becomes a designation granted by FDA to an approved technology, and approved isn't a key word there, to streamline the development and review of new products made with that technology. This designation is specific to an approved product. It's not applicable to our discussion today. So we will not be talking about platform designations. A platform trial is a type of perpetual trial with multiple treatments assessed for a disease under a master protocol. This type of trial does not necessarily use a platform technology. It is a generic term that could apply to any drugs. I will come back to this term on a subsequent slide, but just a note for today's discussion, when we use the word platform, we will be referring to platform technology, the first term on the screen. Next is leveraging prior knowledge, is a term you may hear used by FDA throughout the day. We will use this term to refer to the ability to leverage prior knowledge from product development programs to streamline submissions and to expedite product development and review of regulatory submissions. thereby leading to quicker approval of products for patients in need. Prior knowledge can be composed of both public and platform knowledge, with public knowledge being widely available, scientifically accepted knowledge, and platform knowledge, while in some cases may be widely available, in most cases it's often proprietary and thus requires approval from the owner of the data to leverage it. And I just put up here as an example that in the CDER ASO guidance, we refer to this definition of well-characterized ASOs, and this would be considered a way of leveraging prior knowledge. And then the last term I'm going to talk about is the master protocol. So a master protocol is a trial that is designed to assess multiple drugs or multiple subpopulations under a single protocol. There are several types of trials that may be conducted under a master protocol. One is an umbrella trial. I suspect that's probably what we would be talking about in most of the situations with individualized therapies. And this is a trial that would be targeting multiple targeted therapies in the context of a single disease. A platform trial is like an umbrella trial in that it studies multiple targeted therapies in the context of a single disease, but it's done so in a perpetual manner so that therapies can exit or enter the trial on the basis of a decision algorithm. And then a basket trial is used to study a single targeted therapy in the context of multiple diseases or disease subtypes. I would recommend that if we're talking about these types of trials today, maybe we stick with the higher level term master protocol, which could encompass any of these, but I think still gets to the point that we would want to discuss about the ability to think about innovative trial designs. So in conclusion, I'll just wrap up here. In conclusion, the initial individualized therapy programs for both ASOs and gene editing therapies presented many challenges, and they continue to present challenges. They require FDA to think beyond our typical requirements and established regulatory processes that were developed for commercial development of products for more prevalent conditions. And notably, the programs that I highlighted were initiated by academic investigators who are not drug developers. And this required intensive engagement from the agency to not only regulate, but also collaborate and educate on drug development and regulatory science. I want to emphasize that despite these challenges, and perhaps because of these challenges, FDA staff are really excited about these therapies. We're very enthusiastic about these opportunities to collaborate and advance translational science and regulatory science, and to expedite the availability of these potentially life-changing therapies to patients. We look forward to hearing and learning from the many perspectives that will be discussed today, and thank you very much. I'm going to turn this over to Dr. Bart Rogers, who's a clinical pharmacologist in CDER.
Thank you, Teresa. Per the introductions, my name is Bart Rogers. I'm a clinical pharmacologist in CDER at the FDA, and I've had the very fortunate opportunity to be involved with all these individualized antisense oligonucleotide programs. So in the next 15 or 20 minutes, I've been tasked with sharing our experience within the CDER realm with these individualized antisense oligonucleotides. You'll notice my usual disclaimer. So when I was thinking about what to convey today to the audience regarding this, I said, you know what, I really need to start in reverse, and that's with the acknowledgments. And I'm speaking for myself, but I know I speak for many of us at the FDA when I say this, that we'd like to acknowledge the patients, the families, the caregivers, the sponsor investigators involved in this space. You guys are superstars in our book, you really are. And we look forward to continued collaboration. I look forward to the discussions today. It's a rapidly evolving space, many unique ideas, many great minds in this audience, so I'm very much looking forward to that. Okay, to my talk. So first off, I want to provide some important context of the distinctions between the CDER program and the CBER program. You'll be hearing from Dr. Brian Stoltz after me. We'll go over some regulatory milestones. Then I'll get into the mechanisms of which these individualized ASOs elicit their effect. And I'll just provide a brief synopsis of our three guidance, CMC, non-clinical, and clinical. And then really the crux of what I want to talk to today for the first time is our current experience in CDER with these individualized programs. And I'll just briefly sort of grease the skids for some of the later discussions of potential pathways forward. Okay, again, important context, because you're going to be hearing from CBER right after me. Compared to CBER, the CDER's programs are not typically for commercialization. And this is an important point of distinction. Preclinical testing in our programs are quite different. There's an abbreviated non-clinical package to reduce burden and delays in treatment, and this brings about different considerations. In addition, the depth and longevity of the data required for the programs are also quite different. The IND programs, if you will allow me to steal a term from the biomarker world, are really fit for purpose. So again, on face, they'll appear different, and we don't necessarily regulate these differently. We regulate them differently in the framework and the context in which they come to us. When looking at mechanisms of individualized oligonucleotides in the RNA field in general, there are a number of different mechanisms which RNA therapies can work through. However, in the individualized space, we're really faced with two mechanisms. The first of these is splice switching to restore a reading frame to produce a truncated protein in the case of a disease where the protein is absent. This would involve a case of a cryptic splice site where an antisense oligonucleotide could bind to change the splicing pattern to produce, again, an internally truncated protein. The second of these, and the most commonly used, is an RNase H-mediated mRNA decay. Now, RNase H is an endogenous endonuclease that cuts messenger RNA when it's bound to a DNA RNA hybrid. And if you'll notice on the bottom of the screen here, this is a very common chemistry. This is a 5-10-5 gapmer. The gapmer of the 10 oligonucleotides in the middle are DNA bases. And when this binds to a messenger RNA, a selected messenger RNA in a Watson-Crick-Franklin fashion, it's then degraded. Now, as a subset of this, we've also seen allele-specific approaches where an oligonucleotide is designed to bind to a single nucleotide polymorphism that's in linkage disequilibrium with the genetic variant that may be causative of disease. And this is important in cases where you want to preserve the function of the wild type strand. So the DNA strand that contains the variant, you want to knock down that message, you want to knock down that messenger RNA, but you still want to preserve the wild type because that may have important function for the patients. All right, relevant history. Dr. Baracchio touched on this momentarily. In December 2017, it really was a shot across our bow. We were awakened with a new type of IND application for the treatment of a CLN7 Batten disease for use in a single patient that I'm sure most in this audience are familiar with at this point. Quickly, other individualized ASO programs followed. And in January 2021, we developed a draft guidance on really the submission process, the administrative and procedural recommendations. And then December of that year, what I like to call our sister guidances came out. And that's our 3 draft guidances on CMC, non-clinical, and clinical considerations for submission of INDs in this space. Now, when looking at the overall scope of the draft guidance, it's important, again, to define terms. An individualized ASO, often referred to broadly as the N of 1 programs. Now, again, I'm talking about the N of 1 programs in CDER, not CBER, are designed to target a unique genetic variant in a single patient, or typically a small number of patients, 1 or 2. This is directly from our guidance. Now, notably, these investigational INDs are for severe, debilitating, or life-threatening illness. This is an important point of distinction. And the oligonucleotide should be from a well-characterized chemical class, and I'll provide some more details there. Key concepts. Severe debilitating means diseases or conditions that cause major or irreversible morbidity. Life-threatening means diseases or conditions where the likelihood of death is high, and oftentimes the outcome or fatal outcomes of death in the clinical trial setting. Now, from a well-characterized chemical class, as Dr. Baracchio was mentioning, this is something we tend to leverage, and this is in the spirit of platform. It's an example of an NDA, which we have a prior approval. So currently there are 21 approved oligonucleotides from the FDA. So if you utilize one of those chemistries where we have, and you have right of reference to those chemistries, where we have substantial experience and clinical experience with that, this would be acceptable for that program. This is not the space to test a novel chemistry. Okay, looking at some high points of the CMC guidance, it's very important that the first clinical drug product should be manufactured consistent with good manufacturing policies in the guidance for industry. Notably, the same batch should be used for the non-clinical studies as well as the clinical investigations if possible, and the relevant specifications should be provided as well as the stability protocols used in the development of that product. Looking at the non-clinical guidance, we ask that sponsor investigators in this space provide off-target assessment, typically something like a blast search, to evaluate for off-target binding of the antisense oligonucleotide. Importantly here, the route of administration should be the same as the intended clinical route. For instance, an ICV mouse would not be acceptable for an intrathecal administration. It can be used to be supported, but we would need another animal study there. This is really the main thing. If you were asking me to boil down the individualized programs here, it's an abbreviated non-clinical program. And what this means is we can rely on a single 3-month toxicity study with at least 2 weeks of in-life data to support the IND submission for rapidly progressing. If the condition is not considered rapidly progressing, then at the time of the IND submission, we ask that the 3-month study be completed. The first in human dose selection should be justified along with the method of selecting the starting dose, and the high dose should be a maximally tolerated dose. Now, this is important consideration here that expansion to a larger population or intent to commercialize would typically warrant additional studies. Again, this is a unique distinction that makes these individualized programs quite different. When looking at the clinical guides, we remind sponsors that they should provide evidence that establishes the role of the gene variant in the pathogenesis of the patient's disease. So it's not just any gene variant, it's a gene variant that is causing the disease for the patient. Sponsors should provide evidence that the gene or gene variants are unique to the trial participant, including prevalence estimates. We realize this is challenging for sponsor investigators. Many of these diseases are rare. There are not very good prevalence estimates, but we ask you to do your best and provide this. Doses should not be increased more than 2-fold. As you'll hear, many of these programs are intrathecal, so we want to start low and go slow, and we take a cautious approach to this. And the protocol should specify a plan for de-escalation or discontinuation if toxicity should unfortunately develop. And both the safety and clinical response, note I picked clinical response, not clinical efficacy, here should be assessed. Now, when looking at the overall landscape, this may come as a surprise to most folks in the audience, and I think really this is the first time this data has been released. We have 49 N of 1 IND programs encompassing 35 unique oligonucleotides for the treatment of 82 patients. And I'll say that and provide some granularity around that. So there's 49 programs. Some oligonucleotides have been used across multiple INDs. Most INDs enroll a single patient, but some INDs enroll more than one. So that's why the numbers are somewhat discrepant there. Now, it's important, when we looked at these programs, we excluded commercial applications, any expanded access programs, non-oligonucleotides such as aptamers. The protocols were evaluated for single patient, amendments, meeting materials, and general IND plans were reviewed from internal databases. Now, when looking across the survey elements, we looked at many things and many types of metadata, including the disease area, the number of pediatric patients, the type of chemistry used, for example, to take in all this and collectively provide our experience, and I'll provide some more details of that in the coming slides. When looking at the program expansion, this probably doesn't come as a surprise to anyone in the room. Again, initially a slow uptake, and there's really been a rapid expansion, notably the COVID year in 2021. Now, to be consistent, I cut the data off at October 1st for the data I'm presenting today, but I can already tell you in 2025, we've already surpassed, so the numbers are even higher than I presented here. When looking at the findings and providing more detail, 88% of the programs were for neurological diseases. 6% were for inborn errors in metabolism, and 6% were for other indications, notably ophthalmology and cardiometabolic indications. 30% of the programs were for an adult patient with ALS, and over half of the population was pediatric patients, so pretty interesting notation there. And almost 70% of these programs enrolled a single patient. When looking at key findings in the CMC realm, there was generally limited characterization. It's quite important to allow for non-clinical and clinical batches to be used in non-GMP or to not be used in a non-GMP fashion, and impurities have occasionally arisen. And given that the majority of these administrations have been intrathecal, it's quite important that the batches are tested for sterility and endotoxins, and sponsors need to use GMP compliant manufacturing process instead of USP 797 for pharmacy compounding. Non-clinical findings. Again, this probably will not come as a surprise, but the rat has been the primary species utilized, and various reasons for hold or delayed start have been incomplete submission packages, inadequate support for the proposed dosing, typically not supporting a 10x NOEL, and a lack of non-clinical dose ranging. Formulation issues. The FDA is uniquely aware of some of the challenges in the field with chelation of divalent cations and subsequent toxicity caused upon administration there. And this is very difficult sometimes to tease out in an acute toxicity in animal species that may be concentration related and is not necessarily applicable to humans, but it's difficult to discern, is it the ASO causing this or is it the formulation? So this is a point of challenge for us. Providing further detail on the route of administration and dosing, as I mentioned, intrathecal dosing was the most common route. 94% of these programs were dosed intrathecally. The starting doses range from less than 5 milligrams to 50 milligram and had tox data from at least a single species. As I mentioned, the rat or rodent was the most commonly utilized species. This does not come as a surprise given that intrathecal mice is quite challenging given the small size of the mouse, and rodent is often utilized given the cost of non-human primates. So we recognize this. Now, 14% of the programs did include an OEL derived from non-human primates. Again, the FDA recognizes the challenge and the cost associated with this. And less than half of the programs included 2 or more species in their tox studies. Regarding dosing, doses were increased in a stepwise escalation, and the final dose was typically increased in median of 5 times the starting dose. Now, a point, you'll notice a large range here, 1 to 50x. Part of that is an artifact of some of the early IND programs where we were quite nervous where we didn't have the regulatory experience with these programs. So we told sponsors to start low and go slow so we would better understand the toxicities of this program. As we've gained greater experience, we have more confidence in these chemistries and the type of toxicities that can be expected. Now, the most common dosing regimen has been every 2 to 4 week escalation and every 12 to week maintenance dose for the intrathecal administration of the antisense mechanisms. For the exon skipping drugs, we've seen a very similar dosing regimen, a loading dose every 2 to 4 weeks and variable maintenance dose. The maintenance dose is typically longer for those as those products aren't protected. They have a longer pharmacodynamic half-life inside the cell. In the absence of non-clinical or PD support, we recommend 3 doses before the sponsor investigators come to us asking for dose escalation. We want to make sure that the patient has had time to respond to those doses before dose escalation. Regarding biomarkers, it's important to note here that less than 10% of the programs have utilized a PD biomarker. Now, we recognize the challenges, especially in the intrathecal space, of quantifying CSF biomarkers. Many of these membrane-bound proteins are below the limits of quantification in the CSF, and I remain optimistic that new methodologies will come forward to allow us to identify PD biomarkers because they are so critical in this space. Now, these PD biomarkers can be used to further validate the target engagement, guide our dosing, and justify the need for dose escalation when possible. So we continue to encourage both the quantification and the development of PD biomarkers in this space. Looking at safety, we've observed reductions in platelets, elevated intracranial pressures, hydrocephalus, and ventromegaly. I will point out that one patient unfortunately did succumb to hydrocephalus, likely caused by the drug. So there are risks and benefits to these therapies. Safety monitoring should be informed by the non-clinical findings. And broadly, we continue to monitor for potential class concerns from oligonucleotides in the intrathecal space, such as thrombocytopenia, radiculopathies, myelitis, and increased CSF protein pressures. So what have we learned from all this? Despite many promising therapies that target the underlying genetic variants, the clinical benefits have not remained well characterized, and Dr. Bracchio hinted at this as well. Now, so that brings us to ask ourselves, why is this? Is there uncertainties regarding the timing of the intervention in the disease process? We recognize many of these patients undergo a lengthy diagnostic odyssey, and they come to us very late in the course of the disease. So again, I remain optimistic that there will be improvements in this space. I really think these oligonucleotides can work if we can get them to the tissues at an earlier status. In addition, we wonder about the adequacy of the chosen dose. Again, this is troubling with the lack of a PD biomarker, measures of benefit, and needed duration of treatment to observe this benefit. Now, another thing that's come to us, and again, Dr. Bracchio touched on this, it is quite challenging to define what individualized means. And it may ultimately depend on the lens that you look at the program. Are you looking at it through the lens of this isn't a single IND for the patient in front of us? Is this for a customized oligonucleotide? Is this for the frequency of the disease variant? At the FDA, we have not wanted to penalize patients because their variant is a little bit more common than another patient when these therapies exist. We've tried to balance the risk and benefit of those programs going forward. And I'm very interested in hearing the discussion with us today. So today, I'm just sort of going to present an optimized scenario. One low-hanging fruit that I think will really benefit everyone as a win-win is having a standardized process with standardized protocols. We would like to see a cookie cutter approach at the FDA. As a reviewer, it's nice to go in and look at these and know where to find certain information. We see quite a variability in the submissions, and we also believe sponsor investigators would welcome this as well. Now here today, I also have outlined really what an ideal scenario looks like. I'm a reviewer at the FDA. I know ideal doesn't always happen. We get it. We see the warts. We understand this. But again, an optimized scenario may look something like this, where you have a unique variant, a rare progressive SDLT type disease, utilizing a well-characterized oligonucleotide. From this, we will start dosing, we will assess treatment response, and then assess the need based on clinical response and a PD biomarker to continue dose escalation moving forward with a pre-specified duration. This would be an ideal scenario moving forward. In wrapping up, I want to thank a number of my FDA colleagues for helping me put together this data and these slides, as well as all our CDER reviewers involved in these programs. I can tell you, when these programs come into the FDA, it's an all hands on deck. I know you guys don't get to see what happens behind closed doors. We work very hard to get these INDs out. We know patients are waiting. We know families are waiting for this. And we know a lot goes into the background. So we additionally want to thank you again. With that, I'll turn it over to my colleague from CBER, Dr. Brian Stoltz.
So good morning, everyone. My name is Brian Stoltz. I'm the branch chief of Gene Therapy Branch 3 in the Office of Therapeutic Products in CBER. And my task today is to introduce you to genome editing products, what we commonly refer to as genome editing products in our office. So this is an overview of my talk today. I wanted to start with a little bit of background information, get into sort of different types of therapeutic applications that we see, and then considerations for developing genome editing products. And then finally, I'd like to discuss sort of some challenges and flexibilities, especially for developing individualized genome editing products. And then I'll leave you with some additional resources today. So what is genome editing? For the purposes of the guidance that we published last year in 2024, it's a process by which DNA is inserted, deleted, or replaced in a site-specific manner in the human genome using nucleases or other methods. And of course, nucleases create a site-specific strand break in the genome, which then triggers repair mechanisms that are inherent to the cell. So there are lots of different technologies that are used for genome editing. These are some of the ones on the slide, site-specific nucleases. The first one, of course, are meganucleases or homing endonucleases. These are large proteins that contain both the DNA targeting as well as the cleavage activity, and they can behave as both homo and heterodimers. Zinc fingers are, of course, the most common DNA binding motif in biology. And so zinc finger nucleases are generally a combination of different zinc fingers fused to a FOC1 DNA cleavage domain. Talons, I won't pronounce all these long names, but talons utilize tail proteins, which recognize single base pairs in DNA. And these are also combined and fused to a FOC1 DNA cleavage domain to create the nuclease. And the last one on my list today is CRISPR, which I think most people have probably heard of by now, CRISPR-Cas9. The Cas9 protein contains two DNA cleavage domains, and this protein binds to a guide RNA, which is what directs targeting to the genome. So a little bit on the mechanism for nucleases. As I said, these result in generally double-stranded DNA cuts in the human genome and are repaired by human inherent cellular mechanisms. These include non-homologous end joining and homology-directed repair. For non-homologous end joining, it generally results, the repair mechanism results in small insertions or deletions of DNA in the cut site. This mechanism is generally used to knock out genes. These are often sort of deleterious alleles. Is what this is used for. Homology-directed repair, on the other hand, utilizes a DNA template to essentially insert your intended genomic edit into the DNA cut site. So there's also some variations on nuclease-based editing. The first one I wanted to cover today was base editors. This utilizes a Cas9 nickase. A nickase is a Cas9 protein that's had one of the two DNA cleavage domains inactivated. So it only results in a single strand nick. These are combined with deaminases. Deaminase proteins essentially are positioned to target a particular base pair in the target region, and through a deamination process results in a transition of that base pair. For adenine base editors, for an example, it would result from an A to a G transition. We also have prime editors. In this case, it's also a Cas9 nickase protein, and this time fused to a reverse transcriptase. The guide RNAs in this case include a template for the transcriptase, and this allows you to write your intended genomic edit into your target location. So lots of potential therapeutic applications for these types of technologies. I'll sort of run through the list rather quick. Those include hematologic disorders, neurologic disorders, ocular diseases, skin diseases, lysosomal storage disorders, even viral infections, and metabolic diseases. So genome editing products can be delivered by several sort of modalities. They include both directly administered products, or what we call in vivo products. These can be administered through mRNA, LNP, or even other viral vectors. We also deal with ex vivo modified cellular products, where the cells are genome edited in a dish and then given to the patient following genome editing. HSPCs or T cells are an example of the types of products we see in this space. So lots of considerations that a genome editing developer needs to consider when thinking about sort of product design. These include the type and degree of modification needed, as well as the mechanism for DNA sequence change. DNA pathways occur at different frequencies, and the degree of modification needed may be indication dependent. For some indications, this may be well known. For example, sickle cell disease, we have a lot of experience with stem cell transplants, and so we understand what a clinically meaningful therapeutic threshold might look like. We have product design and delivery. This includes in vivo versus ex vivo modified cells. How will you deliver your genome editing components? What advantages and disadvantages to each sort of product design? Optimization of genome editing. So all the modalities I discussed at the beginning of these slides all sort of have different optimizations, ways to deliver, and that needs to be considered how that will be handled in your particular product design for particular indication. On and off target editing studies, what assays are appropriate for your product. Additionally, directly administered genome editing products present additional challenge in target tissue specific editing, as well as distribution of the vector and immunogenicity. There's also clinical trial design is the last thing on my list here. I'm hoping that we'll hear a lot more about that in the last two sessions this afternoon. So regulation of genome editing products, like all gene therapies, we regulate them using a science-based approach with considerations of both the risks and benefits for each particular product for the treatment of a specific indication. This includes considering the severity of the disease, the potential for clinical benefit. Just so you're clear, we actually use the same assessments, whether we're talking about individualized therapies or therapies for more common diseases. They use the same science-based, risk-based approach, assessing risk and benefit. Genome editing products, the primary safety concerns for genome editing products is, of course, the unintended consequences of genome editing. This includes off-target editing. We do not endorse any specific methods. You should use what works best in your system, provide a justification for the tools that you choose to assess off-target editing. For in vivo products, editing of off-target cells and tissues is also a concern, and that should be evaluated. We also consider the unintended consequences of on-target editing and the adverse effects of DNA cleavage, such as translocations. It is important to detect deleterious events even at low frequency. Finally, of course, there's also concerns for immunogenicity. They could include immune reactions to the genome editing components themselves, such as the Cas9, the editing outcome, or the delivery system. So I wanted to discuss some of the challenges with developing these products, especially for individualized therapies. So product quality and safety, because these are permanent genomic changes, the safety information needs to be established early so that we can justify a risk benefit for these products. Even for individualized therapies, CMC and clinical represents commercialization need to be considered early in development. So we understand for these individualized products, your phase 1 study is essentially going to become your pivotal trial. And so thinking about the commercial considerations early will help you run your clinical trial, and so that we can get the best evidence moving towards a commercialization of a product for these individualized therapies. Of course, establishing a process for demonstrating the safety for new individualized products is very important. And then determining clinical safety and efficacy from a small patient population, which again, I hope we see a lot of good discussion on this later in the afternoon. So we offer a lot of flexibilities for individualized gene therapies. Those include sort of leveraging pharmacology and toxicology and CMC knowledge across product platforms. This is very important. Allows you to speed sort of newer products to market. Hopefully, again, we'll hear more about PT or preclinical studies in the second session today. We also allow sort of individualized drug product variants to be studied under a single IND. An example here would be multiple guide RNAs. For example, a monogenic disease that could have anywhere from a few to thousands of genetic variants that cause a specific indication. We have flexibilities to be able to study different product variants under the same IND, provided they're sort of scientifically justified. We've also heard about master clinical protocols for individualized products across related indications. So I wanted to leave you with a few final comments. This is my final comment slide. So I hope I've impressed that genome editing products encompass a wide variety of modalities. I think they have a great potential to treat the underlying cause of the disease. There are many challenges of developing genome editing products, especially for individualized therapies, and we highly recommend that you engage with our office early in development so that we can help guide you to developing your product for commercialization, especially. So these are some additional guidance. I don't have a lot of time to get into sort of the specifics of CMC, but this guidance, which was published last year on human genome editing products, provides you a lot of guidelines. This would be a great resource for what needs to be submitted in an IND submission for a new genome editing product. Subsequent to publication of the guidance, we also released a webinar which covers a lot of the highlights, answers some questions about the content in that guidance, and that's also available on the FDA website. And then finally, here's sort of contact information, not only for myself, but for the Office of Therapeutic Products. On here is the Office of Therapeutic Products main line, as well as an email to the RPM group. If you want to interact with our office, that's probably the best email source for you to go to, as well as some other links of other resources for our particular Office of Therapeutic Products. And I think that's all I have for today, so we can move on to the next session, I believe.
I'll go ahead and ask the session 1 participants to please join me. As they are coming up, just a sincere thanks to our FDA contributors, Amy, Teresa, Bart, Brian. Thanks so much for helping us get a a footing on the regulatory side of things before we expand to bring other friends and colleagues up. Okay. Very excited to introduce our first panel. It's titled Current Landscapes of Individualized Therapies. I have the great pleasure of moderating this panel where we'll be discussing current advancements, but also, kind of leaning into accompanying challenges in delivering individualized therapies as a setup for the rest of the day. I'll go ahead and start off by just a quick walkthrough of introductory names, but then as I hand off to each of you, I'll keep my introduction brief so that you can introduce yourselves, both your roles, experiences, et cetera, and also just dive in with some opening remarks related to some of the key excitements, advancements that you're most excited about. But let's jump into remaining challenges also right off the bat. Carrie Jo Bichelle is CEO at Combined Brain, Vice Chair of the Tennessee Rare Disease Advisory Council, and lecturer in neuroscience at Vanderbilt University. Third one down from me. Sarah Glass, Chief Operating Officer of N. Foundation, far end. Dr. Michelle Hastings is the Pfizer Upjohn Research Professor of Pharmacology at the University of Michigan Medical School and Director of the mRNA Therapeutics at the University of Michigan, two down. And lastly, but not least, because she's right next to me, Sonia Valab, Director of Prion Therapeutic Science at the Broad Institute of MIT Harvard. Maybe I'll do this in chronological order now. Sonia, please introduce yourself. Share some opening remarks to kick us off.
Absolutely. Hi. So I'm Sonia Valab. I run a Prion Therapeutics research lab at the Broad Institute. I came to do this work from a previous career. A million years ago, I went to law school. I ended up giving all that up along with my husband to retrain in biomedicine after we lost my mom to genetic prion disease in 2010. So this is a rapid, universally fatal neurodegenerative disease with no standard of care that took her over the course of weeks. And we didn't know what was happening until her autopsy. And that's when we learned that it was prion disease, it was autosomal dominant, and I was at 50-50 risk. I tested positive for that mutation in 2011, and that is when we left our previous lives. So since then, we've done the things, and now we run a lab at the Broad, and we are exquisitely mission-focused on pushing forward meaningful therapy for prion disease in our lifetimes. So I'll just cue up a few of the things I talk about and think about all day that have already sort of been resonant in the remarks today. Number one, prion disease, genetic prion disease is not N of 1. But I want to propose to you guys that exact N isn't our most important framework. I think our most important lens is risk benefit. And when we look in the literature, how fast does prion disease kill people? The literature wants to tell you 5 months for symptom to death. Maybe that sounds rapid. I want to tell you that my group has done our own natural history detective work. And that first symptom is like some storytelling, speculative, retrospective stuff. Like actually, the moment of diagnosis is the moment when CSF is drawn and tested and you say this is prion disease is when you start counting. In the meantime, death is probably you've been on life support with 0 quality of life for some time, as my mom was, and I will regret it for the rest of my life. What matters is, when was quality of life gone? When did you go on life-extending measures? The window from that starting point to that ending point is median of 32 days. So when I come here to talk to you guys or go anywhere else, my 8-year-old says to me, I'm scared because I am worried when you travel that you could die of genetic prion disease and I won't be there. And if you're going to die, I at least want to be there. So when I think about what we need to do for patients, of course, we need to treat symptomatic disease. And there's a lot to say there because we have molecules that have been good enough to put into symptomatic patients for quite some time, given that these people come to me on the threshold of certain death, begging to devote the rest of their lives to science. Who am I to say no, right? So that's the symptomatic situation. But I can't possibly sleep at night feeling that that is the only bar we're setting for therapy, right? For my cohort, for people at risk, we need to be getting to them sooner. We need to be giving therapies preventively. And, in my lab, we have siRNA therapy that we are trying to push forward in that paradigm. We are cleared right now to dose a small number of symptomatic patients. We are on hold to put this reversible oligonucleotide into pre-symptomatic patients. I submitted a response to clinical hold on October 31st, folks. I do not have the money to dose 48 monkeys, and I never will. But I think that we have a whole lot of data to suggest that reducing the single causal protein in the brain down to 0% and for your whole life is a safe thing to do. And I think that in the risk benefit of the exact biology of this disease, which is a lightning strike, that we are justified to move forward. So the exact biology matters, right? The exact story of each disease matters. And risk benefit is always a highly individual thing, even if we're not talking about a variant that has only existed in one person. And also, we don't want to incentivize me to look less hard for people who could benefit from a therapy, right, that we develop in our lab. And we don't want to go back in time and incentivize me to develop a variant-specific molecule, which it's a choice we could have made, but it wouldn't have been the right thing to do, actually. We can hold this therapy up over a larger umbrella of people, and that's the right thing to do. So, you know, I'm just going to say one more thing as like in response to something we heard earlier, which is I appreciate that there's so much, like the complexity of your jobs at FDA, I have the deepest respect for, and how much you see that I don't see that is informing all of your decisions, I also have tremendous respect for. We have been in the position of receiving what feels like contradictory feedback, where on our irreversible, you know, one-time AAV gene therapy, we've been told, we see the rationale for treating symptomatic and pre-symptomatic people in your first study. And probably you'll dose symptomatic people first, wait 4 weeks. You know, you will do the things that make sense, but that paradigm makes sense. For our reversible oligo, we've been told, dose your symptomatic patients, come back to us with more data for pre-symptomatic. And I think from first principles, that might be exactly the opposite of what you would expect. As someone who spends half my time thinking about gene therapy, the reversibility of oligos is so super beautiful. And no, it's not a 2' mogamer. It's not the exact same chemistry. I love the guidances around that exact ASO chemistry, and we will never be able to keep up with technology via the mechanism of chemistry-specific guidances, right? The siRNA that I'm trying to push forward is going to be obsolete in a year if we're doing our jobs correctly. So, I mean, that's another thing for us to think about. I want to pass the baton because I could talk to you all day long. So thank you.
Thank you so much. Appreciate the immediate introduction of risk benefit as anticipated, also tying back to Bart's presentation and identifying the need for earlier treatment, timing being so much of it. Also teeing up some non-clinical points for later. And consistency of feedback, as Amy and Hub talks about, that's a big driving goal of the Hub's creation at the jump. Michelle.
Hi, good morning. Michelle Hastings. I'm a professor of pharmacology at the University of Michigan. I'm a basic science researcher, really turned drug developer in the last couple of years. I spent probably, I guess now almost 30 years studying RNA and RNA splicing in particular, and have had the incredible experience of seeing a molecule that was really nobody even knew what, most people had never heard of, to becoming sort of something that the general public understands and my family asks me about every day. And from there, even now, as I said, becoming a drug developer and seeing discoveries that happened in my lab advancing into the clinic and helping patients. And that was the experience that I've had in recent years was a antisense, a splice switching antisense oligonucleotide from my lab that we developed in a treatment for CLN3 Batten disease to treat a very rare mutation affecting two girls. And I'm here not representing myself, actually. Karen Kahn, who is the mother of those two girls, was invited to be a patient voice here. And so she wasn't able to come, so I have a statement from her. that I would like to read. So I'm so sorry I cannot be with you in person today. Our foundation's director is my dear friend who has been my rock, lost her father suddenly, and now is my turn to show up for her. When my twin daughters, Amelia and Mackenzie, were diagnosed with CLN3 Batten disease just before their 7th birthday in 2017, the 4 Batten Foundation was formed alongside my husband, David, with the mission of funding research and supporting the Batten community. The foundation has changed our lives almost as drastically as the diagnosis. It has given our family an outlet for grief and rallied an entire community, reminding us we are not alone. It has also given me a sense of purpose. For 8 years, we have trusted the same team. They are brilliant, they collaborate, they share, and together we learned perseverance. Our early naive hope for a miracle slowly matured into understanding that science takes time, even as bad Batten continues its relentless toll on our girls. 17 months ago, Amelia and Mackenzie received their first dose of zabroncacin, a personalized ASO created to their unique CLN3 mutation. This was the culmination of years of dedication from our team. Zabroncacin is named in honor of Zebra and Monkey, who have comforted Mackenzie and Amelia at every doctor's appointment and snuggled them for every goodnight kiss. Today, I want to speak as a parent about the challenges we still face in advancing individualized therapies. The truth is, families are often the driving force, not because they want to be, but because every other door has closed. The year leading up to treatment was the hardest of my life. Endless meetings, way too many acronyms, financial strain, juggling the girl's illness, and carrying the weight of the ultimate decision. It needs to be acknowledged how much it costs families emotionally. Carol and I often joke that Lexapro should sponsor rare disease, but that joke comes from a place of truth. I would not have survived that year, nor would my marriage without it. Later this morning, when you discuss how the benefit to risk ratio truly weighed for individualized therapies, I ask, what about families who have spent years preparing? Should that count for something? And what about quality of life gains we can't always predict? If we've spent years fighting for a treatment, do we stop inches from the finish line, or do we move forward, knowing our children may become trailblazers for those who come next? Believe me, as a parent, I have overly analyzed and worried about ethics enough for this entire room. Don't assume that by treating their child, parents are expecting a cure. We're smarter than that. The challenge remains that even after treating our girls, I still lose sleep at night because other children are waiting. I wish I could hand off the baton and enjoy the time I have left with Amelia and Mackenzie. But even after achieving the goal we set 8 years ago, getting to treatment, we still feel the responsibility to keep pushing. At 4 Batten, we believe hope isn't passive. It's not just wishing for change, it's taking action to create it. Thank you, Michelle, for representing our Project Butterfly team. And I know a lot of Project Butterfly teams are watching this, and I thank you as well. And thank you all for listening.
Thank you, Michelle, both for participating and stepping in, but particularly for ensuring that Karen feels like she's here. Terry Jo.
Okay, I'm Terry Jo Bichelle. I'm the CEO of Combined Brain. It's a big patient advocacy group. We're a consortium of 120 other patient advocacy groups. Each one of them represents a rare neurogenetic disorder. I'm a late bloomer. I'm 66 years old. I'm a translational neuroscientist. I've only finished my PhD 9 years ago. It didn't take me 30 years to do it. I didn't start till I was 49 and a half. I started it because I have a son, Lou, who has Angelman syndrome. He's 26 years old. It was the most interesting thing I could possibly think about doing. So Lou I hope at 66, I'm not past my peak. I really hope that I have a lot of years to keep learning and doing good for the world. But Lou, with Angelman syndrome, it usually peaks at about 5 years old. It peaks at about 5, and it peaks at about the skill set and age equivalent of about 1.5 if you're lucky. So people with Angelman syndrome, they may be able to say a word like mama. They may or may not sleep all night. They walk, but their gait may be pretty ungainly, unsteady. They can yell when they're mad. They can hit and pinch and grab when they're frustrated. They don't have a lot of communication skills. So my son's 26. At about 5, if he peaked, luckily it's not a fatal disease. The thing is, we think people with Angelman syndrome live a normal lifespan. We don't know for sure because most of the very elderly people with Angelman syndrome were never given a genetic diagnosis because it didn't exist when they were put in the institutions they were put in way back when. We know there's a man in his 80s in Canada. We know there's a lady in her 90s in Switzerland. We know there's another lady in her 90s in Australia. So we know my son probably has a good 60 more years on him, and I don't. So if he peaked at 5, and then what all the graphs show is that at 15, he starts to lose skills. It's not dramatic. It's very vague. It's the kind of thing where I look on Facebook and I see, look at that video I posted 8 years ago. That was Lou jumping. When's the last time Lou jumped? I guess he can't jump anymore. You know, it's gradual, it's vague like that. It's when's the last time did Lou actually walk across the room without someone having to hold his arm to keep him from falling down? And now, think about it. What if he gets an ASO or another gene treatment? And what if, when I get ready to come to this meeting, I say, hey, Lou, I'm going on a trip. I'll be back in a few days. And what if Lou says, Baba, did you hear that? Lou said a word. He said bye-bye. Lou, you said bye-bye. Did you say bye-bye? Did he really say it? What if, now that I'm 66, it takes me a beat longer to get off the couch than it does him to head into the kitchen to get a cookie? And what if, as I'm trying to get up off the couch, he's already rounding that corner, the one that always nicks him, the one that makes him fall, the one that gives him stitches, the one that breaks his bones? What if I don't get up fast enough to grab him? And what if he goes around the corner? He makes it. He gets into the kitchen. He gets his cookie. Wait a minute. Is his gait steadier? What if he sleeps a couple nights a week, all night? He never does that. What about that? That makes all of our lives easier. It's been a long time since he pulled my hair. He stopped doing that? Is this worth the risk? Who decides? I think my family decides. My daughters, who are going to be taking care of him long after I'm dead, decide. My family makes up the mind about, is that risk worth it for him? I want Lou to have the chance to be a late bloomer.
Thank you, Terry Joe, for sharing the very personal experiences and level of detail. It also highlights the range. We always talk about heterogeneity of experience in rare diseases. But often with these severely debilitating and potentially fatal diseases, we think so much about the very young or later onset, not this range of a multi-decade experience. We'll go back to some of the patient decision-making risk benefit, other contributors after we hear from Sarah.
Thank you so much. Hi, everyone. Sarah Glass. I am Chief Operating Officer at N. Foundation. So I'm a geneticist by training. I've been in rare disease drug discovery and development for about 20 or so years, really working in clinical trials, clinical operations, mostly focused on precision medicine and the role of genetics in drug development. I also, coincidentally, as the universe had it, have a son who was diagnosed with the N-of-1 mutation in 2020. I'm not going to speak about my child. I'm here to represent the story of N-Lorem and actually the progress that we've made. And I think want to tell you kind of where we've gotten to from our perspective. And I think it's amazing hearing it from the FDA perspective. So thank you for sharing that. So N-Lorem is a nonprofit foundation. It was started by the, essentially a gentleman who created antisense oligonucleotide technology like 35 years ago or so, and also obviously, has really kind of been a partner in this individualized space as well as moving these antisense oligonucleotides forward. The idea behind N-Lorem was to really think about, could we industrialize this process? So I joined N-Lorem about a year after it had been established. At the time, the idea is, can we potentially make drugs for a handful of people over the next couple of years? Let's see if we can do it. I was really pretty keen. I thought at first I would go in as a parent and threw that hat off and really thought, wow, this is a really fascinating goal, right? Can we actually make a biotech, scalable process to be able to make these drugs efficiently and effectively? And so ultimately, we have established over the last 5 years, we've created processes from the point of patient selection, patients ultimately after they're nominated by their physician, if we assess the genotype and phenotype, move into our sort of R&D, then biotech pipeline, if you will. To date, we've assessed over 400 applications. So just to give you guys a sense of, one patient, we start with Mila and move through many, many other patients. These dozens of patients today are now hundreds. So you guys mentioned 50 or so INDs. We have at least that many coming next year just from N-Lorem. And so I think really and truly, this is such a timely conversation. And I just can't express how much this means actually to have these type of players together, because I think we're all really trying to work towards the same goal. And so ultimately, we have moved 400 patient programs through these applications. We've accepted about 200 so far. So we have about 100 active drug discovery and development programs in progress. We have submitted, I think, 37 INDs so far. And so we're treating those cover, I think, about 50 patients. So ultimately, what that has translated to is 40 patients on treatment today. We have created, I think, about 30 different unique REDCap databases. So I think the data and that piece that's going to come up later is so important as well. We've really tried to insert kind of the pharmaceutical, the biotech, perspective into enabling these drugs to move forward, to be able to assess the patients and enable the physicians to make that benefit risk decision, make that treatment decision to be able to continue to treat them long term. And that's translated into like 130,000 data points. So I think that's something it's just really important to emphasize. We have done what we can, and we continue to make these drugs, and we want to make sure that we're partnering with all of you to be able to use this data in a way that can actually be meaningful in the approval and enabling access to much broader population. So I think importantly, what we found is that across all of these drugs, we haven't had any ASO-related serious adverse events. We've had one adverse event that we think is really disease drug related, that interaction. And from a clinical benefit perspective, it's actually been pretty extraordinary. And I'd love to figure out how we can share this better with those that matter, because I think contrary to what it sounds like, maybe you guys we haven't done a good job sharing that data, because out of all of the patients that are being treated, we have 90% showing clinical benefit according to their protocol, according to their treatment goals. 90%. And those that are not seeing clinical benefit were actually all ALS patients who were very, very severe at the start of their treatment. So it's really pretty extraordinary. We're seeing a teenager who has never walked shuffling around his house, brother saying, Dad, what's he doing? What's he doing? You know, we have seizures decreasing across the board, patients coming off of their seizure meds. You know, it's just really pretty exceptional what we've really been seeing. And so now the goal is, I think when we first start, the question is, can we actually industrialize this? I think the answer is yes. The cost to date, really the reason that we're able to do this is because of the community. We have 35, and I think this is broadly in all of us in the individualized ASO space. There's probably lots of you here who are part of this process. So every step of the way, individuals who are contributing a manufacturing batch, here, a decrease in different animal studies costs, those sort of things, brings that cost down to 1.2 million per patient. That's still a lot of money, obviously, but when you think in the grand scheme of a treatment that these patients could use for life is pretty extraordinary. And so now we're really focused on enabling access more broadly. And so I think we want to continue to share what we're learning, try to push the envelope, challenge it. We're so excited about, I think, this, this plausible mechanism. I think everybody saw that paper and we're like, oh my gosh, this is it, right? And they're like, wait a minute, we got to figure it out. So I think I was glad to see that logo. I think everybody's going to take a breath. But it's very, very exciting, and just appreciate so much sort of the progress that the FDA's made on behalf of these patients, and I think on behalf of my son as well, hopefully someday.
Yes, hopefully someday indeed. Thank you so much. A lot of what you said is helpful for teeing up the afternoon session on kind of clinical trial design, data aggregation. You talk specifically about Lorem, 400 patient programs, 140,000 data points. Something that was a recurring theme in so many of our kind of 34 stakeholder interviews is how to, in some ways, kind of replicate or frankly build a much broader collection and integration of data, noting the kind of individuality or and a few components here. So I'm looking forward to that afternoon session. Okay, so we've got about 23 minutes. I'd like to get at least 2 or 3 questions out with you all, and a reminder to the audience online and in person, the Slido code, please, QR code, please send questions. Team in the back is aggregating and reviewing. We'll incorporate where we can, but we'll also make sure that everything gets to the FDA as follow-up. Can we go back to risk benefit, patient community, and patient input in kind of clinical decision making, and if a couple of you could please build off of Terry Jo's opening, and offer any practical examples for how that patient community, patient voice, clinician voice has been incorporated into the FDA process or engaging with academics that are developing therapies, et cetera. What can you share related to having a greater input into determining benefit risk, measuring efficacy, et cetera? We'll have a mic to the front after this one. Can I ask 1 or 2 to respond, and then?
Yeah, I mean, I guess I've already queued up the way I think about this, but for both of our populations, symptomatic and pre-symptomatic, I think we are failing to hear them. And for symptomatic patients, I think it is a matter of respecting the point that they have come to, a place where none of us have ever been, where death is a certainty, and it is coming very soon. And I mean, I think we can all look across our drug development ecosystem and agree at some high level that we are waiting way, way, way too long to gather the very first human data. And once you have that, like, you can do all the thinking, you can do all the animal studies, you can do all the everything. You will be proven wrong about nearly everything you thought you knew as soon as you have the first human data, right? And there's a tremendous leap there. But what I want to make sure, when we think about this, like, there is a lot of value to be debated in the various forms of de-risking that we do in animal studies. And I also want to make sure that we are not holding on to particular formats of animal studies as a security blanket that cannot ultimately deliver the security we are looking for, because nothing can. And that's scary to me every day, I promise, right? Like these are my friends. They have become my family. And when we put an experimental therapy into them, into me, we will be taking a huge risk. That will be true no matter what we do in the interim. So I think we need to be having a serious conversation about when humans get the right, under what circumstances do they get the right to cut the monkeys in line? My pre-symptomatic people, like it is so important for them to be able to say, do I experience my life as a totally normal, healthy person? And is that, is it appropriate for us to be thinking about pre-symptomatic trials as putting a risky, innovative, experimental thing into a perfectly normal, healthy person? I can tell you that they wear their risk in hundreds of different ways because they are hundreds of different people. And some of them have spent years of their lives looking into the eyes of their loved ones who are worse than dead, right? My friend Amanda, who has gone public with her story, has lost 5 generations of her family to this disease. She's asymptomatic today. Is she asymptomatic, like totally normal and healthy? No, right? She's living in this very individualized zone of risk where if I could go to her today with a reasonably justified, reasonably safe experimental therapy, she would go on it 1 second later. So I think that's the conversation we need to be having in my disease.
Anybody else want to
Yeah.
add?
I mean, I think from our perspective, because we're looking so broadly across many different patient experiences, I think what's really important actually has been the relationship physicians have with their patients and understanding what matters. So, and many times it's not with the patient itself, with the family and with the caregiver. And so really what we've done is to try to build in that family perspective. It's like, what is your primary treatment goal? It's, and then also for our team to try to layer in that, okay, and also can we think about this in a standardized fashion to the best extent we possibly can? So really thinking about how are we individually looking at the holistic story of each patient, what would be an improvement in their life? And that can include many, many things, right? It can include, how much effort does it take to actually get the patient to the treatment site? What are potential adverse reactions? What is the potential benefit and the impact on a family? Has one of the parents had to actually quit their jobs because they can't actually work? So I think that benefit risk is, we're trying to center that around data that is specific to each patient, try to have as much consistency across patients as we can from a data perspective, the standardization, but then really thinking holistically. And that it just involves, really working closely as closely as we can with the clinicians who are honestly a real part of the heroes here as well, because they are putting in so much into every patient.
I also just want to point out for the programs that I've worked on, the one I just mentioned with the cons, and I don't know if this is always practical, but for us, what has worked just beautifully is having the patient or the patient advocate. We work on pediatric diseases, so always the patient in each of the meetings all of the meetings that we have, and they're the ones that show up every single time to every single meeting, and they're with us talking, learning the science, and that makes everybody feel better. First of all, it motivates everybody incredibly and dedicates them, but it also makes everybody feel better that they're the ones that ultimately are making those decisions, not the clinician and not the scientists. It's sort of the conflict of interest seems to be leveled in that case.
Mm-hmm.
And it really has made that process, at least early in these days, I think work really, really beautifully.
I think we've got to come up with, based on what you said, to a really standardized way to a global scale, to administer global scales, that there has to be a multi-domain responder index that we can all use, or a clinical global impression scale that we can all use, that is basically asking, these are individualized therapies, so we have to be able to ask the individual and maybe the physician, is the patient doing better or worse? Do you feel that meaningful change has happened? In what domain has that happened? Can you describe that? If we can describe that, come up with that and build that, then it doesn't matter if the risk is extreme or the risk is the benefit is mild. You can still be able to judge it based on the individual's response.
No, I think it is a remarkable challenge, though, right? The individualized nature, yet the goal towards kind of standardization on a global scale. Things like symptomatic versus asymptomatic, and just talking about one kind of bucket of disease state. Okay, all very helpful. We have somebody raise their hand from the audience. We ask when audience members do talk, please introduce yourself, even if you probably don't need one.
It's Jan,
Oh, I think that.
okay, Janet Woodcock, private citizen. I'd like to introduce some thoughts about actually how you assess benefit, okay? Both incorporating the idea that it should be patients and families who contribute what is meaningful, but also we need something, we need rigorous approach to this, because otherwise people are not going to be convinced, and we need measurable outcomes that are measured. I think what I propose is, because you have to get away from ordinary drug development, because this is not ordinary drug development. Okay, so put that out of your mind. I think what you need is serial self-controlled studies. Okay, so treat each intervention that you're doing as an experiment. Okay, you can observe the patient. It's going to take a while to develop this intervention. You can observe the patient and determine those things that are limiting them, or what symptoms they have, whether that be seizures, whether that be ambulation problems, whether it be communication issues, and so forth. You can actually pre-specify which ones you're going to measure, and actually measure them before you do the intervention. This is a level of rigor I don't think we're seeing right now, because this has been early experimental work, okay? However, if we're going to really push this forward, that's what you need to do. So measurable outcomes that you pre-measure and you pre-specify. Then you can also talk to the family, the patients, and the clinician, what is meaningful change? And you can pre-specify that as well, okay? And then you treat the patient, and then you see what happens to that patient. Now, that way you're not, people like global impression and so forth, they're too fuzzy. And people are saying, well, George is doing better and he's getting around more and everything. No, we have to have quantitative, measurable outcomes that will convince, like, insurance companies, all right? Because let's really think about this, okay? We need something that can be reimbursed, okay, that shows quantitative benefit. So I would really like people to think about different ways of assessing these individual interventions. Then you can add them up, okay? So in Lorem, you treat 10 people with certain types of mutation, neurodegenerate. What is your score? How many of those met the success criteria that you pre-specified for them? Okay. Now I can tell you many drugs, okay, they are efficacious and the p-value is less than 0.01. Okay. But maybe only 20% benefited. Okay. You're able to randomize enough people, okay, to find that signal. Okay. So you can't expect that 100% of people are going to benefit from any given pharmacologic intervention, but you need to pre-specify maybe what your success rate is, that what you would overall success rate for the cumulative interventions that you would consider a success as well. So sorry that was long, but I do think we need to start thinking out of the box, different. This is different than standard drug development, phase 1, 2, and 3, and randomization and all that. That's not gonna happen here.
Can I just?
Yeah, me too. Yeah, I was say we are doing that, I think.
Yeah.
Too. Go ahead, Michelle. Yeah.
Have, and
Same
obviously like have the standard quantification and assessments built into it. What I want to say is a lot of these diseases which have never seen any kind of a therapeutic, you don't know. And so some of the benefits that we're seeing in the girls are things like dysphagia, neurogenic bladder, constipation, things we didn't even think about, but very dramatic, so again, you have to be open also to this idea of quickly like assessing after having these types of outcomes.
multiplicity problems here in the same way as you do with a hypothesis testing. So these are all bad symptoms, right?
Yeah.
And you can, but you need to quantify them beforehand. So you know that
If you don't know beforehand.
well, you know they have dysphagia, you know they have neurogenic
Right, that's
matter.
true, yeah.
Okay, so you have to, what's wrong with the people? Okay, so that then when it's better, you can actually demonstrate that.
Yeah.
Okay, so
history, yeah.
So what you're saying though is that you could actually categorize all the different things that are wrong with somebody without actually saying this is the one thing we're going to measure it on. If any of these things improve, it could be better. But even then, I have another challenge. How do you measure halting progression? You don't really see a whole lot of improvement. But if you see nothing, it's a win.
Well, you need a trajectory beforehand. You're not trying to convince me, okay? The insurance company, okay? All right. And so you need a trajectory, maybe external controls on the natural history, right? Okay. For slowly progressive, rapidly progressive, it should be okay. You can get a couple months of observations, okay, and then inflection in the curve, right? But you need to, my point is, we have to be rigorous about this and have quantitative, measurable outcomes. I think the statisticians would give you a hard time if you have 20 different outcomes, 20 different symptoms and you see improvement in one, but that's fair, okay? Is one, how big of that improvement is that? Is that enough? Have you really changed the disease? But at least you've measured it all and you have measures beforehand and you have measures after. And so, you're in a different world of actually quantitatively understanding what your intervention has resulted in.
Yeah, and I was just gonna add, that's what we're doing. So I think 100%. So as soon as a patient get accepted, we start to work with that physician on what matters to that patient. And, so some of the patients do have data. So there is natural history. Some you don't have any at all, right? Potentially they might have a very severe disease, but don't have acute needs with the physician. So they don't, but that's exactly. So I think, and the idea, even when a second patient is treated with the same ASO, you're not saying, oh, well, forget the individualized treatments, let's combine you. You're saying we're still going to compare you to yourself.
Right.
For and during.
Measurable.
Measurable, absolutely.
You're going to need videos. You're going to, need, all kind of,
Circles.
and everybody,
Have all
this contingent over here may say, well, it hasn't been validated in this disease. Well, that's never going to happen. Oh, good. They're denying complicity in that. Okay.
So, Sonia, and then we've got about 7 minutes. I want to ask one last question to tee up the follow-on sessions. But Sonia, your response?
Yeah, I was just going to say that like where we are talking about either more than one patient or there's natural history that we can compare to, and there's inherently going to be heterogeneity in that. And maybe it is a list of things and we need to measure all of them. And we also need the patient and family input. How do we weight each of these things? How much do they matter? And we need to build that whole picture. But what I also want to propose is that in cases where we are lucky enough to have a rock solid pharmacodynamic biomarker, and we're like, look, this one protein drives this whole thing, and we've lowered the amount in CSF, and we've validated that that means we're lowering the amount in the brain, and that is reasonably likely to predict clinical benefit. There is your through line that weaves through, like every patient portrait is going to be different, but you've got this beautiful through line that is linking them all together. And in my perfect world, that is like a paradigm in itself. And like, okay, CBER, CEDAR, historical reasons, acronyms, whatever. Like in my perfect world, there would be genetically targeted therapies that we think about in this way. And people say to me, you want to lower the bar. How are you going to prevent like snake oil, whatever. There is just plausible mechanisms, right? There's simply a different way of thinking about something where the genetic prior is in place for you, and that itself is measurable. And now you can tell a coherent story, even against the backdrop of every patient being different.
Oh,
Hi, it's Teresa Baracchio. So I just wanted to comment on what Janet said. Janet, I wholeheartedly agree with everything you just said. We definitely want rigorous, systematic collection of data in these trials. We want good quality data. I'm less concerned about validated outcomes in this situation. In this situation, when we're dealing with one patient, a few patients, very small numbers, that's less critical. But we want rigorous, systematic collection of data. We are open. I think this has actually been really want to hear from Sarah later. We'll talk later about the benefits that you're seeing because they aren't being reported back to us. My comment was more that we haven't seen the benefits that were reported with baby KJ, which were pretty outstanding. I expect, though, that benefits are still hopefully occurring. It's just harder to quantify when we're dealing with neurodegenerative diseases where the trajectories aren't well described. And it's a greater challenge for us, but we do still consider slowing disease progression or no change to be a benefit. We do still consider that. And I want to see Michelle Campbell. Do you want to add anything?
Sure.
This is
Michelle Campbell, Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience. And I also want to just come over and just hug Janet for what she said, because she was feeling my already messages going, are you having a heart attack with what, some of the things you're hearing? And Janet very nicely, adequately said everything that we would say. And I would just want to add that you don't have to convince us that no change in a progressive disease is meaningful. We know that, and we expect that. I think we're having a very lovely discussion, and I think the things that you're talking to us, we hear about all the time, but it hasn't been collected with the rigor for us to be able to say, make that conclusive statement. And we are very flexible in what that can look like and what do we need, but we have to have that conversation because this is the situation we hear about afterwards. And we never knew. We're like, oh, we didn't know that. We could have actually figured a way to help make that be capturable, measurable, right? Everyone comes and looks at me because I am the outcomes person, I'm the measurement scientist, but I don't need to bog down and I must have the most perfect valid scale. I will not be that person. I will say, what is the best way to capture the data that is rigorous and standardized, and what can we glean from that information? And those conversations are going to vary based on your population, your disease, and what the drug's supposed to be doing, what that therapy is supposed to be doing. But we are open to those conversations of how to help best design that and collect that information.
I have greatly appreciated the audience engagement over the last 15 minutes or so. It has now run out the clock on this session. If I may take moderator's prerogative and return to our esteemed panelists, sincere thanks. A couple things came in from the audience, and I'm not sure that we're going to have any time to address them now, but thank heavens we've got about 7 more hours of this. Coming in online, I appreciate these stories. It's incredibly important that we continue to pursue these treatments, but in today's reality, the insurance companies that determine what treatments panelists can undergo, what type of lobbying with the insurance company is ongoing. There are a couple other points in that regard. I would also say, our last question was intended to be, you all know what's coming next, right? We've got a non-clinical session, we've got a clinical session, we've got clinical trials, data integration, et cetera. Can I ask for a final kind of 30 seconds, rest of the day, anything on any of those topics that you most want to hear, non-clinical, clinical trials, coverage, CMC, et cetera?
I could start. It'll be a 1 minute, no, 30 seconds. When patients and their families calculate the risk benefit, we can amass data bit by bit that can be used as the prior data for the next studies, and we cannot lose time.
Bravo. Sarah.
I guess what I'm interested in hearing about is kind of an extension of the FUS story. So we heard that then turned into a trial that required additional non-clinical studies. So I'd be interested in hearing a little bit more and having some discussion around the perspective on once you have data in a patient, several patients, how is then additional non-clinical data considered, and what's the rationale, and kind of, and how all those things might come together to be able to get to that approval, which I think, in line with the question online, our understanding and all the sort of lobbying conversations we've had is with the payers is they need an approved drug to be able to have the conversation. And so for insurance coverage to have coverage for an experimental drug is a much bigger hurdle to get over.
Michelle or Sonia?
Yeah, for me, just looking forward to understanding more how to overcome barriers, which for us, a lot of times are cost and, just the science really, and maintaining rigor in the face of things happening very rapidly. And I think that sharing is a really important component of this. So prior knowledge and having access to that in the public domain rather than these privatization is really important also to make some of these things happen. So I think today is a start. I mean, not just a start, but it's a great opportunity.
And Sonia?
Yeah, I guess I would say, one of my motivating questions all the time is, what can we do? Like, we very, very, very understandably have systems set up to try to protect people. But how can we make sure we're not protecting them against their will? And the second thing I will say, my other big motivating question is, what does urgency look like? We have certain pictures in our head. We have seen ourselves respond in extraordinary ways to actively dying cancer patients, very, very, very sick children, and global pandemics. Okay, sometimes urgency looks a whole bunch of other different ways, right? Sometimes it looks like a high-functioning person in a cute yellow jacket who is going to look like this and function like this until one day, suddenly, she's dead. And then you all have to have a RISE workshop being like, how exactly did we let that happen? And I won't be there to help you through it. So I need us all to think about that. Thank you.
Thank you all for sharing your professional, but also very personal experiences. And also actually, thank you to my co-moderators in the audience. That was a first. We're going to take a 10-minute break. And when we're back, we'll be discussing non-clinical considerations with Charlie Gersbach as our moderator. Thank you.
Welcome back, everybody, for the next session. My name is Charlie Gersbach. I am a professor of biomedical engineering at Duke University, as well as the director of the Duke Center for Advanced Genomic Technologies. And I've got the pleasure of moderating this next session on non-clinical considerations and introducing the first two speakers for the session before we move on to a broader panel discussion. First, I'd like to introduce Tim Yu, who I think needs no introduction to this audience. He is a physician scientist in the Division of Genetics and Genomics at Boston Children's Hospital and Harvard Medical School, and co-founder of N of One Collaborative. Following Tim's presentation, we will hear from Lauren Black, distinguished scientist at Charles River Laboratories. So with that, I'll pass it over to Tim to start the presentations, and join me in welcoming Tim.
Hi, everybody. It's a pleasure to be here, a real honor. I'd like to thank the organizers for convening this incredible group of participants in the community that is facing a really interesting and really important challenge. And I'm just really buoyed to see what this field has grown to be. And we've made a lot of progress, and working together, we can make a lot more. So let me see. I'm going to be starting off this section on non-clinical considerations. Of course, I'm a clinician scientist, and I'm not a pharmacologist or a toxicologist, but still it's a pleasure to offer my perspective on this from the standpoint of an investigator who's been invested in these trials for a long time. And I'm luckily accompanied by Lauren Black, who will be following me as an expert pharmacologist to offer much more detailed remarks. In order to get us going, I thought that I would start by trying to provide some clinical context around the non-clinical considerations that we're discussing, and to illustrate some historical examples of how we think through and use these clinical parameters to guide our decision-making around non-clinical validation. my disclosure slide. And this slide, I think we can do without. But we all recognize the long tail challenge that we're working with here. We're working on the far end of an extreme distribution of rare orphan diseases out even to the once seemingly nonsensical dimension of N of 1. And we're doing this because we are working with a group of diseases, 85% of which are too rare to be considered commercially viable, and finding creative ways to address this true market failure. We've talked about Melisyn. I'd like to just illustrate a couple of examples for how Melisyn has guided our thinking in non-clinical sufficient evidence to move forward with programs such as what we're discussing. We worked on Melisyn because of urgent clinical need. And our goal in that program was to develop a reachable and accessible and logical mutation-specific medicine in partnership with CEDAR, the Division of Gastroenterology and Inborn Errors. And we were able, through a successful collaboration with CEDAR, to bring this to bear for a patient whom we had just met 1 year prior to our first dose administration. And just to illustrate the components and the parameters under which that negotiation took place, we were working with a platform technology, antisense oligonucleotides, which have also been adequately introduced to this group, with certain capabilities, great flexibilities, certain liabilities, and certain unknowns. But we knew that several characteristics made them suited for the task at hand. In working with a neurodegenerative disease, a very challenging set of disorders, which neurologic symptoms underlie, as we heard earlier today, upwards of 80% of the severe symptoms seen in patients with genetic disorders. We were working with a technology that had been demonstrated over years of painstaking work by many others to be capable of eliciting broad cellular delivery to the central nervous system, meaning that some of the basic fundamental unknowns about biodistribution, not all, but some, were addressed. We were working with a system that is capable of supporting in silico drug design, allowing us to move very, very nimbly to re-engineer our drug to its target and to be able to risk mitigate somewhat via in silico measures, some empirical measures, as to where the target specificity might be, what the target specificity might be, and what liabilities might exist. We were able to work with in vitro models fit for purpose to the application at hand, using patient cells in a situation when there was no time or resource to devote to making animal models available. And we were able to do this at multiple levels of scientific validation to look at RNA effects of our drug on its intended target and prove that it was engaging in the way that we expected it to, as well as demonstrate functional biological rescue in those same cells, giving us confidence as clinicians to sit and speak with the regulatory team and our patient and describe that our prior for success was higher than most other non-traditional or most other traditional compassionate use efforts. And we were also able to negotiate with wonderful expert CMC, pharmacology, toxicology, and regulatory advice, expedited paths that were fit for the clinical purpose to complete manufacturing and single species safety testing in 4 months. These efforts seem to be paid off by clinical response. And it has been my pleasure to see us and many others in the field move on with this type of paradigm for other carefully selected patients. Patients with compelling medical needs, patients with single gene disorders, identified mutations, scientifically feasible fixes that could be demonstrated in the lab, often in vitro settings, and as we're talking about, specific mutations in small numbers of patients. And now we have, as you've heard, growing experience with individualized ASOs, beginning with CLN7 Batten disease, a program launched in January 2018, programs at Columbia by Neil Schneider, Jason Fusen, following in 2019. This slide highlights a couple that I know most intimately out of our group. ataxia-telangiectasia in January 2020, in which we were able to treat 2 children with a progressive cerebellar degenerative disease before the onset of major neurologic morbidity. This program is currently, I think, the longest continuous running ASO program on an individualized basis that I know of, running now over 5.5 years. A program for KCNT1 epilepsy for a very, very severe epilepsy of infancy in 2 children who were born and accessible to us in the Boston Children's Hospital catchment, who had a very devastating form of neonatal onset epilepsy. And most recently, in collaboration with Sarah Glass's team at NLORM, a program for intraocular treatment for a form of progressive blindness for a young girl at Colorado Children's Hospital. She's seen here with her clinician, Emily McCourt, and she's doing wonderfully. Each of these programs I could comment on. The clinical characteristics of these programs are results. I'm going to refrain for the moment, since we're talking about non-clinical considerations. These were supported by cellular studies that allowed us to show functional benefit in the lab on an in vitro basis. For ataxia-telangiectasia, we were able to illustrate ASO-based rescue of the DNA damage response using patient fibroblasts. For KCNT1 epilepsy, we were able to make a comprehensive series of electrophysiologic measurements that demonstrated rescue and restoration of normal electrophysiologic firing patterns in iPS-derived neurons. For FLV-CR1 retinitis pigmentosa, that's a mouthful, we were able to make retinal organoids and demonstrate rescue of protein expression as a function of treatment with our experimental ASOs. These programs have been carefully crafted in a way that tunes and increases our prior to be able to go into the clinical program with some risk. But even the careful tuning that we've done to try to streamline this process as much as possible isn't always enough. I don't often actually get to share with my FDA colleagues stories of patients who actually didn't make it all the way through this gauntlet. I'll share a few of those. But I will say that as we are working through these programs, we are working to establish processes that can, and high scientific standards that can be first put forth, polished, refined, but to move towards a standardized process, parameters under which we can show in vitro that this might work. Of course, all of this has to be backed up by appropriate safety studies, which will be the subject of Lauren's talk to follow. But it's still not easy. Here are 2 of the cases that we did not get to bring to the FDA's attention. Here are Addie and Cassie, 2 twins with Niemann-Pick disease that we began working with in 2018. And we developed antisense oligonucleotides that were capable of rescuing expression of the broken Niemann-Pick protein in their cells. And I was on a telephone call with their father in July 2019 to tell them the good news that we were preparing to move forward with CMC filing, safety studies, and IND filing with the FDA, when he had to share with me that his twins were in the ICU with a respiratory virus and 5 days later passed away. We had an excellent preclinical proof of concept package that went for naught. A similar story could be shared about a more recent case, a baby born at MGH a few years ago, Lincoln. He had Zellweger syndrome, a devastating neurometabolic condition caused by defects in the peroxisomal biogenesis pathway. His cells didn't make peroxisomes. We treated his cells with an ASO that restored the generation of peroxisomes, and we're filing our pre-IND discussions with our colleagues at the FDA when he needed to go on a respirator for an unexplained respiratory illness, and he also passed away in the ICU before we got to that point. There are other cases on the cusp that we're working on right now. Henry is a young child with alternating hemiplegia of childhood, a devastating condition that can lead to paralysis of part of his body for hours to days at a time, recurrent seizures, and a looming threat of death at any moment from sudden unexplained death of epilepsy. Sybil, on the bottom right, is a young woman with Batten disease who graduated from college, played high school soccer, but then began losing her vision and began having difficulty with her coordination, and now is having seizures. And she, if untreated, is going to go the way of other kids with Batten disease. Another case that we're working hard on to try to avoid them not meeting our expectations. So I share these only to provide color for our discussion, the important discussion to follow, to remind folks of the urgency, the need for time efficiency, the careful balancing of disease risks and treatment risks, and this management strategies that we can all engage in together. And I'll offer just a few thoughts about how this growing ecosystem can help find paths to navigate those very difficult questions. We've heard of the dozens of N-of-1 patients that have been dosed to date via ASOs. We've celebrated the amazing advances that now allow this approach to be applied to gene editing as well. But we are finding more and more cases. Just with ASOs alone, the N-of-1 Collaborative has pushed out some consensus guidelines for how to recognize these cases, how to find them without having to phone an expert. And early applications of these guidelines to patient genetic cohorts from either disease cohorts like AT, or from single medical centers, or from an epilepsy cohort across a consortium of multiple hospitals, have found 5 to 16% of kids evaluated as eligible for ASO treatment. In these academic ivory tower publications alone, right here, are 85 kids that deserve treatment, just from these reports alone, and that's without even looking in the real world. So we do have to work hard. We should celebrate our advances, but there's a long way to go, and this slide is meant to remind us of that. Just some closing opinions from a humble clinical investigator. How do we manage to keep up with this clinical need? We need high scientific standards. We need fit-for-purposes processes. Some of these I've outlined by illustration. Efficacy in animal models. Can we progress towards efficacy towards patient cellular models? I think we're already doing a good job of that. But how far can we push that? Can we continue to expand innovative, creative uses to make this even more efficient? In safety studies, well, for ASOs, animal studies are important. But what can we do to shave off the time and resource required to do each one while still maintaining high scientific standards? And some thoughts here on the slide, moving from large standard safety studies to smaller, more bespoke studies, multiple species to single species, targeted tissues for pathology analysis, short-term studies. Importantly, I'd like to stress the last point here, siloed results. Our ability to cross-reference studies and learn from platform technology results is something that is unique to the FDA. I'd like to make that less unique. I'd like to make that, broaden that to the community of investigator sponsors who are putting these forth on behalf of patients. The FDA does a wonderful job applying lessons from one program to inform and guide us on our programs. But it would be wonderful if we could do that in a more intentional way, in a more open way. And gradually shifting our mindset from products to processes with this iterative learning that Janet referenced in comments earlier. Last slide of perspectives. How do we manage risk? This was poignantly discussed in the last panel. And I think that the answer is evident. We're finding ways to share the load. Sharing the load between not just regulators and companies, which often have felt like the ultimate arbiters of these very, very difficult decisions. But through these programs, we are working closely with medical centers, partnering with them to discuss and navigate these risks. And I think that we've done a good job of helping our decisions be guided by clinicians and as messengers for their families. Could that be formalized? A provocative idea is that, are there ways in which medical centers might work in a more seamless way to continue to distribute that load with us, for instance, medical centers that might specialize in this type of work. Engaging patients, disease advocates, listening to their messages, but also sharing governance with them, not just as voices, but as co-governing entities. And then harmonizing these risk frameworks across different divisions and specialties and attitudes. One last concrete thought. For data sharing and public transparency, I think that we all can do a better job committing to that. This has been one of the driving principles of the N of 1 Collaborative, and perhaps it could be broadened. Perhaps we can look to precedents historically in the medical and scientific realm. For organ transplant, we have UNOS, we have the Scientific Registry of Transplant Recipients, who carefully collate both positives and negatives from the transplant experience across the United States. Some of you may remember the Recombinant DNA Advisory Committee that was convened to help bring together scientists and the public to allay concerns and to smooth the adoption of what used to be scary technology, the ability to splice genes, into industry, into medical practice, into clinical trials. And perhaps one of the things we should discuss or could discuss is there a role for that type of body to help distribute the load and to come up with decisions that can navigate this difficult between highly personal decisions and system-wide effects. To me, this is all about building a system that's more resilient to risk. Yes, we need to customize risk-benefit, but we also have to think systemically about building a structure in which risk is understood, accepted, and taken upon many people's broad shoulders. So thank you very much, and I'll pass it along for the next talk.
Questions at the panel?
Good morning, and thank you again to the organizers, to Duke, to the FDA, to the facilities folks and the ones running the AV. I'm very grateful to you all. And I'm especially grateful to the people in the room that have had to sit in the shoes of being parents and family members of folks with rare disease. I just wonder if I could take a round of hands for someone who's got a rare disease person in their family. And people that have lost children from this kind of problem. Yeah, I lost my son while I was working at FDA. He had a very ultra rare heart abnormality, so he was operated on at CHOP to no avail. But we had the best attention that the world could have provided to him. So that was at the end of 7 years at FDA, and I moved on. As you can imagine, I needed a change in my life, and I've worked 23 years at Charles River since. And I've had the pleasure in the last 7 years to enjoy a whole new career working with Tim, with Neil Snyder, who I have to apologize in advance. The spelling didn't get corrected. And yeah, okay, sorry. And then within Lorem and in helping train people to look at toxicology differently. I have the benefit of working with almost purely high-risk disease and life-threatening disease during my time at the agency. I started off working on oligos in '92. Followed by transplant medicine, followed by working in CBER during a time in which most diseases we tackled were in all different organs and didn't have any cures. All of those trials were in patients. So I think that's a key problem I want to face you with, is that most of our international toxicology and pharmacology guidelines that are pertinent to regulations were written in a time in which the diseases were easier to cure. They had they had better defined conditions that have been studied since, heart disease, when Leonardo da Vinci was taking hearts out of dogs and looking at how they beat differently. When we were doing transplantation and looking at the lymphocyte infiltration in the 1950s and wondering why those organs rejected. All of those things were studied for many years before we started embarking on antidepressants and antihypertensives. We're now in a situation where we've cured or have significantly ameliorated a lot of these diseases, and we're now down to the hard ones. The things where emotions run high during a time when funding is running low. And we feel a deep, deep uncertainty about our future of being able to sustain this type of technological innovation when we know it's real and present. Right? We can touch these, we can feel them, we can pick up a surgical knife and pick out the right blade, and we can cut based on what the surgeon's opinion is of what sort of tissue they're working with, right? But for genetic medicines, we also have these kinds of tools. We can pick an editor, we can pick a promoter, we can pick a targeted oligonucleotide, and we can surgically edit these very specific problems that can be tested in utero, if we knew or were able to do that, or certainly after a diagnosis. But we all recognize the frailties of diagnosing something after it's already started to develop. Why? I just got the news. I've been a neuroscience researcher since NIH in 1990. And suddenly I found out the other day through a newspaper article that 80% of the neurons have been lost in the substantia nigra before disease, the symptoms are actually visible in Parkinson's disease. I'm like, oh, that's what they're talking about. So for someone who's already manifested disease, and I hearken back to your talk, Sonia, by the time someone gets diagnosed, 80% of their residual Mother Nature's redundant functions are already been stripped from them. And so we are treating the remaining 20% of the neurons and hoping that they can restore function. I mean, that is kind of a pipe dream, isn't it? So we really need to get ahead of these diseases and start treating patients much, much sooner. As far as I know, Tim's application of the oligo for AT was the first time that we were able to vet that with the FDA and say these kids aren't bad yet. Can we go ahead and treat them with the rat study? And I think that was a huge achievement. So with that introduction, I'd like to go ahead and try to put some very unpopular opinions up. This will probably put me at odds with every toxicologist in the room, and I apologize in advance. They know my propensity for liberality, but please, now that you know my background and you know that I've worked on risky disease 35 years, I don't really have a lot of regard for a lot of the international guidelines that were written to suit the masses. We need, and I think like Janet said, we need a specific targeted approach for these things with all the caveats I already listed, and so has everybody else today. So I'd like to mention, I work at Charles River Labs, and we actually do drug development. We're not just the rat sellers. Although we do have lots of cute rats, and I especially like the Zucker. Has anybody seen the Zucker rat? I wish I had a t-shirt. It would remind us all to diet. So Mila and JC, I've just indicated a crude timeline here. And in the writing, let's see if I can get this to work. Is the sparkle thing. Cool. All right. So Mila and JC and another unannounced party all received, either were about to receive or were receiving their treatment back in 2018. So 3 INDs got started at that time. Only 2 of them are public. In the next year, everything started to take off. You notice I raised this up relative to this number. These numbers are what Charles River has worked on. These indicate the height of the number of INDs that have probably been in FDA's purview. If you add up all of these numbers, you come to the number that Bart was acknowledging in his remarks. So an ALS oligo that hasn't been announced, as well as J-Fusin, and subsequently over the next couple of years, a set of 11 N of 1s were overseen by Neil, all FUS ALS, that became the database that convinced Ionis to pick it up for commercial use. But his JC and her subsequent 11 colleagues were all treated over this period of time and were just published in July of 2025 in Lancet. EPEC, and this is AT, the disease AT, Valeria, KCNT1, TECPR2, a child that unfortunately the drug was too poisonous and couldn't move forward. And then another ALS oligo occurred in this time period. These are all ones that I did regulatory strategy for and tried to refine the number of animal studies downwards in order to justify the risk benefit for those high-risk diseases. Here we have genetics. I wanted to mention this, although genetics was the first Angelman's company, commercial entity. They brought the first premise into the psych division for treating Angelman's, and their first cohort was 10 patients. Note that's not much different than JC and her 11. Sounds like rock stars, right? JC and 11. So we have genetics, and they're 10. We're the first treated for Angelman's with an oligonucleotide. And this is when we were able to get the FDA had a quorum, if you will, of different cases under their belt in order to make some sort of coherent guidelines. I have no beef with the guidelines, except, let me just put it out there, they ask for 3 months of data. I can see all the oligotoxicities I need to see within 1 month. And Charles River has worked on over 100 CNS oligos, and we can tell what's going on in the brain by 1 month duration, not 3. That would cut 2 months off. If I had cut 2 months off of the ability to treat Mila, we would have been able to save a lot of her functionality. Same with JC. So the AAVs start coming in around 2022. Dialogues on them originate the year before. Fundraising occurs. Terry Horgan was treated at University of Massachusetts. Unfortunately, he passed away from a combination of his disease and the treatment. And then Michael Paravolakis is a wonderful story. If you look up Terry Paravolakis, his dad's website. Michael is now able, after his treatment, to run to a tricycle behind his mom across the neighborhood, laughing madly, trying to chase his mom down. That's a pretty good quality of life. Although there are many things he cannot do, he's better off than if he had not been treated. So the AAVs really start to come into N of ones here, and this is when CBER starts becoming very active, and I'm sure they had dialogues far before. Then we have the ocular that was already referred to with N of Lorem and TEM. And then we have another number that I haven't really quantified. There probably a lot of these are N Lorem. Vanda did an oligonucleotide, and they're going to be starting to report out that young lady's results soon. And then we have one that we're working up right now that's going to be an LNP mRNA system to deliver into somebody with acute fibrosis of the lung. And then Tim and I are going to be working on the next one that's going to be an oligonucleotide that's going to have to be given into the eye and to the CNS. Now, would you be surprised, knowing my background, that I've proposed that be in a single rat model? Say no. Okay. Because ordinarily oligonucleotides in the eye are done in monkeys. The eye is a very close anatomical correlate, and that seems to be the obvious solution. But have it be known that an oligonucleotide intraocular delivery in a monkey of the type that CDER usually requires costs about $2.5 million. And Sybil does not have the money, and Tim doesn't have the money to pay for that ocular study. Therefore, we are going to be proposing to the FDA imminently that it be done in a single animal species. And there's a reason for that. And that is that oligonucleotides are part of the ocular system, part of the sensory system, the ocular tract, ocular nerve are all part of the CNS. If I'm an FDA reviewer, which I sometimes play, you give the drug into the eye, it can travel up to the brain. It's going to be at the front of the brain. You give the rest of the drug from the back, but that ends up in the brain in a different mix. So you have a biodistribution interaction, and the drug's going to have a different amount of concentration at the front of the brain because some might have been coming in from the eye. I don't want to do 2 separate studies to characterize that, because that's not going to tell me what the patient experiences in terms of drug concentration and in what parts of the brain. So I think there's a logic there, and we'll see what FDA says next month. God bless y'all. Sorry, that's going forward. If I make it through 4 slides, I'll be doing really well. How much? I have 1 minute left. Oh boy. Well, let's just cut to the end. Needless to say, I just wanted to point out, these are all of our existing guidelines. You're perfectly happy to look at these particular clues. I said this is like a scavenger hunt of looking for flexibilities in all the different non-clinical guidelines. The happy news is they are there. They are there. The end of one guideline here, of course, is heavily referenced, but again, I think it stops too short by saying, I think we can do with 1 month. Here, the compassionate use regulations, all of these trials should be under compassionate use and be considered for accelerated approval. One trial should be conceived of, right, that culminates in accelerated approval, such as Terry Pirovalakis's, where he's got half the kids in the country with SPG 50. He's treated them all. That's all the kids he can muster. It's a phase 1, 2, 3 trial. That's what he's going to be able to bring to bear on the issue. The generally accepted scientific knowledge, the GASC, oh, what an unpleasant word, document is probably one of the least appreciated FDA guidances out there. It says you should reference everything you ever knew about the product class, everything you ever knew about the animal, and everything you knew about the disease. And you should put them together in a well-written high school essay and express to FDA about that balance pan that Tim, you said, the risk of the disease and the risk of the drug have to be looked at as the primary drivers of how you design your non-clinical work. And I'll end there. Thank you.
Great. Thanks so much, Tim and Lauren. I'd like to invite the panel up for the discussion and Q&A, including, I think, all 6 of our panelists, who include Tim and Lauren as well. We can come sit up front.
Thanks to you.
Hi.
Okay. In addition to Tim and Lauren, who have already been introduced, we're joined by Lois Freed, the Director of the Division of Pharmacology and Toxicology in the Office of Neuroscience in Cedar at the FDA. Val Myers, who is a Pharmacology and Toxicology Reviewer in CBER's Office of Therapeutic Products at the FDA. Neil Schneider, the Claire Tao Professor of Motor Neuron Disorders in Neurology and the Director of the Eleanor and Lou Gehrig ALS Center, co-director of the Claire Tao Center for Motor Neuron Biology and Disease at Columbia University. And lastly, Fyodor Urnov, the Professor of Molecular Therapeutics at UC Berkeley and Director of its Innovative Genomics Institute. I figured to get started, Tim and Lauren already had a chance to say a few words. We can maybe take a minute or two for our additional panelists to introduce their thoughts and role on this topic, and then we can open it up to Q&A. So maybe we want to start down at the end and come back this way.
Sure. As they mentioned, I'm the Director of the Division of Pharm Tox in the Office of Neuroscience, and I have focused mostly in the area of neurology and have been involved in, I think, all of the individualized therapies submitted to our group.
I am Val Myers. I am a primary pharmacology and toxicology reviewer, so I get to review the files that are submitted, and it's exciting to work in this space with all of you and build programs together.
Good morning. I'm Neil Schneider, Columbia University. You've heard a lot about our efforts this morning. I work to develop J-Suffusin, an ASO for FUS ALS. What began as an N of 1 grew into a large program. We've now treated worldwide over 30 patients through the expanded access mechanism that began with JC. The first 12 of those patients were published earlier this year, as Lauren said. The FUS program, as you heard, de-risked this ASO and convinced Ionis to launch the phase 3 study, which has now completed enrollment. But for many of the genetic forms of ALS, this kind of approach was, commercial approach, I should say, was not possible. And this really led to the establishment of Silence ALS, which I founded together with our partners at Enlorum, shortly after the guidances were issued in 2021, and those guidances were key to making all of what we do through silence, and happy to talk more about that possible. We've had a lot of learnings, both from FUS and now silence, that inform our development of ASOs for the larger ALS population. So thank you.
I work for Jennifer Doudna. The reason I have this honor is 23 years ago, I had the good fortune of developing the toolbox of human genome editing, co-naming the technology and being part of the team that took all of its first in human applications to clinical trial. More recently, I was the lead on the team that discovered the gene editing strategy for Casgevy, which is the first approved CRISPR gene editing medicine. And thanks to Rebecca Arens-Nicholas's and Kieran Musunuru's leadership, I was part of the team that developed the medicine for the child with CPS1 deficiency at CHOP. Earlier this year, my institute received a generous philanthropic gift from the Chan Zuckerberg Initiative to treat 8 more children at UCSF in the next 2.5 years who are living with severe pediatric genetic diseases. And in my remarks later today, I will tell you exactly how we're proposing to do that, with the explicit goal of forward integrating every microliter, every nanomole of what we move towards approval under the plausible mechanism pathway that, while still under construction, I totally hear the agency representatives on that, we would like to provide the first few bricks to try to construct that plausible mechanism pathway with a view to approval.
Great. So I remind the audience that there's the opportunity to submit your questions online using the QR code that's up on the screen. But to get the conversation going, we can start with the first question for the group. What's unique about these class of medicines that we're talking about today is the ability to make very small changes to otherwise very complex macromolecules, such as a few changes in a nucleotide of a guide RNA or an ASO, or potentially a few amino acid changes to a protein, while keeping the vast majority of the drug the same to repurpose that drug for treating different patients. And so the question in this session on non-clinical considerations is, what are the opportunities to reduce the necessary preclinical studies to take one of these drugs and repurpose it for another patient's mutation? So open that up for anyone who wants to take that first.
Since I'm closest to Charlie spatially. So first, folks, we know that single children histories can make phase change differences. Julia's daughter and the child who is Rebecca's patient with CPS1 deficiency create a sense of #yes we can, unlike anything else. More generally, it's really hard to predict severe adverse events from non-clinical studies. And I say this from 23 years of experience doing these non-clinical studies. In my field of genetic therapies, gene therapies and gene editing, not a single SAE in the past 23 years was predicted from non-clinical studies. This doesn't mean we should stop doing mice, but this does mean we cannot fool ourselves into thinking that another 10 years of experimenting on NHPs will make safer therapies. That's not going to happen. So as a specific, we have elected to pursue an indication space where there is essentially no for-profit sector activity, largely for commercialization reasons, where there are 500 distinct diseases that are amenable technically to gene editing today, and where clinicians guide us that we can bring together children, despite having mutations in different genes, under the benevolent umbrella of a master protocol. So in the next couple of minutes, I'll just give you 3 high-level takeaways of how we're hopeful to leverage this unique feature of CRISPR gene editing, that where you change 20 nucleotides in a guide RNA and you get an entirely different medicine, and take that through clinical development at UCSF. So we, first and foremost, benefit risk is the #1 consideration. We have elected to work on severe combined immune deficiencies where children left untreated die, and not everybody is eligible for a transplant. We've also elected to work on a devastating T-cell disorder called HLH. And the 2 things you need to know about this is 30% of children don't survive to transplant. And post-transplant survival times have not improved since 1994. Every mutation that causes this disease, one of 6 different genes, can be corrected using gene editing. There's not a single biotech company in the world working on a single one of these children. Carrying the torch of benefit risk commensurate, we asked ourselves, if a child dies in 3 months, What is the utility of a non-clinical pharm tox package that takes 6 months? We're going to put that filing on the child's grave? I'm dead serious. But so rather than just hand-wring and be histrionic about this, we partnered with Danaher. And first of all, I want to acknowledge I get salary support and research support from Danaher. And specifically, we partnered with 2 leading CDMOs in the Danaher constellation, integrated DNA technologies that make guide RNAs, and Aldevron that makes messenger RNAs and proteins. And we asked ourselves, can we manufacture and de-risk a clinical grade editor in 11 weeks? Because if we take 24 weeks, the child will die. And so what we have under review at the agency is the following 3-component package that I'll review briefly and then pass the floor over to others. Number 1, children with HLH get treated with etoposide. It's a topoisomerase inhibitor. That's the standard of care. People treated with etoposide develop treatment-related AML. So let me ask this audience, I'm sorry, what is the significance of doing off-target analysis for a gene editor for a child who was on 2 months of etoposide? This doesn't mean we should treat children with dirty guides, but this does mean we have to adopt a benefit-risk commensurate mindset. Do you want to say something?
No, that was a very good point.
Oh. Hi, Lauren. I love, obviously, Tim's story about the 2 patients he had who passed away before they could get dosed. So brass tacks terms, we teamed up with the clinicians who treat these children at UCSF. And together with our team, we have developed a package that de-risks the off-targets of the most bioinformatic concern that we find for a given gene editor on demand within 3 weeks. Is that the full standard pharm tox package we would normally submit for something like Casgevy? Of course not, but people who are living with sickle cell disease have years. Children with HLH have single-digit months. So we have submitted this for agency review. We're very excited for agency feedback coming out soon, and as soon as we get it, we will publish it. Our goal, we're a public university funded by philanthropy. We want to be the rising tide that lifts all boats. Number 2, we want to maximally leverage the process. If we have to redevelop the process, good luck to us all. So we're leveraging the process in 2 ways. First, the active pharmaceutical ingredients. Full-scale cGMP guides takes months and many hundreds of thousands of dollars. Children don't have that time. I really want to honor integrated DNA technologies for collaborating with us on that. And in a close partnership, they have worked to develop a process where a guide RNA that takes 5 weeks to manufacture has functionally indistinguishable properties in terms of potency and specificity in the clinically relevant cell type from one that is manufactured to the full-scale, full-chested cGMP. I want to assure the agency here, we are not treating children with dirty guides, but we have developed a process that is benefit-risk commensurate for the fact that the kid has 3 months to live and that guide RNA will never be used again. Again, we will publish all the learnings. And last thing I want to say is, we talk about platforms and that word has many meanings for many people. Our plan is this. We're going to build the plane as we're flying it. We're going to build the platform as we go along. Here's what that means specifically. We will treat one child in that mode. We will see how that child does. We will speak with the agency. And then for the second child, our goal, and I really want to thank the agency and many representatives here who have made a major contribution to this, we're not going to redo redundant studies. Once we have clinically addressed the known risk related to a particular accelerated path we have developed, we will argue to the agency we do not need to redo that study again. If there's an SAE that's treatment-related that's due to the editor, we will stop. But honestly, folks, I'll close by saying, and I'm very eager we have wonderful panelists here. I think when we look at 2025, we have every opportunity to say to ourselves that this is the year, 2026, when we as a community, I really want to echo Julia's vision here and Sonia's as well. This is the year when the entire community of scientists, manufacturers, regulators, clinicians, and the patients, most importantly, came together to realize what a remarkable technology constellation we have. And we together developed the courage to advance them through clinical development and to approval in a safe and responsible manner.
Thank you.
Excellent.
Yeah, very good. I want to see if anyone wants to add or respond to that. Maybe Val?
Yeah, I would happily. Thank you for all those thoughts and information, Peter. That was really helpful. And I'm sure you'll get wonderful CMC feedback from my colleagues. I recognizing that we are all here to develop safe and effective therapeutics. We also know that very small changes in guide RNAs can have significant differences on safety. So just like we don't default to saying you must use an NHP for your safety studies, we also don't default to demonstrate safety in animals. Especially with guide RNAs, you can use cell lines, and in that manner, you can do things quickly, and we're here to support that.
I might add that the challenges for the ASO community, I think, are very different. I mean, the guidances of 2021 were based on decades, really, of growing experience with this particular chemistries that didn't just suddenly appear. And though our challenges are very different with ASOs, there are still many challenges to face in the preclinical, non-clinical, and clinical worlds that we're trying to, we will discuss today. I think that we have learned, as we did in the ALS case, a lot from our experience with FUS. These have informed our development of other ASOs. In each case, though, there needs to be a really, I think, important focus on the specific biology of this of the specific ultra rare form of the disease, because there are issues related to strategy and dosing and in clinical management that are unique to each subtype, even on the subgene level, different variants leading to different phenotypes and different requirements. So, I mean, I think that the challenges for the ASO community are different, maybe more mature, perhaps, than it is for the gene engineering. I think that through the ALS program, 30% we heard of the INDs are from ALS. I think this is what we have here as a paradigm for others to follow as we try to broaden this technology.
Yes, can I say that, as mentioned, the N of 1 guidances, the individualized therapy guidances, were developed based on a good battery of data that we had on specific platforms, or I won't use the term platforms, chemistry backbone. And so those guidances were based on that general information. And that's what we use to reduce the kind of non-clinical studies that are necessary. And I will say with the individualized guidances, the non-clinical guidance, all of the discovery, all of the data selection, all of the candidate selection, I'm assuming sponsors would do that in any case. And it is an absolute truism that many, many of the candidates that are developed for that one specific patient are discarded because of adverse effects in those in vitro assays. The only thing that we have determined based on our extensive evaluation of the data at the time that we worked on those guidances was the need for one study, a 3-month study in one species with that species justified. It does not have to be a non-human primate. We prefer if it's a pharmacologically relevant species. In many of these cases, there is no pharmacologically relevant species. We understand that. And in that case, we certainly accept, and we're on record as having accepted rodent. At that time, a 3-month study seemed the best for us. We didn't have data suggesting that shorter duration studies would be sufficient for this very, very, I have to emphasize, very, very truncated program for these. And in cases where there was rapidly progressing, life-threatening indication, we only asked for 2 weeks of in-life data. And that is, I mean, someone that's been here for a very long time. That's extremely minimal. But at that time, we felt because of the indication, because of the concerns, because of all the things that people have, mentioned in these patients, how critical they are and how desperate they are for therapy, we thought that would be perfectly reasonable. And we think, the minimum that we could possibly review in order to determine whether or not we feel comfortable enough. And we do need to determine that we believe that we're comfortable enough that the safety of this is sufficiently demonstrated that we can allow this to proceed. And so, we have based those very, very limited data that we need from a non-clinical standpoint based on our understanding of those backbones.
Great. Questions from the audience?
Is that on?
Can you hear me now? Yeah. Okay. I'd like to make two relevant comments. I think first on GMPs. Now, my colleagues here might disagree with me again, okay? But that's supposed to be a fit for purpose regulation. It is not supposed to be an ironclad. And if you actually read the regulation, which sometimes I wonder if the inspectors read it, okay, it says, adequate to do this and adequate. If you're making small amounts, do you really need a separate quality control unit? Do you need a separate loading dock with inventories and double sign off? No, I do think that is like a false flag out there as far as what you actually need. And I would push back against the agency or anybody, okay? GMP regs were written for mass production by factory workers, okay? And so obviously you have to have a great deal of controls in there, okay? And things still go wrong, as you know. So that's one thing, because I would hate to see like this, and I've seen such huge investment in making very small quantities in laboratory conditions, okay? And for investigational use. Now, obviously, the product has to be well characterized and well controlled, but that's different than than having a separate, whatever, all these things. Number two, I don't think this burgeoning field, which we saw from Lauren, I don't think we should repeat some of the mistakes of the past. We've seen this with the small molecules, and now we're seeing it with the AAV vectors, okay, which is the data are not public. And so people don't know what's going on. There are people in this field sitting right here who are surprised by what Bart presented this morning, right? And we'd probably be more surprised if we could see the tox data from all these. But why should it just be sequestered in company, or academic files, and then the FDA has to, like every 5 years, try to put it all together, okay? This is going to be a dynamic field. And I think like the AAV vectors, the public does not know. They don't know the fatality rate. They don't know the denominators. They get all upset when they hear different things because there isn't transparency and sunshine around this. So I know we're talking about this afternoon, but I think for the toxicology, it shouldn't just be a small number of people who set the standard and then everyone complains about it. Everybody should be able to look at this and say, this is appropriate, right? It's because it's a community consensus, it shouldn't just be one or two people. And if Lauren, who probably has the greatest experience in looking at this, from Charles River, is saying one thing, well, then we ought to have the ability to look at that and say, that's the case, or there's some outliers over here, it's not the case. But instead, we're sitting here and we don't know. It's you and then what Lois is saying, and we just don't know because the data are not available. So I'm making a really strong pitch that as part of this, if we really go down this route, there should be real, and the RAC or something like that, that was what, because people were afraid we'd have the Andromeda strain and, we'd all grow yellow ears or something, right? That was very, very helpful in giving sunshine to the new recombinant, technologies at the time and settling people down. And I think we're going to need the same thing here. Thank you.
We're about out of time. We've got maybe 1 minute left. And so once again, a great discussion and great participation from the audience. Maybe to wrap up, we can just go down, to hear from our panelists one more time for 30 seconds, maybe commenting any concluding thoughts, but also with the flavor of how we can take advantage of the data that already exists to make these more efficient non-clinical packages. Maybe starting with Lauren.
So of the 15 slides I didn't get a chance to address, in the back, there's an ask. In there, I went specifically through what I would wish for in terms of a new guideline on toxicology for these. I hope you consider that feedback, call me, berate me, whatever. I'm more than happy to carry that into the N-of-1 collaborative and then have them debate with me along with the medical officers, because I believe, unfortunately, a lot of pharm tox has been conceived of based on old pharm tox for big honkin' drugs. We need to look at this under the compassionate use regs, which, if I want to summarize in 10 seconds, say we have certain risk of the disease. We have not no whale, we have probable risk of the drug. Certain risk of the disease, probable risk of the drug, and possible benefit of the drug. And that turns the FDA risk assessment for ordinary pharm tox on its head. And I think we need to turn that upside down or through the looking glass and re-examine it.
As mentioned, thanks to Rebecca Arntz-Nicholas's leadership, I had the good fortune of being part of the team that developed the medicine for the child at CHOP with CPS1 deficiency. So round numbers, about 220 people touched that medicine over the course of its route to that child. And I can tell you from my lived experience, the number one phenotypic marker that every single team member exhibited was courage. Courage is defined by Andy Rooney as, quote, "It's not going into a room full of danger unafraid. It is being afraid and still going in." And I really want to salute the agency, and there are several folks in the room who made a direct impact on that. And anyone listening, thank you. Danaher, thank you. Bigger picture point. We are going to have to go into a realm of Rumsfeldian known unknowns. The only way to learn is to treat children and learn from the clinical trial data. We have to jointly display courage, known as benefit-risk considerations, as we advance these experimental medicines to first in human and learn from the trial data as to how to make them safer.
Perfect. Risk is felt by everybody. And this conversation about risk is not focused on any one party. It's felt by the families, it's felt by the patients, it's felt by the investigators, by the pharm tox reviewers, by the sponsors, by the government officials that endorse this work by setting up these structures. And the one point that I'd like to underscore is this idea of a new social compact around accepting this, proactively delineating the framework by which every invested party, which is everybody, contributes to helping set that risk tolerance. And that's because we need to think not only about permission to proceed, but to ask in advance for forgiveness, knowing that we're going into territory which is very difficult. And this concept of resilience, it's my favorite word right now, I think is really vital, and I'd love to see that conversation still happen.
I don't have too much to add other than to say that I really appreciate the collaborative, progressive, thoughtful approach that the FDA has taken to this issue as we begin to move these ASOs internationally into the ALS community. I see this, the FDA's leadership is very clear. No other regulatory agency is, I think, thinking as hard and working as closely with the disease and physician communities as the FDA is. I really, I think Janet was the one, and Sonia too made the point earlier that what these efforts are allowing us to do is to treat patients with therapeutics for which in a way that we don't really have enough data going in, but we're learning in the course of treating them all kinds of things about the disease, about what response to therapy looks like, about what the potential of these therapeutics is in the pre-symptomatic versus symptomatic populations, and learning how to apply learnings from one genetic form of disease to another, and from one disease entirely and beyond. So anyway, I think this is a really exciting moment, and we're just beginning to realize the potential of what you've all done in the last 10 years.
I will also be fast, hopefully. I think maybe the last thing that I want to say is I hope everyone comes in and talks to us early and as much as possible, because we want to help push on the uncertainties that you have so that when it does go into a patient, it is as characterized and safe and effective as possible.
Well, let me first say, I agree with Dr. Woodcock that it would be extremely helpful for there to be a very public sharing of information that helps us, that helps everyone. But in addition to that, I think it would be very good, and I will apologize to my colleagues because they're all very, very busy. But the more information that we have, the better decisions we could make. So if there's someone out there who has a lot of information indicating that 1-month studies are better than 3-month or as good as 3-month, well, share those with us. We will review them, we will look at them, and we want to do what's best. We want to do what's the most efficient and not ask for things that we don't need, but we need the data in order to make those kinds of decisions. Exactly. That's what I mean. And well, we have failures, but of course we ourselves. And that's the things we learn from the failures as much as we learn from the things that proceed. And just one last thing, I promise, is that I'm assuming that the best from sponsors, the things that are the safest. We don't even see the things that have failed to that extent. Yes, exactly. Thank you.
Okay, fantastic thoughts. Really appreciate all the discussion. We now have a 1-hour break for lunch.
So, as promised in our introductory remarks, it's a bit of an ambitious day for the amount of topics that we're trying to cover. The interest has been phenomenal. I don't think we said this in the introduction, but we had about a little over 2,000 virtual participants for today's meeting. We have been a bit rushed in the Q&A sessions in general, which is fine. We want the dialogue, but we have been receiving an enormous amount of questions online, and we're yet to be able to incorporate them into any of our sessions. We send them to this iPad that the moderators have. So I do want to kind of implore our moderators to try and make sure that we incorporate some of those questions as well. Great first half of the day. Let's have a second half. Scott.
Good afternoon. Welcome back from lunch. I'm glad to see that I'm not talking to a room full of empty chairs, because I do always worry about that having the first after lunch session. I'm Scott Winicky. I'm from the Rare Diseases team at the FDA Center for Drug Evaluation and Research in the Office of New Drugs. And it's just great to be here to be able to introduce our next 2 speakers. So joining us virtually is Rebecca Ahrens-Nicholas. She is Assistant Professor of Pediatrics in the Division of Human Genetics and Metabolism and Director of the Gene Therapy for Inherited Metabolic Diseases Frontier Program at the Children's Hospital of Philadelphia. And in person, we'll have Richard Finkel, Director of the Center for Experimental Neurotherapeutics in the Pediatric Translational Neuroscience Initiative and a member of the Department of Pediatric Medicine at St. Jude's Children's Research Hospital. And now I'll turn it over to Rebecca to kick off her presentation.
Thanks, and thanks for having me. I'm sorry I can't be there in person. So to kick off the session on clinical considerations, we're going to start by talking about gene editing therapies for metabolic diseases as a sort of a use case for how we're thinking about this. So if you could advance 2 slides, and I apologize, I'm going to be asking a lot for this These are my disclosures. Advance one more slide, please. So if we're going to be thinking about clinical considerations, we need to think of about what the disorders are. And I'm a physician scientist that takes care of kids with metabolic diseases. We've already heard about a wide variety of metabolic diseases today, everything from CLN3 disease, spoken about earlier, to urea cycle disorders, which I'll discuss now. But when you think about these clinical disorders, it's important to realize there's a huge unmet clinical need in our patient population. So this risk-benefit analysis that we've been talking about all day is incredibly relevant here. And for many of our diseases, there's a logical translational path because we know what the cause of the disease is. And for many, but not all, there are biomarkers that can help guide the drug development process. Next slide. And when I speak today about a platform technology, I'm not using that in a regulatory term of art. What I'm really speaking about is an end vision where in my patient population, you can adapt these technologies that are so transformative to be able to help a wide variety of patients with a wide variety of genetic changes. And the technology I'm going to be speaking about today is liver-directed base editing. This is work that is done in full collaboration with Kiran Musunuru. So lipid nanoparticle delivered editor mRNA and a guide RNA designed to perform a corrective edit in the liver of a patient with a metabolic disease. Next slide, please. This is actually great. Now we can go to the next, that works well. Okay, so when we started thinking about this, we thought about the clinical characteristics of the population that we would ultimately like to treat at the very beginning. And I think that's very important as you think about drug development. And we started with PKU for a reason that lots of groups think about PKU. There's a large unmet need, and there's patient populations of adults that could participate in clinical trials where a therapy might actually have a real potential for benefit. And we had a nice program in the preclinical space around this, designing base editing therapies for PKU, and they work. This is just showing you some in vivo data from a mouse model showing full normalization in the orange line of the neurotoxic phenylalanine accumulation in a mouse model of this disease within 48 hours. And so this is very powerful data. And if you do additional preclinical work, you can see that it normalizes brain biochemistry and other markers of disease. But when we saw this data, we really realized the potential that this potential technology could unlock for other rare genetic disorders and other rare metabolic disorders. So our clinical considerations shifted. So if you go to the next slide, please. And we started thinking about how we could adapt this technology to an entirely different population. In this case, very sick infants with neonatal onset urea cycle disorders. And I just want to take a moment and say this shows the power of this technology, but how you also have to adapt the technology to the patient population, the endpoints, the clinical condition of each individual group of patients. You have to know these diseases if you're going to adapt these technologies. So for the case of urea cycle disorders, where kids are born, they're unable to break down protein, and it results in a toxic accumulation of ammonia in their brain, which can be life-threatening within days of life. So typically they become very sleepy, poor feeding, and can slip into a coma by about the third day of life. And if you don't intervene to improve and get rid of that ammonia, they'll die. And so the time course that we're talking about here is so different clinically between our first case of PKU, where we could be thinking about treating in adults, and a urea cycle disorder, where you're going to have to adapt this technology in real time quickly. Next slide, please. And just to drive home this point, for the urea cycle disorder we're going to talk about here, CPS1 deficiency, 50% of the babies die in the neonatal period. And while we have a standard of care, we have what liver transplantation can improve metabolic control in this patient population, kids have to grow big enough to receive a liver transplant, and they often undergo neurologic injury while waiting. So again, knowing the disease is important, and knowing the speed with which one has to work is also incredibly important. And we knew for urea cycle disorders, we would have to work rapidly. Next slide, please. So I'm going to talk through the process that we went through in developing the first personalized gene editing therapy for a urea cycle disorder relatively quickly so we can get to some of the learning points. But I think it's important to illustrate this as a case example of what one needs to think about clinically when they're working to develop personalized or any sort of platform technology. So if you go forward, please. So many people have heard this story before. We've heard of baby KJ. He was born, he had a very high ammonia level and was very sick in the neonatal period, and he was rapidly started on life-saving dialysis at our institution. Metabolic testing, thank you very much, confirmed he had a proximal urea cycle defect. And this was really by day of life 4. And it's important to highlight here that you have to be able to diagnose the disease in order to be able to treat it. So rapid diagnosis is so important. Next slide, please. Again, molecular diagnosis and rapid molecular diagnosis is very important. And this is not the case for so many of my rare disease patients who have a long diagnostic odyssey. This particular indication, we tend to get the answer pretty quickly, but for a lot of our populations, it takes much longer. So we need to expand access to diagnostic testing. He had variants that looked like they could be amenable to adenine base editing. And importantly, he had incredibly severe variants. So it was that clinical knowledge that we thought that he had the most severe form of a urea cycle disorder that allowed us to start thinking through that risk-benefit ratio for this particular individual, because we were really worried that he would either die or have brain injury prior to transplant. Next slide. We then transitioned him to our standard of care. And I know not every diagnosis that we've talked about today has a standard of care, unfortunately. We do for this group of disorders. And again, when we think about the ethics of translating a lot of these transformative technologies, I think it's important that we offer standard of care and we make sure we are doing our best to care for these patients while we are trying to develop these therapies. Next slide. In the lab, we developed a lot of preclinical models, including in vitro analysis of his variant. Next slide. And did a screening process. And this is, I think, some of the first screening data that's been shown today in terms of how one actually tweaks or targets that guide RNA that has been spoken about to individually address a patient's variant. And I think this is also important because we've talked about the clinical considerations in terms of the severity of the patient, the course of the patient. But through this process, you start to think about, do I actually have the tools for this individual? And matching whether or not you can develop that tool in a way that has the potential for benefit. Do you see that there's editing at the intended site? And whether or not, as we'll talk about in a minute, we think that editing is enough, knowing the disease, to make a difference. Next slide. This, I think, is an important clinical consideration, and we've spoken to this already, and there's already been a lot of discussions around the medical ethics of this. But thinking about how we approach families in an ethical way, both about experimental therapies, but also as these become, hopefully, in clinical approved drugs down the road, how do we ensure equity of access to these therapies? So having our medical ethics colleagues guide our actions is incredibly important, or help us guide our actions is incredibly important. And for the case of KJ, they were involved in every step of the way. Next slide. So as we continued down the rapid drug development process that we led for KJ, we developed mouse models, again, going back to the earlier session about having the right preclinical models and whether or not this is necessary for every future case going forward. But obviously for this first case, it was. Next slide. We did off-target analysis of those models, or off-target analysis of our guide RNA. Again, we spoke about this earlier, but doing this in vitro models of human cells is actually very relevant and important because we want to make sure we are evaluating the risk of unintended edits or unintended genomic consequences in the human genome, not in a mouse genome or another representative model. Next slide. We had a wonderful interaction with the FDA around last Thanksgiving around our request and our thought process for this. And it was really a great partnership. Many in the room have highlighted the fact that sometimes academic physician scientist sponsors are not, savvy regulatory agents. And so it was really nice to have a robust dialogue and get bidirectional input essentially on what we were trying to do. So we appreciate that. We went forward, in our case, we ended up doing a non-human primate toxicology study, again, to think through safety. Again, this is probably not necessary for every subsequent program, but we felt for this first case where it was in some ways precedent setting, we needed to do this. Next slide. We did proof of concept studies in our mouse models, again, to show the potential for editing or corrective editing in a mouse model. And keep going. And then we got our clinical batch. And we tried it in cells and it worked as expected, which was very reassuring. So it was with these data in hand, if you go forward, and with our final off-target editing data, which again has to be thought through in the clinical context of the patient, right? That risk benefit of unintended edits or unintended genetic changes needs to be considered with how severe the disease is. Furthermore, when we think about clinical considerations, we did our off-target editing analysis targeted to our patient's specific genome. So it was actually personalized to him to help guide that risk benefit in his unique setting. Next slide. We then submitted our single patient expanded access IND application. And after a week of going back and forth, a very helpful way from the review process, we received clearance to move forward. And he subsequently, if you move forward, ended up receiving 3 doses. So that's the timeline and sort of what our thought process was around clinical considerations for this particular use case. If you go forward. Okay, so I think when we think about trying to design a single patient expanded access IND or a clinical protocol here, it's a little bit different than what we're going to be thinking about as we move forward for formal clinical trial studies. But in the context of this patient, I think there's some guiding principles that could be carried forward as you think about clinical considerations for targeted base editing therapies. We wanted to maximize safety. And for an infant, we thought that meant that you had to start at a low dose. And with LNP base editing therapies, you have the opportunity to give repeat doses to try to dose to effect. So it was mentioned a lot this morning, start low and go slow. This was our thought process because this had never been done in an infant. For practical purposes, we were going to give a maximum of 3 doses, and all the doses had to be in by 120 days because we didn't have long-term stability testing planned, I'll be honest. And we have to think through the potential benefits versus the costs of things like long-term stability testing when you're intending to have a drug treat one individual. We thought a lot about immunogenicity of not necessarily the drug product, but a corrected protein product. If this worked and the CPS1 protein was corrected, what would happen? And we decided on a course of immunomodulation. I think this is a field in terms of clinical considerations that we need to think a lot about as we think about these personalized therapies. And then importantly, we had an oversight committee, somewhat similar to a DSMB or Data Safety Monitoring Board, that helped guide our decision process. Next slide. This is what his actual dosing schedule looked like. Thank goodness he had no serious adverse events. Go forward. Just to make a lot of data short, after dose 1, he could tolerate more protein, and after dose 2, his scavenger medication could be decreased. But these are clinical observations because this is an expanded access IND. To make more definitive claims about safety and efficacy, as we've spoken about today, we need to move forward to some form of formal clinical trial. And I'll speak about that in a minute. Next slide. A lot of people have asked what has happened since our initial report. So what observations have we continued to see? One is in terms of thinking about measures of potential target engagement, we are seeing an increase in something called the blood urea nitrogen, or the BUN, which is a marker of ureagenesis, especially after dose 3, which is very reassuring. And if you go forward another one, when you're actually looking at the patient and thinking about how he's doing well. So he was discharged from the hospital at about 306 days of life. He's now tolerating more than the recommended daily allowance for protein. We keep him on a low dose nitrogen scavenger, partially because in some ways that's a security blanket. Those are medications that are thought to be life-saving if a child becomes sick with a urea cycle disorder. So this idea about how do you titrate off medications is one clinically that we're going to have to think through. We successfully weaned off his first immunosuppressant, and we're working on his second one this month. And he's had a number of infections, including things that typically would absolutely trigger hyperammonemia, like croup requiring steroids. And these are just showing you some of his biomarkers. So again, to highlight the point made this morning about how each patient can be their own control, you need to collect data before and after the intervention. And this top graph is showing you the BUN, that urea number. And you can see it was essentially undetectable most of his life. And now, especially since being discharged home, he clearly has a normal BUN. Similarly, glutamine, which is a toxic or buildup metabolite, has normalized and is in the normal range. So these are guiding thoughts or guiding biomarkers suggesting that something might have happened. Next slide, please. Okay, so in terms of where we are, KJ is still going to need urea cycle management. And I think this is important as we think about these programs clinically, is that these kids, these patients, these subjects that we treat don't lose that underlying diagnosis, and they still need ongoing surveillance and management to make sure that they are safe. We showed hopefully that it's possible to do this in 6 months, but it's not sustainable, right? You need to come up with a more sustainable model, and that's something I'm going to try to speak about. We need longer follow-up studies and non-invasive biomarkers of editing, because for things like liver-directed gene editing, liver biopsy is not the standard of care and is actually thought to be a bigger risk than benefit in this patient population. And so how do we measure whether or not we actually are doing what we think we are? We need delivery to other organs, but ultimately we really need to move from N of 1 studies to platform trials for a number of practical reasons that I'll speak about quickly. Next slide. So if we think about this, how do we move from editing solutions in my lab or Kieran's lab or any of our labs into the clinic? The preclinical side of this is actually becoming relatively more straightforward, and there's a clear path in terms of how you just assess whether or not a genetic variant is amenable to a base editing therapy. This is just showing you data from a variety of variants that we have. The real difficulty is how do you actually move from the lab into first-in-human clinical trials and then hopefully to an approved therapy. And that is what is really essential if we're going to scale this for patients. Next slide. So the questions that we've been trying to ask and try to get clarity on, and we appreciate the partnership with our FDA partners and regulators on this, is what do we need to do to be able to move from variant 1 to variant 2, 3, 4, and 5 in order to make sure that this is safe and this has a potential for efficacy? Do we really need to keep going back to non-human primate studies like we did this first time? Are any animal toxicologies studies beyond that first sort of gold standard package necessary? When really what we want to make sure is happening here is that each guide that we change in our drug product for variant 1 or variant 2 or variant 3, whatever subject is in front of us, that guide RNA isn't having unintended off-target editing consequences. And really that should be done in human cells, not these animal models. Next slide. Oh, we skipped a couple. Go back a couple more. As he goes back, I'll tell you, I think, what the slide is going to show us. So essentially, what we've asked permission for from the FDA is around this idea of having a master protocol as it was nicely reached out or described today. I don't know if we can go back a couple more, but the idea is that we have a single master protocol clinical trial in which patients with any one of 7 urea cycle disorders can be enrolled as long as they meet the inclusion criteria. And the importance of this is that we can then take a broader view and understand, okay, if I have one subject with, CPS1 deficiency and a different subject with citrullinemia, what can I learn across the population of urea cycle disorder patients? Well, we can learn a lot. We can learn a lot about safety because the safety is really dictated by the delivery mechanism. And then we can learn a lot about the potential for efficacy because we can see whether or not in patients across the spectrum of urea cycle disorders, you can have a meaningful benefit, both thinking about biomarkers, but more importantly, thinking about those things that our patients are telling us matter. Things like, can they tolerate more protein in their diet so they can grow? And along those lines, can they be stopped? Their medications be stopped? Do they need a liver transplant? And so these are the type of endpoints that we are thinking about, and we've shared sort of our plans publicly as to what we're thinking about doing as we try to launch a clinical trial. And I don't know if the slides can be fixed to go back to my next one, but I'm happy to keep going without them. And so if we are able to do these master protocols, why does it matter? And in the end, it really matters because for those of us who are practicing clinicians, we know we spend most of our lives trying to get things approved. The prior authorization process with payers can be, a little bit of a discussion. And why approval matters is that we need to work with payers to get these approved and reimbursed so that we can scale. Since this KJ story has broken, we have had requests from thousands of patients asking for help. But we don't, in all honesty, have the money or the support or the infrastructure to be able to do that as a series of single patient expanded access INDs. And it wouldn't be beneficial to the community. We need to have these clinical trials, generate the data to be able to support approvals, whether it be through the plausible mechanism pathway, and I know we're still very early days learning about that, or through another pathway. We need to get to a point where we can have a drug that we can prescribe and then convince payers to reimburse for. And some of that convincing is going to be some of the hardest work. And we're working on that up front to start thinking about how do you make the case to payers that for a kid with a urea cycle disorder, it not only is clinically beneficial to avoid a liver transplant and have a one-time, one-and-done therapy, we hope, but also it's cost effective. And with the case of the urea cycle disorders, we have hard numbers we can use to make that case. A liver transplant costs about $1 million, and a year of nitrogen scavenger medications is about $750,000 a year. So you can very quickly start making that argument. And I think that's important as a field as we think about clinical considerations of these therapies, that we have to make that argument. Because if we get these drugs approved and then they're not reimbursed, well, it's not going to help anybody. And so I think this is a continuous process that we all have to work together to achieve. But a huge part of that is getting regulatory approvals and regulatory agreement, essentially, about what we're trying to do. And with that, I'm sorry you couldn't see my last couple slides, but I'm happy to talk about anything during the question and answer session.
By Dr. Aaron's Nicholas on base editing. I'm going to switch over to ASOs and give a somewhat parallel discussion. And I'm going to pivot quite a bit from the prepared slides, and I'll have to go off script because I want to really address some of the topics that were raised this morning. So I'm a pediatric neurologist. I work at St. Jude, and I'm so pleased to be invited to participate here today in this workshop. So I was assigned the task of talking about both challenges and strategies for ASOs in this context of personalized medicine. These are my disclosures. I'm going to build on a bit of what you heard from Tim Yu earlier on what is an ASO, but more specifically, how do we in the clinic consider them, and how does that relate to the topics we've heard today? So you've heard that ASOs are generally short, about 20 base pair nucleotide segments on a DNA-like scaffold. They enter the cell by endocytosis, but they don't get there systemically. So for neurologic diseases, which is largely what I work with and the topic here today, we're talking about largely intrathecal delivery, but then it has to get into the target cell. Okay, and then through Watson-Crick base pairing complementary, it's going to attach to a unique area in the genome and that area of interest. And it's either going to be the messenger RNA in the cytoplasm or the pre-messenger RNA in the nucleus. And it's prone to endonuclease degradation, as you heard. So we have to consider all these factors of what is the drug and how do we get it to the target. There are two main classes. There's the 2-MOE, it's spelled out there, which is the more common one that we've been talking about today. And that's primarily good at getting into the brain and liver, not good at getting into muscle. Whereas the PMOs, or the morpholinos, as they're commonly called, are very good at getting into muscle. So the FDA approved exon skippers for Duchenne. Those are in the morpholino category. Pretty much all the other drugs we're going to talk about are in the 2-MOEs. But there's a new strategy, a new generation coming along as well, peptide conjugated, this and that. But as you heard this morning, we're going to be focusing largely on those that are tried and true when you're dealing with this N of 1 or N of few population. There are two main strategies that you heard about earlier. So there's non-allele specific, where your ASO is going to attach to both alleles. And if it's a dominant disease, it may knock down the healthy or wild-type allele as well as the mutated allele. And then there's also an allele-specific technology that you heard about, and that might target a SNP on that mutated allele to try to knock down just the mutated one. So those are other important things to consider. And then the two main mechanisms of action, one is to try to upregulate, in other words, increase the amount of a deficient protein. So nusinersen for spinal muscular atrophy, a splice switching oligonucleotide would be a good example there, because that's a loss of function situation. And then the other, which is actually more common, is where you have a toxic gain of function situation and you want to knock that down. And that's where the gap mirrors are useful by recruiting the RNase H enzymes to break down the toxic messenger RNA. Okay, so all of those really build into the clinical program. These are some examples of ASOs that have been in clinical trials, and I've just bolded some that have actually been used of N of 1 or N of few. And I'm not going to belabor it other than to say systemic, intraocular, central nervous system of many different types, and then peripheral nervous system as well. So broadly based. There are some considerations that have already been touched upon. There's some advantages of ASOs. It has a fairly long tissue half-life. So the pharmacodynamic effect is such that you have to retreat, yes, but retreat relatively infrequently, every couple months or so. But it's also good that if you do see toxic side effect, you can stop it. It's going to it'll melt away over time. There are different modes of delivery, again, IV, subcutaneous, IT, and even in some cases intraparenchymal that are being considered. And it has generally low toxicity, particularly with intrathecal administration. We do know that we need to monitor for systemic side effects, renal, in particular coagulopathy. But generally speaking, with intrathecal delivery, current experience has been quite favorable from the toxic side. Okay, I want to turn now to everyone's favorite article from last week, and I've abstracted what I felt were 10 main takeaway points, and those are highlighted here in the bold print. And I'm going to try to speak to each of those in the ensuing slides. And so this didn't come from the authors, but this is what I think we want to think about as we consider what's the next step here. Are we going to be getting a new guidance that will help us move this field forward? So I'm not going to go through each of them or read them, but they're there, and let's consider them now in the current context. So as a first step, let me remind you that this is an evolving community. You've heard earlier from Tim Yu about the N1 Collaborative, which is really a facilitator, an accelerator. It brings together scientists and clinicians to try to promote the field. And the NLORM, which you heard from Sarah Glass, about how spectacular they've been in being able to actually submit these multiple N of 1s, get these drugs to the patients. And it works. So they have an industrialized program, as you heard. And then I'll mention at the end, there are some international efforts as well. So this is not only a US effort, but I think the world is looking from this latest FDA communication for some guidance on the path forward. I work at St. Jude. We've been trying to promote development in this topic of personalized medicine by a series of workshops that are listed here. And that's been quite successful at bringing different stakeholders together and being able to share ideas. At St. Jude, the mission is to advance cures and means of prevention in pediatric catastrophic diseases through research and treatment. So obviously, what we're trying to do here in our neurology, our neuroscience initiative, is within the mission of the hospital. And so our program, it has these three parts. It's a preclinical part, a clinical part, and then the Office of Strategy and Alliances to help pull the community together. And that's been very effective as we get our program off the ground. Our personalized medicine program we call Gemini Genomic Medicine Initiative, and it's a translational effort. It's purely philanthropically based, but we're trying to generate knowledge and share that with the broader community. And so how do we develop an ASO for one individual, because this is all patient focused, but develop something that knowing that there are likely to be other individuals out there, and once they're identified, how do we actually get the drug to them? How do we do that responsibly for the broader community? And we're not the only ones considering that, but it's certainly a challenge. So we try to approach this with an initial focus on, are they really catastrophic neurologic diseases? Are they ASO amenable? And we realize that currently these are in effect, compassionate use, single patient INDs. But there's an opportunity, and we think an obligation, to actually make them into research type topics. So we're trying to make sure that at each step that there's some hypothesis-driven activity, and we can try to answer questions with each patient and learn from that, and there's some accumulated knowledge that we can share. We want to not only develop some of these best practices, but share our data as well with the broader community. Our focus has from the start been on N of few rather than N of one. And we realize we have to work within the current environment of the FDA guidances, but we've learned, particularly from the NLORM experience, that you can do multiple N of ones. There's a little flexibility there. But how can we leverage that for an N of few? And more importantly, what's the path forward towards getting this into a more formal clinical trial? We try to do integrated genomic and functional analyses. And importantly, we're trying early on to engage with both policy and regulatory authorities to guide us so that there's this interaction with the idea that eventually we do want to be able to get these drugs covered by the payers so that we can at least have some of the cost covered, because we need to build a sustainable model. At one point, these are really interesting scientific questions that we're trying to address in proof of concept. But at the end of the day, we need to be able to develop something that is sustainable and can help the broader population. Our program has these 5 integrated working groups. There's a preclinical, clinical, manufacturing. We do manufacture ASOs in our GMP facility for internal use, not for commercial use. We have a data analytics group, and then the external alliances with many in the room here today. So how do we actually approach this? Well, we have certain selection criteria, and I'm not going to go through it all for the sake of time, but just to highlight that there are 2 kind of parallel pathways. One is gene-driven by the patient's variant, and we have to first identify, is that a pathologic variant? Do we know for sure? Are we certain that we know that it's a loss of function or gain of function? And in fact, in some cases, in a particular gene, it could be both. But for that individual, we need to be sure that we know what's going on with that variant in those cells, because we don't want to develop an ASO to knock down something and it turns out, whoop, that wasn't the right strategy. So we have to be absolutely certain we understand the mechanism of disease. And at the same time, there's a patient-driven component. So because it's typically driven by that patient that comes into the clinic or patients of interest to us from other individuals working in the lab. So we say, okay, gee, you have a disease of interest. You have a variant that we've identified. What's the next step? How do we actually take this through some sort of strategy so that we can bring it together, both the gene-directed and the patient-directed strategies, to be able to select and say, yes, this is a disease we're going to focus our resources on. We're going to try to develop an ASO. And we think that this is a good candidate. Okay. These points have already been addressed. I'm going to skip over this. I was asked to talk about clinical outcome measures, like, well, we need metrics. We heard that this morning. We need yardsticks. We need to be able to measure the response to the intervention, to the ASO. So typically we have about 1.5 years, maybe 2 years from that point of saying, okay, here's a candidate, we're going to try to make an ASO. Tim somehow goes on turbo speed and could do it in 1 year. The rest of us take 2 years. But you have maybe 1 year or 2 with that patient to do a run-in observation period. And that's what I want to show you here. So on the screen here, let's see, do we have a laser? Yeah, okay, maybe. I'm going to just call your attention to the left-hand panel. So these are 3 examples of things we've done in little children, 3 to 5 years of age. Well, try to get a 3-year-old to do anything reliably is a challenge. So, but we're doing a whole bucket of different tests and trying to see which of these are informative so that we can get a run-in and get a sense of what are we going to do once we have an ASO for that child. Okay, and you heard earlier from Dr. Ernst-Nicholas, her approach. This is on the clinical side. So supine to stand, ask a kid to stand up. Sounds easy. A child with ataxia should be difficult. Well, actually not so. They pop right up, the same as controls. So you can see the red and the blue are about the same. Pediatric balance scale. Well, there is a difference there. You can see in the middle panel that they separate a bit. Maybe there's a little bit of decline in our patient in the blue, but not a big change. But that's only over 6 months. So we need a longer observation period. The right-hand panel is actually pretty interesting. 9-hole peg test is something that we do for fine motor skills. And it's validated for children, for adults. It's used for a lot of different conditions. So if you look at this right-hand panel, you can see that from the start at 4.5 years, this little boy took twice as long to complete the test. So it already segregated from typically developing children in the red. But over 6 months, it didn't change really much at all. in him. And then the next 6 months, you can see it jumped up. It went from what, 61 to 80 seconds. So that's suggesting to us that the progression in these patients is not linear. Okay, they're off, and you heard that earlier in the heartfelt story about Angelman. It seems like the child makes some progress to about age 5, and then there's a plateau, and then about age 15, it starts, maybe there's a slow decline. So we need to better understand that as well as we're evaluating these patients. Okay, so there's some challenges here. I'm going to not dwell on this. It's already been discussed earlier, but equitable patient selection is important. How to capture and characterize adverse events. We think we need to do a better job at that. And so do we use measure terms? Do we use CTC, AAE? Let's pretend we're a clinical trial. Let's characterize adverse events before we start the drug so that once we start it, we have some mechanism to do that. We need stopping rules as well, both for safety and potentially for futility. Because at a certain point, let's say we've gone 3 years and we're seeing nothing clinically, we're seeing nothing in our biomarkers. Is that a point where we might say, gee, maybe we're adding risk by repeated lumbar punctures without the prospect of benefit? We don't have an answer to that yet, but those are the kind of questions we're struggling with. So there's a current N of 1 strategy that you're familiar with. It can evolve into a few. There are two different protocol considerations, and I know the next group is going to talk about this, so I'll just give a light dusting. But there is an umbrella protocol opportunity where you have one disease, you have 4 patients, they have 4 different variants, you have to develop 4 different ASOs, but you can do them under one protocol. Common inclusion, exclusion, outcome measures, safety measures. And yes, you're going to have to do things to identify the safety for each of them, but we think there's some commonality there that we can learn from. And this goes back to something that Dr. Woodcock published some 8 years ago, which has sort of prompted this thinking. The second point is, can we be a little clever here and use some adaptive design? So can we take what we learned from the first patient to inform the second patient and the second to the third, et cetera? So with that, I'm going to wrap up. I just want to highlight a slide that was shared from some colleagues in Europe just to say they're thinking about these same things. So what we're doing will impact them. I think that this is a great opportunity to move the field forward, both in the US and globally. With that, I'll stop, and I really appreciate your attention. Thanks.
If the rest of the panel would come up, please. So with us joining on the panel, we have Dr. Shelby Ellenberg in the bright blue, clinical team lead and acting branch chief in the Office of Therapeutic Products and FDA's Center for Biologic Evaluation and Research, and Emily Freilich, director of the Division of Neurology 1 with the Office of New Drugs in CDER, or FDA Center for Drug Evaluation and Research. And joining us online, we have Amy Simon, Chief Medical Officer at Beam Therapeutics. So I want to start off with one question. I'll go to the panelist, and feel free if you want to pick up on what Rebecca or Richard said. But what data is both feasible for clinicians to collect and necessary to assess both benefits and risk? We've been talking about benefit and risk all day, but now that we're at the clinical part of this, what data do we need to assess that that balance?
I can start.
Okay.
Sure.
Emily.
So it's a good question and one we struggle with all the time. As the division director in the Division of Neurology 1 in CDER, I have evaluated the large majority of these single patient N of 1 so-called INDs. And when we were specifically talking about one patient, then the data we're looking for is specific to that patient. How much do we need to know that it will be safe to give this patient? What is the risk benefit? And then how do we know if we should continue dosing this patient, right? ASOs have to be dosed every few months. And at what point do we stop or do we keep going? So you do need some baseline safety and efficacy data collected on the patient. And this can vary depending on the disease we're talking about, the age of the patient, et cetera. But we do want, general safety exam, labs, physical exams, neurologic exams for most of our conditions. And then any biomarkers that are available. We heard earlier that biomarkers can be so important in this space because we don't always have objective endpoints, but are we having target engagement? Are we lowering the protein we think we're lowering? Are we saturating target engagement? Can we go higher in the dose? These are all decisions that we need to make during the course of treatment. And if we have a biomarker, whether it's a PD biomarker or of the protein we're lowering, or a imaging biomarker or a seizure diary that you can capture, those are all important factors into deciding whether to continue to treat that patient. I think that other efficacy measures we already heard. We don't need validated outcome assessments in all of these patients. What we need is rigorous data that can help us know, is the patient getting better or, as we talked about, staying the same in the case of neurodegenerative diseases, right? Are they not progressing anymore? And that can be very telling. So once we get past the first patient and we're talking about 2, 3, 4, maybe we want more data, right? It would be nice to know that there was some benefit in one if you're going to give it to another, and so on. So the more patients you want to treat, the more data we need to support that treatment. I'll pause there. I'm sure there's others.
I was just going to jump in. I think it's a really important topic that we are thinking about constantly at FDA. And I think especially in rare diseases because, the baby KJ case is a great example of a successful treatment of an individual patient. But I think it sets a very high bar if we're expecting every single one to look like that and have very objective data that we can interpret. That's just, in a lot of cases, not the reality of the situation. And so I think we're dealing with very heterogeneous diseases where even if you treat 3 patients, those 3 patients may be very, very different. And so the data that you get from those patients may be very different. And so a phrase we use a lot in CBER, I don't know about CEDAR, but we talk about totality of evidence. So even if something isn't your primary endpoint, and yes, of course, we love to see big treatment effects on primary endpoints, but we are always going to look at the totality of evidence that, for a marketing application specifically, or to, try and figure out, is this doing what we think it's doing? So encouraging you to look at all of those different aspects in these programs. But I think one thing that is very, very challenging for us is in the benefit risk is how important the patient and family perspectives are on that and tolerance for risk. This is something we're constantly trying to understand better because it's going to vary from disease to disease and patient to patient. And we just don't hear enough about what risks patients are willing to take on in potential benefit. So we often hear about successes. Sponsors come to us and say, look at this great case of somebody who improved a lot, but we don't get to hear about the ones who had SAEs. And if they knew that that was going to happen, would they still have chosen to try that therapy? And so I think as much as there can be transparency in the safety side of things, I think that will inform a lot.
Amy, do you want to comment on benefit risk and data needed?
Sure. I mean, I think, again, things change very much depending on if you're doing a large clinical trial or, to this point, an N of 1 and very rare disease. A lot of it will depend on, do you understand the natural history? Do you understand it enough to understand what benefit is, either with a biomarker or changes in halting progression? And I think people have mentioned this, that probably using a patient as their own internal control is probably one of the most powerful things we can do to add to kind of some gaps in data that we may have. And, going to the families for these N of 1 cases and advocacy groups and saying what matters, because as people have indicated earlier today, we may not have a validated PRO. We may not have something that is, but if it's a dramatic difference and someone can now put a peg into a hole and perform on a test that was never appreciated before and could never be done just by chance, I think this is important. I think, this is going to be a partnership with many stakeholders. And I think that the tolerance for benefit and risk is going to depend on both how severe and debilitating the disease is, as people say, depending on the actual patient and their family. And then understanding a little bit more about how to work with the regulators with each unique situation that comes up about what is acceptable, what is possible. So not everyone can be a CHOP, and not everyone can do things that quickly. So what can we build here so that this can go beyond CHOP? Like, can we think about having centers of excellence like a CHOP, and then having clinicians feed into that center of excellence patients that come their way, and then partner with industry if needed to, number one, help with the tools. So maybe we can provide a toolbox, for example, of different editors. Maybe we can provide lipid nanoparticles very readily. So how can we all work together? Because we each add different, I think, strengths to this process. But I'd like to think that it's going to look very different. For example, my base editing trial now in sickle cell patients ex vivo is going to have 50 patients. We never thought that would be the case with no placebo. That's because the results are so dramatic. This is a different trial and the requirements will be different than, let's say, this N of 1. But there are certain things that we all have to understand and that we can't go without, which is understanding the benefit risk from multiple perspectives and then kind of aligning on those. And I almost sometimes a case by case basis now for these N of 1s because things are very different. And then partnering together with the various stakeholders, because I think this is an evolving area now. And I just want to say we're fully on board with it. But I think, again, it's very different if I'm doing a clinical study of 50 patients versus an N of 1. And we kind of have to understand that things change based on that, even down to the manufacturing requirements, et cetera. So again, I look forward, and I think we all look forward to kind of adding the building blocks to this kind of process, which has been going on for years, but I think is now the accelerator button has been hit since we have such precision medicines that are able to kind of uniquely treat diseases, even down to the single base.
Richard, did you want to comment on that, or you look like you wanted to say something?
I'm going to hold it because I imagine there are more questions to come up, and let's get.
So I wanted to get to some of the online questions. As you know, we have a couple thousand people online in addition to everybody in the room. So this one is: improvement in quality of life is often a huge benefit to patients. We sort of heard that earlier about these small incremental things that you may see of someone who couldn't do something and maybe they're a little bit less ataxic, they have better balance, they're able to jump, that sort of thing. How is quality of life viewed in the context of a study endpoint? So I think that's a challenging issue because, you know, what to measure in these things can be very challenging because you don't necessarily know exactly what's going to improve. Maybe the baby KJ example, they know exactly what they're looking at, but in many of these, you may not know exactly the sign or symptom you're looking for in improvement. So how do you approach that?
So do the same guidelines apply here for survival, how the patient functions or feels, or not? I mean, I think to me, yes, quality of life and how the patient feels is incredibly important to capture, but doing so is a bit of a challenge. We don't have established PROs for a lot of these. We have generic ones, the pediatric UL, you know, things that, and we have the clinical global impression of change type yardsticks, which have some validity to them, but is that alone going to be sufficient to be persuasive? So let's say we have a good PRO, everyone agrees, and it improves quality of life, but we're not able to measure it otherwise, either through a biomarker or through our fancy metrics. And Dr. Woodcock has the answer for us.
I'll just add before Dr. Woodcock goes, but I think it's important also to define what we're looking for, like what is the end goal, right? So if we're talking about a marketing approval, then the standards for effectiveness might be different than if we're, what I was saying earlier, just deciding should we continue treatment or should we continue to treat more patients with this? Should we move it into a bigger development program? So quality of life is definitely important in all aspects, but how we measure it can be very challenging, but it is one of those key features of how a patient feels, functions, or survives is the quality of life, getting better. And I think, there are scales like you mentioned, global improvement scales or global severity scales that can be used to kind of capture like a snapshot of how the patient's doing in time. And those can be very helpful to determine if the patient's improving. Now we know there's a lot of, things that go into that. So other aspects that are true in all rare diseases, we also try to incorporate the voice of the patient or caregiver, through interviews throughout the trial, talk to the family before they get the drug. What is the most bothersome symptom? What are the things you're seeing? And then talk to them later and ask the same questions again. And that can be really helpful too. And it kind of comes back to that totality of evidence, but yeah, definitely it's important.
I just quickly wanted to weigh in. I think that it's very important. We often in rare disease trials say, look at it as an exploratory endpoint, but we absolutely want to know. But I think it's really challenging because these are not going to be placebo-controlled trials. So a lot of patients will say that they are getting benefit just out of participating in a trial and having some of that specialized care. So I think that's a really important point here.
I want to get back to a point that was touched on a little bit earlier about dosing. In baby KJ, they gave 3 increasing doses. So the question is, how do you decide when to escalate a dose? And conversely, at what point do you know that a therapy should not be continued? So how do you make that call?
Yeah, I'm happy to take the first crack at that, if that's okay. It's a great question. And again, it's going to be very disease specific, right? There are some diseases where you can measure data that is a little bit more directly related to either target engagement or outcomes than in others. And in all honesty, a big reason why we chose to start with urea cycle disorders in the clinic is because there are things like biomarkers that can help guide some of this decision making, knowing that there's so many other unknowns in terms of how to advance an N-of-1 therapy. And so in the case of KJ, it was not up to me, it was up to an external committee or oversight committee that looked at the totality of evidence, some of which was clinical improvement in terms of stability during illness and biomarkers and things along those lines, and then the risk benefit with if we had seen any safety signals. That risk benefit is not going to be as easy to measure in many of the other rare diseases that I treat clinically, right? Where we're using different measures and the measures over time are very different, whether it be a global impression of change or stability of a neurodegenerative process like we would look for in some of the neuronopathic disorders I take care of. I think you have to tailor that decision process to the indication, which is part of why this is so complicated. I will say one thing that I think is incredibly important that was touched upon a little bit earlier, but I think we need to remember at the forefront of our minds is that we have to learn from every patient. We have to learn about the decision-making that we make around dosing, about adverse events from every patient, but not just the patients we treat. We need to share information about, dosing decisions, about adverse events, about potential signs of early efficacy across programs, because these patients are so precious for so many reasons, but the information coming out of them is really going to inform the entire field. So for this world of ultra rare and a few medications, we really have to share data. And I want to put just one other point out there is that if we don't share the data as from the physician, the scientist, the industry partners side of things, the patients do. Rare disease communities are very tight knit. So oftentimes I hear about adverse events and trials that have not been reported publicly from patients coming into my clinic who heard it on Facebook or on social media because, families support one another. And so I think the information will get out there. And I think it's important that we share what is happening as we build this field so that we can all learn from it, both in terms of dosing decisions, but everything else down the road also.
I just wanted to add to a point you made, Becca, which is this idea about sharing your experience. I mean, right now, a lot of the cell and gene therapy trials have a 15-year commitment to follow-up. And a lot of people, including ourselves, have these separate long-term follow-up studies. I would like to kind of advocate that maybe we put people into registries and it's not so company specific. These decentralized kind of registries where you put all the patients and gather important data over time for durability, for safety, and for other things, where you look at adverse events of special interest, would be less burdensome to the patient and less burdensome to sites. And everyone would gain from that knowledge, to your point. And so how can we now craft these registries with very small patient populations and larger ones like the sickle population and other diseases that we're working on, like alpha-1, to benefit from our collective experiences and decrease the burden on patients with these long-term follow-ups, which are important, but still can be quite burdensome.
I agree.
I just wanted to add from the ASO perspective about dosing. You know, this is something we also struggle with a lot. You know, how do you know that you're at the right dose? And especially with you're only treating 1 or 2 patients, you know, you don't have the collective experience from a phase 1 or 2 trial. So there's, I'll put in the plug Bart mentioned earlier for pharmacodynamic biomarkers. You know, that is really helpful to know target engagement. Are you reaching saturation? Are you having an effect? Is there room to go higher? I mean, that would be ideal. That is also not the case for most of these N of 1 or N of few ASOs that we get. They often don't have biomarkers. So what else can we do? And right now, you know, we recommend monitoring clinically for any obviously adverse events, but also for improvement. And to say, you know, well, if they're improving, do they need a higher dose? But if they're improving, could they improve more? And so, you know, there is this fine balance between trying to find a dose that is they're maybe plateauing or they have made slight improvement, but you still would like to see more. And then there might be a justification for increasing the dose as long as it's being well tolerated. But that's definitely a decision we have, discussion we have with investigators all the time.
I think adaptable study design is important too, especially if you're talking about an N of 1 study. I mean, having very rigid stopping rules might not make sense. You know, having, any grade 3 SAE, you know, we're not going to give more doses. Well, if that's easily manageable, you know, with further immunosuppression and the patient is benefiting, you know, I think you kind of need to be able to adapt some of those things as you go.
I also think as you gain experience with like toolboxes, let's say of an LNP or a delivery device that you're using, now you know kind of clinically and based on your clin pharm experience and tox studies, much more about your, let's say, lipid. And then from that perspective, you know, if you've used that lipid before, you feel much different with like understanding the safety the second, third, and fourth time you're using that lipid. So kind of building on your prior experiences. In the case of lipid, usually Cmax driven as far as side effects, et cetera, acutely. And so I feel like as we get more and more things in the toolbox and can kind of use those cross program, that will help not start back at the very beginning every time.
And then what about the flip side of that question? When do you decide not to continue a therapy? Shelby, you touched on that just a little bit. But what are some of those things that you look for, signs to say, okay, well, we haven't seen any effect, now it's time to stop?
I think futility is a really important thing. We talk about stopping rules for safety, but you know, having some criteria for futility as well. And this is where I think having some of those independent data monitoring committees can help. So not just for safety, but I think for some of, you know, having some of those criteria that they're looking at as well from a futility standpoint, because then you're just giving them something that's not helping and increasing the risk of cumulative exposure.
And I will say, I agree. I think having those oversight committees to think through futility is really important because as the physician sponsor or just the physician or investigator taking care of these patients, having that discussion with the family around stopping a therapy is very challenging. And it is very helpful for them to know that outside experts have looked at this data, have thought through this, so that they understand that they are not taking something away from their child that people are recommending that they give. Because in the end, you really have to make sure you're supporting the families through all this, whether either the patient themselves or the caregivers. And so having more objective data or at least opinions on this, I think is really important and helps the families.
I just want to thank everybody. I think that's a great place. We're out of time. Just want to say that despite the technical issues with Dr. Rebecca Ahrens-Nicholas's slides, they will be available online. You'll be able to see all the slides there. So thanks to all the panelists and speakers.
been a great day of discussion so far. My name is Brian Cantor. I'm an assistant research director here at the Duke Margolis Institute for Health Policy. Excited to be here and looking forward to introducing our next presenters and panel. And we're going to get to a little bit of the nuts and bolts of clinical trial design. But first, we're going to have a few presentations to level set a little bit more for the audience. And first, we'll hear from Klaus Romero, the Chief Executive Officer and Chief Science Officer at the Critical Path Institute, or CPAth. So Klaus, please take it away.
Hey, thanks everybody. Right. Yeah, no, this is great. Thanks to the Duke Margolis team for putting it together, and Garrett and team, by the way. Love the choice of colors. You'll see why. That's disclaimer number one. Disclaimer number two is that we're here for the living experience that we've heard about throughout the entire day. Those of you who know me know that I wear my Critical Path Institute pin very proudly, but I switched it today to wear the Wheelers Warriors pin that was gifted to me today. But this is all about Wheeler. This is about Mila. This is about the countless people that we can help with what we're discussing today. Okay, so what is a Critical Path Institute? We were established 20 years ago thanks to the vision of Janet Woodcock. When FDA put out the Critical Path Initiative document, I encourage everyone to read the original version of that document. That in 2004, the FDA came out to the world and they said, look, we have the mission to regulate the products. Industry has the mission to develop the products, whose mission is it to make the process efficient. Can't be just FDA, can't be individual companies. Why don't we set up public-private partnerships to work together on fixing the process for everyone, making it efficient for everybody? So that's what we do. We set up collaborations that include, in a neutral environment, with the legal infrastructure, the scientific infrastructure, and the regulatory infrastructure, a neutral environment for FDA, global regulators as well, industry, the living experience voice, and our academic colleagues to come together and share information that would be very hard to share otherwise, and use that information to turn that into solutions that can remove bottlenecks from the process. We never do anything alone. Everything that we do is through the power of collaborations. And we're in front of what could become one of the most transformative collaborations of our lifetime that can bring drug development, as we've discussed all day. This is different, but this is modern. This is the material demonstration of what personalized medicine could truly be. So this is why we come together, because no entity can do all the science that is going to be needed to transform the process as a single entity. But we cover, gosh, what, 50 or 60 therapeutic areas? How can we do that when we're only 160 people strong organization? That's because we've always concentrated on 5 core competencies. Everything data, everything advanced analytics, everything biomarkers, everything outcomes, and regulatory science. Those 5 core competencies are very portable, and we're putting them on the table to the benefit of what we're discussing today in terms of transformations. But the proof is in the pudding. What have we been up to over 20 years to really transform some really challenging problem children areas in drug development? So through the power of generating true insights from the data that we've been able to integrate, now we have disease-modifying drugs for Alzheimer's disease. We have the first new drug and the first new drug regimen for tuberculosis in over 60 years, and a healthy pipeline of more than 60 drug combinations being developed today for a disease that is completely neglected. or is to be no longer neglected because of all that development. The first disease-modifying drug for polycystic kidney disease, based on the transformation of how people think about trial design, selection of patients, and tracking their progression. The first type 1 diabetes prevention medication, thanks to a transformation through solutions that were based on data. Labeling expansions for symptomatic medications. Yes, we are about to transform the trajectory of diseases, but we should never forget about the symptomatic benefits. The first disease-modifying drug for Friedreich's ataxia, I'm going to dive into that one a little bit deeper. And then the first non-steroidal option for all variants of Duchenne muscular dystrophy. So Parent Project MD, I know you're in the room. Kudos for the way you've jumped into the collaboration that you've helped us set up. Okay, so it's all about data, but what about the data? It's all about transforming data into actionable knowledge. First, it's all about the data that are out there, but we have a process through a data platform that can make the data speak the same language and turn that data first into information, then into insights and into knowledge that can be transformative. Through the trust that we have been able to build with our industry partners, we have a data platform that integrates 722,000 individual records across clinical trials from pharma. That includes control arms and active treatment arms, observational studies from academia, patient registries from patient groups, and electronic health records. But we're not a data vendor. This is not about integrating data for the sake of integrating data. This is about integrating data for the sake of generating solutions based on the data. So you can, my undergrad is in biophysics, so I'll just nerd out on physics for a moment, not putting Michelle to sleep, which is the cardinal rule of every time I nerd out on data science and advanced analytics. You can think of data as the mass, but you can transform mass into energy. To take the mass and transform it into energy, you need a process, and that's the process that our data platform provides. But what's the point of transforming mass into energy? So that you can generate power, which is the transfer energy per unit time. We want power. We want that energy to be transformed into movement that can actually transform not just the process, but ultimately the life of people in need. And in doing so, we can materialize the true vision of personalized medicine. Okay, so let me share a few details of the example of Friedreich's ataxia. So we went from mass, the data. We have the largest integrated database for Friedreich's ataxia on the planet, thanks to the collaboration from FARA. Ron Bartik was a champion of this. We put the data through the transformation process of energy to mass, and then we generated what was actual power. The sponsor ran a trial for the first, what was hoped to be the first disease-modifying drug for Friedreich's ataxia. And the trial was statistically positive. But there was always the question, does that evidence of statistical differences demonstrate evidence of clinical benefit? In a different world, in a different time, the answer could have been run another trial. In a devastating rare disease where the patients that could be enrolled in a trial were already enrolled in the trial. Ethical cost considerations, all manner of issues. But the FDA was visionary enough to set up the funding for us to be able to set up the data analytics platform in the first place. And because the FDA knew of the existence of the data platform and the sponsor knew of the existence of the data platform, they said, why don't you go into the data and extract some external controls through a propensity matching score and then plug the data into the totality of evidence that you're showing us, and let's have the conversation again. The error bars shrunk, and that led to the full approval of the first disease-modifying drug for Friedreich's ataxia. Okay, fantastic. But this is different. We're talking about going even more modern, going even more transformational. And we're looking at a collaboration that could be the place where we go from, N of one or N of few to millions, because this will not just be about one individual one time or 7 individuals one time, but a continuous approach to learn and confirm and get help to the people that actually need it in a regulatory grade way, in a scientifically rigorous way. All the components that you see there are aspects of what we have envisioned as key components of what a collaborative effort could look like that helps FDA's vision become material, that helps the voice of the living experience have a meaningful conduit, that helps our academic colleagues have a trusted process, and that helps our pharmaceutical industry partners have certainty for what they're getting into so that they have the confidence to continuously invest in the development of these therapies. And here's a dream. How about that for a timeline where we aim to, in no more than 3 years, have the key aspects of the transformative process set up based on data, based on rigorous science, based on collaboration, for every one of us to have a new process at our fingertips that we can use to materialize the vision for personalized medicine. So with that, I'll stop, but I'll break another cardinal rule from my mentor to never show equations in a presentation. But work with me. Success is a function of time and people. So people, thanks for taking the time to be here today and ensuring that we will have a successful outcome and have a process that actually transforms medicine and healthcare in the 21st century. Thank you.
Thanks so much, Klaus. When I was looking through the slides, I was trying to work through what all those different letters stood for, and you made it clear there. And I think also, the point about the proof is in the pudding is true, and it's also important to remember that the expression continues with the proof is in the pudding and the eating. With that, we'll hear next from Marshall Summer, the Chief Executive Officer at Uncommon Cures.
So good afternoon. It's actually great to be here. My name is Marshall Summer. I'm a clinical and biochemical geneticist. I was in academic medical practice for around 40 years before venturing off into the great unknown of doing clinical trials full time. Somewhere along the way, I had the privilege of seeing over 32,000 patients with different genetic rare diseases. I was part of over 200 clinical trials, primarily in rare diseases, whether as PON or PI, and hopefully learned something along the way. But I think, like so many of us here, I've also been greatly influenced by knowing Dr. Janet Woodcock, who has taught us so many things and really has paved the way for a lot of that. So Janet, thank you for this. And I'm also going to show some data we've been working on. So just a heads up there. So this first slide is actually, okay, how many rare diseases are there? It's a great question. Current estimate is there's around 16,000. And it's growing every single day from that. And that's based on new discoveries from a lot of the sequencing companies, old data, things like that. So it's gone from a kind of a ivory tower. We drag people from the laboratory specialty to an actual field where there are so many different conditions, so many different diseases that we have to actually think about it. And these are very impactful on our healthcare system. These are patients who use a lot of medical resources. Finding treatments for them actually can have significant impact on the economics, plus the fact that these patients need treatments and they need therapies. Now, here's an interesting one. This you haven't seen before, unless you've seen me talk recently. We went through and looked at prevalence data for rare diseases. Now, I'm not including cancer in this. I'm kind of including your classic definition of a rare disease. And here's what we found. There's probably only about 200 conditions that have a few thousand patients. About 90% of rare disease have fewer than 150 patients. They're very small numbers. About 80% probably have fewer than 50 patients in the United States. So I'm basing this mostly on US data and EU. So when you start thinking about your classic models, of rare disease clinical trials, you suddenly find that for a lot of the new diseases that have been described, a lot of the existing conditions, the patients just don't simply exist to fill some of the old statistical models and the old study design models. So we really do have to look at new models just to do good science. So if we look at approvals, obviously FDA for the last 5 years has been tracking over 50% approvals per year for orphan. A lot of those are for oncology products. EMA is starting to catch up. I don't think that's tremendous news to anyone. The success rates are interesting, though. When you look at orphan disease, the success rate tends to average out, and there's about 3 studies that have looked at this, and we went back and dug into clinicaltrials.gov and saw the same thing. About 75% of rare disease trials fail. The average from phase 1 to 4, not just in any particular phase. That's actually better than most, but while that sounds encouraging, you have to then take into account that for a lot of these disease conditions, they get one shot on goal. It takes tremendous resources to be pulled together to try to do one of these trials. GLP-1s are great, but there's a lot of them out there already. If one of them doesn't make it, the world is not a lesser place. Same thing for statins, things like that. But for a lot of these families, they get that big one chance. And a 75% failure rate is just simply not an acceptable outcome from that standpoint. Now, why do they fail? It's not science usually. Science is maybe 15 to 20% of the time why these things get withdrawn. If you actually look at it, the most common cause of failure is recruitment and retention, which comes back to study design, which comes back to the models, which comes back to the burden of the study itself. So one of the things we have not done as good a job as I think we can is actually taking into account putting the patient at the center of trial design and the realities of the patient community. We all have a golden ideal of what a clinical trial should look like, but that's designed around a model and based on statistics that simply don't play in the world of rare disease and never really will. We have probably thrown away a number of good drugs simply because we have tried to fit them into trial designs that are not well designed and they're not ever going to work. So, like I said, when recruitment and retention are your biggest failure rates, it means you're not doing something right. So what's different about rare disease? Well, they're rare. I mean, that's kind of the starting point. And as Chris Austin used to say, we have a PR problem because the second you say rare, most people on the Hill's eyes glaze over and it's like, well, I don't need to worry about that. But when there's 16,000 of them, you got to start to think about it. The other thing we don't pay attention to as much is genetic heterogeneity. In a classic clinical trial, you want to have your patients selected so they all look as much alike as possible so that your control arm looks like your treatment arm, and that is just simply impossible in rare disease. In the urea cycle world, which I work with, which the talk earlier was fun because CPS1 was the gene I actually sequenced back in the 90s. So that actually was kind of, yeah, I know, it's kind of fun seeing that come full circle. But when you think about genetic heterogeneity in urea cycle, I can have patients with the same gene defect, maybe not the same sequence, present at 6 hours of age or 80 years of age. So there's enormous variation in how the patients present, their clinical course and everything. So you really have to start thinking about that patient's clinical journey as that individualized to that patient. So start thinking about the concept of patient as their own control, and we're going to come back to that. There's very sparse natural history and outcome. It's getting a lot better. CPAT's done some great things. NORD I Am Rare program, which Janet helped fund from FDA, and I worked on when I was NORD's board chair. There's a lot of things out there, but we're just scratching the surface. And then can you get good patients matched to that serve as a control arm? And the answer is pretty well, not perfect. Challenging patient identification. These patients sometimes have clusters, but by and large tend to sort of randomly pop up. And so if you're using the old model where you define where your centers are going to be, unless you're right next to an academic medical center that happens to be one of the sites for the study, you're probably not going to get to participate. So we almost have to flip that model around too. And do you look at opening centers where you actually have identified patients to come into studies? And then effect size and frequency, and we'll come back to that. So basically, the one-size-fits-all model of the rare placebo-controlled randomized clinical trial really just doesn't fit well. You don't have the patients, you don't have the model. Yeah, I mean, I like cartoons. Sorry. I'm from the South. We're simple-minded folk. But we're using a design that just doesn't fit the target we're trying to hit. All right, definitions you can use. Brian asked me to include some definitions. I will not read through all these, but umbrella trials, basket trials, platform designations, adaptive trial. I am going to stop on adaptive trial. One of the things we do not build into our clinical trial design enough is an adaptive pause. Because when we come up with power calculations and study designs, let's be blunt, we're guessing a lot of the time. We're thinking that might be what would work. And maybe we're right, maybe we're not. But if you don't build in a pause saying, hey, we've done a little bit of work. Were those calculations right? Does it look like it's heading the right way? Or does it look like it's not going to work at all and we could stop now? You're just kind of wasting some time, and you're probably wasting another failed trial as well too. So I really like the concept of putting adaptive trial, master protocols, all of these things. I think the slides are available if you want them, and then you can read these at leisure. Oop. Okay, I got it to go back. So this is a concept of looking at like diseases. So urea cycle is a great example. There's 7 kind of urea cycles, 5 primary enzyme deficiencies. I worked on this since 1988, so I actually know all the enzyme names. I can bore you with that later. But why can't you use those like diseases to generate data around treatments? We've actually done that when we were working on the oral medication for treating hyperammonemia in these patients. We actually were able to use data across 5 different diseases. So you can go back up the tree branch and find like conditions that then you can use to kind of group together or use information across your study. Or let's say something worked there, the molecular pathophysiology is similar. Why can't you pull those things together? Okay, man, this thing's sensitive. There we go. Okay, so there's kind of 3 overarching trial design styles I'd like to talk about quickly. And there are variations on all of these and sub-variations ad endless nauseam. First one is the RCT, double-blind placebo. That's kind of what everyone would like to steer you towards. And it's got a lot of power to it. I think it's great if you are doing agricultural statistics or large patient-based statistics, which is where a lot of the p-values and things come from. By the way, the first randomized clinical trial, as best I can tell, was done in the 1700s in France to study animal mesmerism. I don't know what animal mesmerism is, but evidently that was what it was used for. The next is, I'm going to skip to number 3, which is natural history control model. You try to collect enough data on patients with the same disease so that you have enough history where you can either pair them up or use them as a group to use that as a match control. The statistical power of that is heavily dependent on how much those patients represent your patients that you're going to treat. And as we said before, genetic heterogeneity, particularly as the group size gets smaller and smaller, can really work against you there. But it's definitely more powerful than an RCT. And the third one, and my favorite one, is patient is own control. Some people used to call this serial N of 1, which sounded kind of ominous and like something not the right way of doing it. So we've started using the term patient is own control. And that's where you collect data on the patient. You know how they're performing with their disease. You then expose them to the treatment, see how they respond or don't to that. And I know there's all kinds of issues around placebo effects and things like that. You got to be careful and build that stuff in. But that way you can start to account for genetic heterogeneity. That patient's slope of their response to the disease is personalized to them. And then you can then compare it across patients. And it's very powerful. And that's what the next slide shows. So we ran modeling. And this is a project Janet and I are working on here that I still owe you the manuscript on, and it is coming soon. She can kick me later. Anyway, if we look at the power calculations for different effect sizes, effect size is how much you move the needle for that patient. 0.5 means you got about a 50% change. I know that's a little naive way of putting it, but we'll put it that way. And then our x-axis is a log scale of how many patients you need. So to get a 50% effect size, which is a robust impact, you need about 160 patients in a randomized clinical trial. For a patient-matched natural history study, you probably are going to need somewhere around 50, 60, somewhere in that range there. You need about 10 to 15 in a patient is own control model. You actually need less than that, but if you're going to correct for edge effects and those variations you get, you actually need a lot fewer patients. So it's very parsimonious of the patients you need. We've seen positive signal in studies with as few as 3 to 4 patients. So when you're looking at design for a rare disease clinical trial, this in some ways is scientifically the most valid model because you could actually build in a duplication cohort here. You know, if you've got 20 patients, you break them into 2 groups, analyze them separately, you can actually replicate your statistics as opposed to trying to find enough somehow to cram into an RCT. So I really do think this is the model we need to start steering towards. In rare disease, for our discussions earlier around the N of ones or grouping together N of ones, it also allows for a lot of variation from patient to patient. So that's kind of my main thesis today is this particular slide here. The last one, I'm going to channel Ron Bartek because I get to work with Ron a lot on the Pediatric Inclusion Alliance, which is we're also missing science and we're missing effect by not including children and pediatric patients in research from the start. And why? What's the point of maximal impact for a disease, particularly a rare genetic condition? The majority of those conditions are pediatric in nature. The biggest impact is had early in the patient's life. If we wait until, I hate to use the term, the cake is baked, you may see no change at all. And you're going to throw away a otherwise functional drug because you didn't study it at the relevant time for that patient. Also, assuming that because you've tested a drug in adults, it's safe in kids has also been shown to be erroneous in numerous examples. And vice versa, showing it's safe in kids doesn't make it safe in adults either. So including the pediatric patients early, we've always had a fear of that. Nobody wants to hurt a kid. I'm a pediatrician originally by training. I don't want to hurt kids, but I also don't want to watch them die while I'm trying to do something in adults while we're waiting to see what's going on. So I think that's probably enough. Thank you very much.
Thank you, Marshall. Really excellent presentation. And I will invite the rest of the panel to join Marshall and Klaus up on the stage, and we'll introduce them as they make their way to their seats. So joining us will be Ashley Munchell, a clinical reviewer in the benign hematology branch of CBER at FDA. Also joining us from FDA will be Michael Pakinowski, the director of the Division of Translational and Precision Medicine in FDA's Office of Clinical Pharmacology at CDER. And then another Michael Panzera, Chief Medical Officer at Neurovati Neurosciences and Grin Therapeutics. And rounding off the panel will be Charlene Sugribi, the Chief Executive Officer at Global Genes. So I should be able to walk seamlessly. It's really amazing how this technology works. I did always want to be an actor, and I, on the Duke Margolis team, happen to, I think, have the least amount of musical experience, but we are in the green with the big clock showing in front of me, so we must be doing something right for this session. I think we have a lot to talk through here, and also want to say that we are going to prioritize questions that have come in from the audience that have come into this iPad that the team in the back will shower me with booze if I do not look at. And so I think just want to make sure that just in terms of to get started, kind of, we talked a little bit about sort of natural history studies and having a well-characterized nature of the disease for treating some of the therapies. And Charlene, wanted to kind of come to you first and the work at Global Genes to kind of see what goes into characterizing a natural history study and sort of what indications that it's been most applicable for the work that you've been doing.
Thank you. And thank you for having me today. It's been a really, really insightful day and excited to be included. So I'm Charlene Sugribi, and I'm the CEO of Global Genes, and I'm coming to this kind of with 2 hats. So I spent my career building big data solutions. I don't know if we call it big data anymore, but data solutions, we do. Okay, good. For both in genomics and healthcare, as well as enterprise software. But I got my introduction to rare disease because I have a child who has a rare neurodevelopmental disorder called STXBP1. And that really changed my life of going from this technologist to suddenly becoming part of a patient family. And it became very personal to me about how important it is to get to solutions for rare disease patients. And so I actually left the for-profit world a few years ago and moved to Global Genes to accelerate the work that we had started in my daughter's disorder around how do you build assets to really advance work in these very rare disorders. And so one of the programs that we have is called RareX, and it is a registry that enables collection of data across rare diseases. And I felt very passionate about this concept because, as Marshall said, we now have 16,000 rare diseases. A few years ago, RareX, as well as the Monarch Initiative, did a count, and it was a little over 10,000. And so we are really in this age where there is, such a curve to these new disorders that we're finding. And we don't have the ability to characterize the disease before we start to collect data. We have to be able to collect data immediately. And so the concept behind RareX was to work in collaboration with individual patient advocacy organizations to collect robust data, because as you know, many patient advocacy groups, they didn't come from a background like most people in this room. They don't have PhDs, they haven't run a study, they haven't necessarily built, data collection, type of measures. And so the idea was to provide that kind of robust background, foundation, and then also to be able to collect in collaboration with patient communities because they're the ones who have the connections with patients. So we have focused very much on ultra-rare diseases since that's the area that is highly underserved in these emerging diseases. We now have almost 100 diseases that are collecting data on the platform, and that's in almost 100 countries. So we are GDPR compliant. As you know, rare diseases know no borders. And the thing that I am excited about is that we have been able to start to build what might be some of the largest groups of patients in these ultra-rare diseases, both for characterization of those diseases, but also to identify patients. Because as I think we're in an extremely exciting time in terms of innovation. Of course, as a parent, I don't think things are moving fast enough. I will tell you, my daughter was diagnosed when she was 3.5, and she is 12 now. I naively thought that we would have a therapy for her in 5 years. And we have made a ton of progress in my daughter's disorder. We are not there. And things move so slowly, but even just in the last 3 years, I have been so excited about the technology innovation that is happening and the opportunity for it to impact many rare disease patients. So we have to go and identify those patients, as you were saying, Marshall, so that we have the ability to, have them ready for these trials.
Yeah, thank you for that, Charlene. And Michael Panzera, I want to bring you in next. You're all the way down there. I can almost hardly see you from over here, but want to see on that theme of trying to identify patients, and know you're doing a lot of good work in the neurological disease space. But be interested to hear sort of how you all prioritize trying to identify patients to target therapeutics and development of interventions.
Oh, sure. So I'm Mike Panzera. I'm Chief Medical Officer of Nervati and Grin. About 25 years of doing, I'm a neurologist, about 25 years doing drug development experience. I've gone from large diseases to small now, so down into category of ultra rare in my last company and this company. Well, it's been this particular Grin Therapeutics, the company that we are working with now and we've created, is a good example of a disease just recently described not that long ago, a disease where we know very little about some of the things we've talked about, the natural history, the locations of the patient, the diagnostic journey. It's not even known how many of these patients come to be diagnosed. And the variants themselves, it's a mutation in the GRIN gene, which causes changes in the NMDA receptor, which is important for homeostatic balance in the CNS. There are gain of function variants, there's loss of function variants, there's the way those are characterized, which is new. So it's a completely new disease. So as you're getting into a new disease, you realize, again, you know very little about that disease. And right now, we know of probably about 1,000 patients in the known registries of GRIN, but there's likely more. So we have a drug that looks very promising, and now we have to do a phase 3 study. So how do you find enough patients to generate that objective evidence we heard so very much about all day? What do you use to assess objective benefit? And in our disease, you have about half of the patients who have intractable seizures. Well, that's something that is good precedent on how to measure seizures. However, about half of the patients don't have seizures, but they have severe neurodevelopmental signs and symptoms, severe dysregulated behaviors, which often are the things that the families say they need the most help with, and there are no real tools to be able to do that. Our approach was to engage early with our colleagues at FDA and come up with ways that we can figure out how to do a study that enables us to assess these things, which always ends up being a little larger than you want it to be. So again, you have to find patients. So back to your original question, engaging the community. Obviously, the families know where they are, and often they aggregate around certain advocacy groups, and families, as you said, form these groups without knowing how to form them or how to do a trial or all these things, but they're out there waiting. And you engage those groups, and you also have to say, okay, well, where do they see their doctors? Often it takes years to see a doctor. So, okay, well, how do we find them? How do we help them see a doctor so that they can find their way to our study? And we have partnerships with genetic testing companies, so we can understand where the testing's being performed, and then work with them to have them reach out to say, hey, there's a trial that might be applicable for you. There are companies of genetic counselors that you can actually set up to have when you find a patient using one of the genetic testing companies, you can send them a letter saying, hey, here's a genetic counselor that you might want to meet with, and then that genetic counselor assesses them and sends them to an appropriate physician. So there's all these techniques that we're now learning to deploy, starting from number one, engaging the community, and then providing the services that enable access. But number one, I think of everything, which was mentioned again earlier today, is if you're going to design a trial and people living with a disease and you don't know anything about the disease, you have to ask them what we're trying to fix. What is the most meaningful thing to you, and how can we measure that? And so we've had to develop specific clinical assessment tools using existing tools. And this is, again, in working with FDA. There are lots of tools out there, but how they work in your disease is unknown. So you have to try and then collect that information, and then, first of all, figure out what's most important to families, figure out which tools might be able to measure those things, and then work on something in the trial that can specifically assess those tools and or those symptoms and what might be clinically meaningful. Now, with all that, you can see it's kind of complicated, and it takes an investment. And this gets to, again, Marshall's presentation about it takes commitment, effort, wanting to do this, and all of it takes investment money, and finding investors who are willing to give it a go.
A lot of thoughtful engagement, and I think Ashley and Michael Pakanowski, that thoughtful engagement can continue between conversations with sponsors and FDA, and maybe you both can kind of share from your own experiences and work, sort of what have some of been kind of the some best practices or some opportunities for thoughtful engagement, really, when it comes to, and just paraphrasing a little bit from a question from the audience, using some of that prior knowledge that was talked about earlier, the preclinical data, some considerations for the risk benefit for patients, and needing to shorten the time to develop some of these treatments because the patients can't make it to get a transplant.
Sure. I mean, so hi, I'm Ashley Munchel. As you mentioned, I am one of the reviewers in the benign hematology branch of CBER. Specifically, as you all are talking, I'm thinking a lot more about kind of the natural history component of this, because I think that's going to be really important as, we're going down the paths of commercialization and licensure, because we need something to compare the patients to. And, it's really exciting for me to hear about all the work you all are doing to help create and maintain these databases, because that is a huge feat in and of itself, right? I mean, that takes a lot of time, money, energy, people, manpower to do that. And it's going to be the most helpful thing that we have when we are trying to really make decisions about how beneficial these products are. I think in that context, the most important thing about the natural history data really, I think, kind of mirrors what we want in clinical trials, which is, rigorous data collected in a standardized fashion. So that way we really can look at, what is happening in patients that are not treated and comparing it to, what is happening in patients that are treated. And so it has to be just as methodical almost as what happens in the clinical trial setting. And that gives us the most useful information, right? I mean, we've had people propose, us looking at registry data over 20 years, which sounds great until you really look at it and you see, there's a patient that was followed every 6 months, and there was other patients that were seen 8 or 9 years apart. And so you don't know if some deleterious thing happens when that started, right? And that's the key to these natural history data, is being able to try to tease down when a deleterious effect started, so that way we know that is something we need to monitor for and try to either stop from happening. I think the natural history data also is helpful, not just to help develop clinical endpoints in trials, but within the rigorous data collection, collecting things that you think might be useful biomarkers would be really helpful. Because it allows us to use that potentially as a surrogate endpoint, or even makes us feel more comfortable about treating patients that are pre-symptomatic or minimally symptomatic, especially in these diseases that are widely heterogeneous. So if you can find a marker that predicts that patients that have this are the ones that have a more severe phenotype, then you feel much more comfortable about enrolling that patient into a clinical trial earlier in the disease process, which we all know is beneficial for all of these diseases. Because it becomes hard when there's a lot of heterogeneity, to know, we think a lot about, are we giving a product to a patient that may have never had a lot of symptoms and may have been minimally symptomatic? So therefore, in this particular case, we expose them to risks that were different than somebody that we knew was going to go down a rapid path. And so that would be very helpful, I think, as far as moving that forward too, is allowing us to feel more comfortable at using this earlier and earlier in different phases of the disease.
Yeah, Marcia, I'm going to let you go in 1 second, but Mike, anything to add there?
Sure. I'll just say that I'm a clinical pharmacologist. I've been, working at FDA for 15 plus years on personalized medicines, and a lot of challenges that come up in dealing with small patient populations, and we have to rely on mechanistic evidence and things like biomarkers. So I would always say, for any natural history study or any investigation of any sort, more data is better. We have to cherish, I think, the patients that are coming into these studies and exposing themselves to risk, and try to get as much information as possible out of them to help kind of support the development of future products if needed. There's a couple different situations we might be talking about here. For an N of 1, let's say, or a few patients, You may have a patient with a specific genetic variant in a rare disease that's much more common, and you may have that natural history data. You may understand the relationship between biomarkers and clinical outcomes, and that is a much more straightforward path. I think the reality is for the vast majority of rare diseases that we see, including those that are coming in, as these individual patients, we know very little about the disease trajectory, and it's very complicated to try to draw out parameters that we can make comparisons to evaluate whether or not the drug is having an effect. Even when we have more common conditions, it's very hard to tease out whether a drug is benefiting a patient, even in clinical trials that are conventional. A lot of our neurobehavioral assessments are very blunt. They don't change over the course of a 1 or 2 year period in some of these conditions. So it does become very challenging. So, I think to that extent, it is very important to look at the pre-post treatment course. This has been brought up several times today, that crossover approach in a lot of the conditions that we're talking about today is really critical, just because I don't think we'll have the natural history most of the time.
Yeah, I appreciate the stratification there, Mike. Marshall, I promised you would get a chance.
No, that's okay. Actually, building on what you guys were just saying too, one of the problems in rare disease is actually getting what we like to call evidence-based medicine. It ends up sometimes being more what we would call eminence-based. But did a little experiment, and try this one if you have a rare disease that's your favorite that's been described in the last 5 years. Look at the first publication on that. It's usually kind of an overall description of what they found, then shows the mutation. Then pull the next 5 publications. Almost always what you'll find is the trend is those were the exceptions to the original presentation, because that's all you can get published. And so when you start going to the literature and start trying to pull these evidence-based groups, you'll find it in the field of rare disease. That ends up being a real weakness. I'd recommend all of you go take a look at a program called RareCap. It's run out of Vanderbilt University. We used to run it out of Children's, but it's sort of like a clinical experience collected around these rare diseases with treatment stuff. And it's an iterative program, but oops, not I was saying, but coming back to the natural history study and the economics of that, those can be very expensive to run. And that's why some of these programs, like the ones from Global Genes, the ones from Nord, and some of the others, I think, are providing some more cost-effective. But when the numbers get very small and the patient and the genetic heterogeneity is so large, it's really hard to come up with that aggregate ideal patient. So you still have to think of those patients as individuals with a like disease.
Yeah, that's a very insightful set of comments, because we're about to just set up the evidentiary considerations for the 21st century. And we cannot forget about the notion of the challenges that you presented in your slides about a very simple opportunity that sometimes gets ignored, and that's patient identification and selection for these types of evidence generation exercises. In the PKD example, so polycystic kidney disease, orphan status designation, yeah, not ultra rare, not by any long shot, but when that community was struggling and they came to us, thanks to the FDA saying, hey, you need to go talk to these guys because you have data and you can turn those data into movement. They came to us with the following ask: we need a surrogate. The answer to all our problems is going to be on a surrogate. And we looked at the data and at the disease, and sure, yes, that made a lot of sense. But even though they had quite a bit of data, 3 registries and 1 observational study, that's where we started. Is at first, let's make sure that we can meaningfully identify and put patients that are truly more likely to benefit from the new therapies in the trials. So that's what we did. And we built, yeah, sure, a longitudinal model that had a lot of pharmacometrics to Mike's ClinPharm background in it. But it was all about getting total kidney volume as the metric to better select patients for trials. Then the company that embraced that use of total kidney volume to select patients, they ran the trial was successful. And the FDA took the same model we had developed with the code that we had submitted, and they plugged in the data from that clinical trial, and voila, it was declared a surrogate. So that's how you can progress, moving the needle with seemingly baby steps, but each baby step has a very, very important effect on the whole equation. But what's important now is that we have data that shows that when companies use total kidney volume to select patients, not even the surrogate part, they stand to save $70 million in development costs. What was one of the counters that we heard? Well, but it's imaging and you're making it harder to recruit the patients and that's costly. Well, now the proof is in the pudding, $70 million worth of savings against the notion of making the trial fail because you don't put in the right people in the equation. Now with the power of getting as many data points as we can from all the ASOs that exist on the planet, the successful ones and the not successful ones, we can start putting these kinds of insights in front of everyone, and we will all learn equally. Same playing field for the academic drug developers, the startup companies, and big pharma. Because we cannot forget about the fact that big pharma does their calculations. They have to do that. Absolutely understood. And if they see that through these kinds of collaborations, they can handle uncertainty and that they will have clarity on the evidentiary considerations, I guarantee you that they'll start investing heavily into this. And that's where we want to get to. So I'll leave it at that.
No, it's very, very helpful to share that success story, Klaus. And I think something else. Marshall, I did make you include some of those terms in the presentation, but I do think there are a whole host of statistical methods and trial designs, and sometimes implementation of those is a lot easier than just what's written down on paper. So kind of when it is particularly difficult to treat patients with heterogeneity in terms of their mutations or in how the phenotypes manifest their disease conditions, how do you go about kind of trying to apply sort of these innovative approaches, thinking in sort of a broad holistic part?
I think one thing to start with is don't think of this as a difficulty. I think one of our mindsets is we're making rare disease appear difficult to work with because we're trying to fit it into a model it is not designed. Oh, thank you, Shannon. It is not designed to fit into. Instead, think about this is almost like a completely different field of clinical trial science, where you have to think of how do we group things together when you have so few patients. How do you actually look? How many branches up the tree? And I'll give Brian credit. He asked me to include the tree one in there for that.
I wasn't going to say it, but thank you.
Kudos, man. Kudos. But how far up that tree branch can you go? Things are still similar enough where the data is translatable. And whether it's N of 1, N of 10, N of 20, N of 30, you're still in that statistical zone, and you're still in that zone where trying to use the classic models, just you're not going to get anywhere. And to be honest, the science isn't as good. So there's a whole thing we need to start doing, one of which is some of us were talking about earlier, is we need to develop the science of clinical outcome measures where we're doing that well before we ever get even close to a clinical trial. And that actually needs to be a robust science where you're testing these things across multiple diseases, multiple modalities, and by collecting that data early on the patients who are affected with those diseases, if a trial comes along, you've already got your baseline control data to move forward with that. And you got to do that working with regulatory agencies. You got to do it working with the patient community, because let's be honest, that's where most of it's going to come from. And I think if we can do that, we're going to see really a whole different field here. So, but I would get away from the it's hard to do as it's different to do.
And I can kind of comment on that. Being in the gene editing space, I think this is really an exciting opportunity because it is in its infancy, right? We're not trying to take a problem and fix it. We don't have a problem yet. So we need to build up, what we have learned from rare diseases and other, treatments. And obviously workshops like this help that, this dialogue helps that, the interactions, between the FDA and the developers help that. obviously that's a big priority of the FDA, and these programs that, are getting put into place with rare disease that hopefully will help facilitate easier communication across, between the FDA. But I do think this is a really, exciting opportunity. And I want to give a lot of credit to the team at CHOP and Dr. Ahrens Nicholas when they were, developing this therapy for baby KJ. From the start, their thought process was like, we want to develop something that goes beyond him. And so that I thought was just really insightful, really ingenious, and the fact that that was already on their mind, even though they were working against the clock for a single patient, they were already thinking about if this is a success, what can we sort of put in place so that we can expedite this program and other programs like it very quickly.
I just want to add that.
Okay.
You can go right down the line.
I just want to add that I think it's actually a false dichotomy to talk about N of 1 and treat it like a completely different class from looking at N of small and ultra rare diseases. And this goes back to, I think, the point about the trees and the branch. And that's a concept that we implemented with RareX, with this idea that if you understand related diseases, that will help you to address the individual diseases. Right now we're very heavily concentrated in especially neurodevelopmental diseases. But I think that thinking about how we can, start to come up with these measures. And, right now, frankly, in ultra rare, we're stuck because we're stuck with trying to validate, this whole process that was really designed for much larger diseases. And we're not sure, do we have to do it? Can we just, use it? What's going to be the implication of using it in a trial, especially with these heterogeneous diseases? And so if we really look at how we're going to remake this whole process and do it, up front, and then create this kind of process across both, N of 1 and N of few, and then ultra rare, I think we are going to be in a much better situation to support the thousands of therapies that are going to be, that are already in development.
And I liked as Marshall was saying, the concept of thinking ahead also to, start, there may not be a treatment now, or there may not be a reason to do a natural history study now, but there's going to be potentially the reason to do that, because that is actually, I think, CPATH has a lot of those data from DMD is a good example, Huntington's disease. The communities, Huntington's disease is a good example because, Gene discovered in 1990, I believe it was, and they just started collecting data, just hundreds and hundreds, thousands of patients of data, to have that ready access database and to have even the imaging that really wasn't at the time the standard, thinking someday we're going to be able to use this. And there was an initiative CPATH that led to a new diagnostic criteria. In DMD, another great example, just collecting early trials, but also natural history studies that at my former company, unfortunately, had a drug that failed. But the clinical trial design there, where we were able to use some of these, not difficult, just different methods to use existing data, borrowing Bayesian borrowing from natural history data to augment the placebo group, so you could keep your placebo group small and just have most people on active treatment, which is, in Marshall's presentation, you've got to make a trial people want to come into. And that was a way to sort of make sure that we minimized exposure to placebo, minimized necessary of contemporaneous biopsies, things that were helpful to get patients into the study because those data sets existed and we were able to leverage them to get the job done. So I can't emphasize enough the importance of that natural history data. And right now we're doing a GRIN natural history study because we don't think we'll need it now, but we might need it later, or somebody else might.
But think about it from the FDA standpoint. Usually what happens at 11:59 is when people bring you, here's the outcome markers we want to use on this clinical trial. You're like, oh crap, how are we going to do it? Oh, sorry, didn't mean to swear. How are we going to do this? Instead, what if you move that process much earlier? And this is where the patient family organizations in the academic world, and frankly, the NIH can step up. No, they can. They can. Got to bring everybody into the table. But if you actually start early before you're doing that 11:59, here's the outcome measures, and you actually get them really rock solid, well tested in the patient community across multiple diseases, that becomes much less of a, gee, do you think this might be an okay outcome measure? That makes it easier from a regulatory standpoint. At least I think. I'd love to hear.
Yeah, no, I agree with that. I mean, I think, we've definitely had a lot of, not a lot, but people have come in and we see this isn't enough patients, this isn't, and then at that point, they're trying to retrofit registry data, natural history data that was not designed to be used in the context of this trial, and it makes it very, very difficult. But, it's like everything. If you come in as part of your plan from the get-go, we have a real world evidence team that is amazing that, helps, you kind of figure out what data you need in order to make this, fit for purpose in the context of the clinical world. And so, that's exactly right. I think any foresight that you have and any pre-planning that you have, I think we're very open to natural history data, but again, it has to be robust. It has to be, and that early on if you come in early on with a proposal to include it. And I think that's the real key, because then we can work sort of simultaneously with doing the clinical trial to make sure that we can make that data as useful as possible.
And then this has been a very, very good discussion. Do you want to make sure that the audience continues to keep those questions coming through Slido, and the team in the back will continue sending them to me on this very fancy iPad, which I will definitely be adding to my holiday gift list, or maybe just taking this one. We want to get to some questions. Peter, I do see your hand, but Mike Pagnowski, I know you wanted to jump in a little bit earlier.
Sure. We're talking about natural history studies and, clinical trials and the ability to make inferences from those clinical trials. And again, I think we still have a much more kind of basic issue here is like we've approached this N of 1 space and these individual oligonucleotides as treating a patient and not as science or evidence generation. I'm not minimizing that point, but I think we're getting to a point today, and I've heard a lot of common themes over the course of the day where there does appear to be common ground that we need to bring broader infrastructure to this. We need to bring some control. We need to bring some specification, whether it's of endpoints or timing of sample collection or dosing intervals. All of those kinds of things are amenable to some degree of structuring. And the aim of all of this is really so that we can take what we've experienced with these individual patients and generate transferable knowledge from that. If not, make inferences about whether or not it's benefiting a certain patient or whether the paradigm more broadly is benefiting patients.
Absolutely. And Klaus, I know you wanted to.
Yeah, before we go to the audience, just real quick, what you said actually is so important, and you don't need to suffer by just walking into setting up a registry blindly. No, and through our partners of NORD, we've helped many rare disease groups set up registries from the get-go with a solid regulatory strategy that allows that reproducibility, data lineage, data quality, information value to be there from the very beginning. So that's something that it's not just because it's a registry, it's not valuable if you set it up right from the beginning.
And credit to Global Genes is doing some really good work in this area as well too. There's a lot of cool stuff out there. We're just not using the tools kind of as much as we can.
Well, I think Fyodor maybe is going to help on that front a little bit. And see, Fyodor, please.
I appreciate the opportunity. Marshall, I second your point about Chris Austin that rare disease has a PR problem.
It does.
Dr. Munchel, I'm really glad you're here because I'd love to speak with you and your colleague, fellow panelists, about non-malignant hematology gene editing. It's a field, obviously, in which we have, since 1996, a pretty impressive track record of lentiviral gene transfer. 18 different lentiviral therapies in that space have proven fantastically effective. Don Cohn, most recently, there's been a beautiful New England Journal paper, 60 subjects treated with lentiviral gene therapies, percentage overall survival, 100%. So these are not incremental outcomes. These are life-saving. And as you all know, commercializing a lentiviral therapy in that space has proven stratospherically difficult. So enter the gene editors. I'm going to get to my question very quickly. And we can make a personalized editor for pretty much 90% of the mutations that cause inborn errors of immunity. So I'd love to discuss the concept of stepping aside from rare disease and starting to think about clinical syndromes. And this is a term I learned from Don Cohn, from Harry Malik, from Jennifer Puck, and all these doctors who treat children with inborn errors of immunity. And when I speak with Don Cohn, he says, well, Fyodor, there are 22 genes that cause SCID. The clinical endpoint for efficacy in all of them is the same. Why am I telling this? You're a physician in this space. CD3 count at 6 months post-dosing. So my heart of a gene editor lights up because I can build editors for 90% of those mutations. And the world-leading physician developing genetic medicines for them tells them that the efficacy endpoint, short-term, is the same. And there's 30 years of track record of gene transfer. So I'd love for the panelists, and Dr. Munchel, perhaps with you, think about no longer fragmentizing, this is not an English word, patient communities by gene and mutation and pooling them, and also pooling clinical trial data based on the clinical syndrome they have, and a near-term either biomarker or actual clinical trial readout of efficacy, whether that can help us in some way.
Yeah, I mean, I'm happy to comment on that. We're in complete agreement with that, right? I think that, if you look at the paper that CHOP is recently published about, their interactions with the FDA, that's kind of what they're proposing in the context of the urea cycle and PKU, with the idea being that because the clinical manifestations in those diseases is irrelevant to the genetic mutation, to have this, she called it the umbrella of umbrella. So an overarching clinical trial. And then, the way you deal with each individual variant is more of a manufacturing preclinical issue. And so from a clinical standpoint, I think we're very open to that. In diseases that we are confident, have a similar manifestation, a similar trajectory, that it is easier for everybody if we kind of group all of that under one clinical protocol, and then, you deal with the genetics part of it, kind of separately. And so, that's, again, this is all new, it's all burgeoning, but it's definitely something we're very interested in and talk about, a lot behind the scenes, and, are happy to obviously engage with people that, in the world that want to start doing projects like that.
Did anyone else just, Jana, we'll go in a second, but does anyone else in the panel want to respond to Fyodor's question?
Well, I can go maybe beyond from a general methodology standpoint. I think what is really important is that we start, when we started building the RareX registry, obviously that's, for a registry purpose, we convened multi-stakeholder working groups around disease areas. So not individual diseases, but across a disease type, so that we could capture the domains that mattered and then be able to do it for that entire class of diseases. And so I think that sort of methodology needs to come into play here to help this to scale.
I also think there's something you can just think about your question, you can pull from common medicine. You don't develop GLP-1s for a specific molecular entity that causes obesity. You treat obesity. Hypertensive drugs don't necessarily drill down to a specific molecular entity. And in some ways, when we're looking at these individualized molecular changes, you can actually kind of lump that. So we have a therapy for this. You got to tweak it a little bit, but we tweak doses for drugs too, to personalize them. In this case, you're tweaking a little bit of the nature of the drug, which is a new thing, but still kind of conceptually along that line.
Yeah. No, Marshall, I think that's a really good point. And then I think just to go back to the online audience, there's a question, and I don't think it's come up. I don't think it's come up quite yet today. And I won't say the two vowels, but Klaus, particularly from your presentation, there was a discussion about how to use some newer technologies and tools, or maybe some automation, because the data collection is so impressive. And so maybe you can speak to a little bit of kind of what the opportunities are there with some newer technologies to apply it to the systems you've built.
Oh yeah, yeah, that's a big one. So the process to make sure that we solve the anxiety of the different potential data contributors, and I'm not making a negative statement about the anxiety, it's just a statement of reality. Somebody in an academic setting may have an anxiety-generating source for data sharing. Somebody in industry may have their own anxiety-generating source. Through the process that we've set up, the legalities cater to that hyperventilation so that the hyperventilation doesn't happen. So that's the first thing. The second thing is we can start by just looking at the data that exists today. So this is one of the things. Let's get all the data from where the data exists, the ASOs, but use that to scale it to learn to other types of technologies. How to get the data management done a lot quicker. That's where ontologies and AI will help. I'll just put that close because I don't want to just get into that rabbit hole about AI for data management, but it is a thing and it can help a lot. But then also make sure that you understand that the data that you start integrating needs to serve a purpose. So it's very important to make sure that we understand what are the unmet needs that would have the highest amount of impact that can be solved by integrating data, and work our way to integrate the data that can actually provide those answers. And it becomes the true learn and confirm paradigm. I'll leave it at that.
I think that goes back to your physics about transforming the mass into energy, but just a little bit of integrating that data also gets to sort of the why, which we touched on a lot kind of earlier in the morning sessions about using some of these innovative approaches or conducting these trials. And so in the last couple minutes, maybe just one more final point about sort of why it is important to integrate that data and how maybe we can get Marshall, your group working with Klaus and working with some of the companies with Charlene and with Mike Panzera and also thoughtfully engaging early with Ashley and Mike Pagnowski. So I'm happy to have just a few brief remarks from each of the panelists.
Okay, I'll go, and then we'll work our way down.
A lightning round.
Okay. So actually, it's funny, whenever you come to a meeting like this, some of the interesting conversations happen in the back of the room in the hallway. And one of the interesting conversations is actually from an academic standpoint, there's a whole field that can open up around building out both, the data collection, the data methods, the validation of trial outcomes, developing new ones, developing tools that can go across multiple individuals and diseases so they're useful in those individual circumstances. And I think there's a real opportunity there to actually move the needle quite a bit. And it starts with the patients.
Plus.
One term, 1 to millions. This is, we're the PR problem. N of one, N or very few. This is about millions of people that can benefit from this. This is one of the potentially most transformational things that we can do together through the power of collaboration. The FDA has set up a vision, pretty cool vision. That's a great what. Now we can figure out the how, and we can work together to make the how happen. That's the way I'm looking at this from the highest possible point. And then we'll work on the details, and we won't reinvent the wheel. We'll make sure that the global genes is part of the conversation. NORD is part of the conversation. Industry is part of the conversation. The living experience is part of the conversation. Our academic leaders in the development of these discussions are part of the conversation. So I'll leave it at that.
Ashley.
Yeah, I mean, again, I'll just kind of reiterate what I said before. Being in the gene editing space is really, and it being brand new, it gives us the opportunity to really build up from here. And a lot of the sentiments I will just kind of echo from what you've said and what Dr. Ahrens-Nicklas has said and other people throughout. I think the way that this rare disease space is going to move forward is with collaboration, with people sharing data. And that has to come from all of the people that are developing these products and being willing to share that data, publish the information that they have both successes and failures so that people are not reinventing the wheel every time. And that's kind of really the only way this is going to move forward. And it's exciting to hear that there is so many people invested in that idea and that idea of collaboration.
And then I am being yelled at on the iPad a little bit, but Charlene, maybe we started with you on this part of the session. Maybe it's best to end with you with the last word.
Okay. So we have been talking a lot about systems change, and that is a huge undertaking. So what I would say is let perfect not be the enemy of good. We've got to do because the patients are waiting. The patients are waiting, and it is too important what we are trying to do. So maybe that's the last word.
A great note to end on. And thanks to all of our panelists for their remarks. And now we'll have a short break, and then Michelle Campbell will come back to introduce our last session of the day.
you everyone for remaining through for this last panel, which I always find these are the best panels. It's the last one. We have a tall order though today, right? Because we are the last one. And we are going to wrap up and really think about in a broad sense the direction we want to go, right? I think we heard some really great things in areas of opportunities for a path forward based on our earlier today's conversations we've had. And let's be honest, this isn't a single conversation today. There will be many more needed conversations. But I think today's been a reflection of a good way of where do we need to go, where are those areas of opportunity that we need to continue to explore, and maybe where there's already some areas of alignment that we did not realize until we were here today. So for those of you who do not know me in this room, which most of you probably do, I'm Michelle Campbell, and I'm the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience in CEDAR. And I am your moderator today. And I want to thank our panelists and who are here today to begin this conversation. And what I like about our panel is we have a diverse stakeholder perspective here today that is really needed to think about what is needed to go forward. So I'm going to quickly introduce our panelists, and then we're going to jump right on into our conversation today. I do remind folks, if you do have questions that are online, please do use the Slido feature to enter your questions, and we will do our very best to incorporate your questions throughout our discussion. And then we will do our best to take questions in the room. We have a live active audience in here today, so I will get to you if I can, but I think we're going to have a great conversation. So next to me, I have Scott Demarest, who's the Associate Professor in Pediatrics in the Division of Neurology and the Chief Precision Medical Officer of Children's Hospital of Colorado. Online, we have Sadiq Kazim, the Chief Technology Officer of Genetic Medicines for Danaher's Life Science Companies. I have Michael Lee. Oh, he just told me to.
Nobody gets it right.
All right. I'm not going to attempt it then. Director of Science and Industry Affairs at the Alliance for Regenerative Medicine. Online, I have Dan O'Connor, Director of Regulatory and Early Access Policy at the Association of the British Pharmaceutical Industry. I have Mark Tersheim, Senior Strategy Advisor of New Things at Tufts Medical Center and the President of CoBio Consulting. And really, I don't know, Julia, do I need to introduce you? You are part of the reason we are here. We have Julia Vitarello, the founder of Mila's Miracle Foundation, the co-founder of the N of One Collaborative in Everyone's Medicines. So with that, let's start this conversation today. So we're going to start our conversation with thinking about and reflecting what we've heard today and using the current regulatory framework that we have right now. What areas of opportunities based on our conversations today and your thoughts are there to broaden the patient access to our individualized therapies opportunities? So Scott, I'm going to start with you.
Yeah, thanks so much, and appreciate the invite to be here and contribute to this discussion. There was a couple of things that jumped out at me from the day, and part of how I would synthesize it is we have a unique moment in history that we're in right now, where for the first time, the science is not the biggest barrier to being able to treat patients. We have an incredible number of patients, for those of us in this field who see patients and evaluate them for these types of opportunities, that we can't even get into the pipeline. I have ones that we've designed ASOs for that I can't take to the next step because of cost of being able to move that forward. Some of those don't even get to the FDA. There's a pileup of patients. And so while we're trying to think about how do we scale this, fundamentally, patients are dying, they're not being treated, and we're worried about, to some extent, are we going to hurt somebody? Not doing something is hurting someone. I think there's a couple of things that have to crystallize in order to move this forward and start to scale, and it really comes down to what is the path to commercialization. There is no sustainability without being able to pay for the cost of developing treatments for patients. I can personally think of at least two models by which we could do that. I think they probably work with an existing statute, though I'm not the expert on that space. One could be that you could have an individual patient that you are developing something for. You have predefined outcomes that if you hit that, it is deemed success. That would be informed both based on the patient's natural history and the run-in, as we talked about, and the importance of rigor in that space, as well as what is known about the natural history of that disease as a whole. And if you can define these things are unlikely to happen and are beneficial, and there is a reasonable risk or downside to it, you should be able to say this now becomes commercially viable. Within ASOs versus gene editing, those might look a little bit different in terms of how that plays out. But I think there's a viable solution there. You may not hit that every time, and you may continue to collect data to try to determine that and sort of have to reset after a certain time point. Or you may find that there's actually not a favorable risk-benefit ratio and you choose to discontinue. And those are all acceptable options. But right now, we don't have that first option. We don't have any way of hitting a predefined mark that allows us to commercialize. I think the other way that we can do it is really to shift from thinking about product to thinking about process. And this means we are defining all of the steps along the way rigorously and demonstrating that through doing that, in most cases, we can design a product that will be safe within reason in the context of the disease and have a good chance of being beneficial. And at that point, that process should be approved and commercialized to be able to move forward and reimbursable. And without having some mechanism to get there, we're doing this on goodwill, which is great. And people in this room are part of that goodwill, and it's incredible. And thank you for everyone who's part of that. But it's not going to scale.
Thank you for that, Scott. Julia, I know you have a lot of thoughts. So I am actually going to give you the option to go last if you would like as you collect them, or would you like to go ahead and start?
Either one.
You want to go ahead and start?
Sure. I mean, I think Scott just said so many important things. It's actually great to know because we've been working together with a lot of you out here that it is, I'll say, usually I'm sort of the negative one in the room. I'll say it's really, really exciting to see after almost 8 years since Mila began treatment. I did not have a lot of hope over these years because I didn't see a lot changing. And I think this meeting is representative of a really pivotal time because I'm hearing more and more people say what Scott and I and Tim and others and Janet have been saying now more recently is that, individualized medicines is here. Like, it's not my idea. It's not Scott's, not Tim's, not Dan. It's here. Whether we like it or not, it's here. And it is a drastically different paradigm than what we've dealt with for many decades. And to see that people are really changing their mindset, especially in the FDA as well, and all sort of looking in the same direction is a huge, huge change. And it's been happening just very recently. I think, Mila's path opened up the opportunity for dozens, as we've seen, of other children and adults that have received individualized ASOs, and more recently, baby KJ. And I think jumping to a different modality and different type of disease shows that this is much bigger than neurodegenerative diseases in children. It's much bigger than ASOs, right? And so I've had the chance to sit here today and hit pause now and take all this in, so many good discussions today, and take a step back for a second and say, what are we trying to solve for? And what we're trying to solve for me is, I don't use the word rare disease, is genetic disease. When I do my homework, I don't really find that many genetic diseases that are large that fit through the normal clinical trial process. I could barely even find a dozen, to be honest. But I'm no expert, but it's small. And we know that there's 10 to 15,000 small, rare, whatever you want to call them, small genetic diseases. Of those, they're being further recategorized. The patients are getting regrouped, often by single mutation. Mila had Batten CLN7. There's like 200 known kids. There's 88 mutations. So you can do the math and see how many kids maybe share a mutation. There are others like Wheeler, that has a different form of Batten disease where you might have 30, 40 kids in the US that have the same mutation, but it's still so small. So the problem we're trying to solve for is that we have a system based on population medicine. And now the reality of genetic disease is like nearly everyone is going to be regrouped into tiny groups. And that means thousands, I'll be conservative and say thousands, but we know it could be more, of medicines
each for small numbers of people. And great news is that people are starting to think about that the right way right now. The question really is, and I'll piggyback on what Scott said, is I think it's pretty straightforward, is that we can't go product or treatment or child by child. We just can't. And I'm going to represent today not only Mila's mom. I feel a huge responsibility. The reason I'm still here, like nearly 5 years after my daughter died, is because I receive phone calls from parents whose children are amenable to today's technologies. That is not okay. And I have to say, hey, you know what? If you want to sell cookies and lemonade and raise a few $1 million and happen to find a miraculous person like Tim and move to Boston, by the way, from whatever country or city you're in, like maybe your kid will have hope. That's literally what I have to tell them today. So on the positive side, we're all shifting our mentality. On the not so positive side, it is longer, harder, and more expensive today to do things than it was for Mila 8 years ago. We have not changed the system. We are still talking about it at every meeting. It's why I don't participate in most meetings. This one was an exception because I think it actually represents big change. I'm tired of the meetings because they are not doing one thing. They're not making exponentially more children get access, dying and sick children that have no other option, inevitable death. They don't have the access to this booming world of technologies that could help them. And so the real problem is you have a sea of faces. I'm going to include Sonia. It's not just children. I try to stay focused on this, but it's a sea of little faces of millions and tens of millions of dying and sick children who have no other option. And then all these incredible technologies, ASOs, CRISPR, and other. And there is literally no bridge. The only bridge that's not a giant clinical trial for large diseases is one that it feels like Tim and I duct taped together with like hundreds of other people. And there's people still going over it, but guess what? It is possible. And Lorem's done an amazing job. Tim and others have done an amazing job. And it's so exciting. We have like 85 treatments, but they are just as Herculean as they were in the beginning. They require a massive effort of philanthropy and massive number of people. It's like 1,000-page IND, 2 years, millions of dollars, cookies and lemonade, grants, 100 people. Like that has not changed. It's just that there's more amazing groups like in Lorem, like Tim, like others that are doing it, but it's not scaling. So to me, the most important question today is, we know individualized medicines are possible. We know that that's happening and it's here. How do we do this at scale? And the only way I can think about that is that we move from product to process approval, what Scott just said very correctly, and that if a sponsor, whether it be academic, nonprofit, for profit, I don't really care, demonstrates with repetition across treatments of different diseases, different mutations, same route of administration, same process, same everything, same chemistries, can they get to a point where they collect that data and they get to a tipping point or criteria threshold where they don't have to come back to the regulator? That is the elephant in the room here that I haven't heard anyone actually say that out loud. You can't come back for every single dying child, because if you do, I'm going to give up. Like, I'm done. It's not going to happen. We might get to 20 or 50 or 100 a year, but we need to get to like tens or hundreds of thousands eventually. So I think it's important that there's reimbursement at the end of that process approval so that we have the other half of access, which is regulatory and reimbursement, so that we can see this, many sponsors doing this. I use sponsors loosely, okay? Academic, nonprofit, for profit, doing this truly at scale without having to come back and having many meetings and looking at every step of the way for every molecule with a regulator. Otherwise, we'll just never get there and everyone will die,
Yeah. Thank you for that. And I'm Sadiq, who's online with us today as a panelist. I'm going to come to you next. I'm going to next go to Michael, but I just want to let you know that first. So Michael, what are your thoughts in the world? Because I know you're more based in the gene editing and the gene therapy, cell-based therapy arena.
Yeah, so we work with our members on cell therapies, gene therapies, including gene editing, and interacting with the regulators on these topics. And Julia, what I think both you and Scott just described is a problem that, many, many people have experienced, patient organizations and also developers, how hard it is to develop these therapies for a small number of patients, let alone scale them and get approval using the traditional paradigm. I do think there's a lot that is encouraging in the suggestion of a plausible mechanism pathway. I do think we need more examples of that. And, we're hopeful and optimistic that the FDA will engage on that. We know that's happening now, with CHOP, for example, and with IGI. Ultimately what we need there is guidance, and for that to become more formal and to describe a route to approval. But I'll say, what we can do today, I think, I still view it as a product, but without arguing over semantics, it's really a family of products, like a very large family that makes the Brady Bunch look like a small family. And the family lives in, or the family is built on a platform, right? And I know we kind of overuse that term, but it's pretty much all I ever talk about. And it's not the platform technology designation, which is great, but it needs to be based on something before you have an approved product to work, like at a pre-IND stage. And we can see, for example, at CHOP, and Rebecca Harris-Nicholas did a great job presenting on this today, and she talked about it as a platform. They're working it right now. So if you have common elements of a development program, whether it's a process or a product, common manufacturing elements, a common vector, for example, an LNP, a common editor, base editor with some variations on it, maybe a different PAM, and you have patients with a similar disease, same phenotype, very different genotypes, you should in principle be able to streamline development and drive down the cost. So, we heard about it earlier, reduce your tox testing and biodistribution testing substantially, realize CMC benefits like streamlined validation testing, simplified release testing, I think, and even cutting down on stability testing, which I think will be important as you're trying to treat more patients, or treat a single patient over a longer period of time with these drugs. And as we've heard today, also construct clinical trials using an umbrella approach, to just make it easier to get to the approval point.
Thank you for that. And Sadiq, what would you like to add from your perspective on the manufacturing aspect of these therapies?
Yeah, well, first of all, thank you for the opportunity to join. I think the big challenge from the manufacturing perspective is really the concept of risk-benefit commercial manufacturing. So manufacturing, there's a precedent. Mila obviously led the way, KJ in the gene editing context. We have risk-benefit frameworks to enable first-in-human clinical trials and clinical manufacturing. But then beyond that, it's ambiguous. And I'll give you two very distinct examples that I think illustrate that case. So if we're talking about something as being a platform, if we look at the baby KJ example, there were actually 10 partners that mobilized with Dr. Rebecca Arns-Nicholas and Dr. Karen Musunuru from across the ecosystem, technology and solutions providers and other CROs that contributed various technologies that enabled the final manufactured drug product. Acuitas delivered the LNP, Aldevron delivered the mRNA encoding the gene editor, and IDT delivered the guide RNA. Now, of those three components, two of them could theoretically be reusable across many different patients. One of them is custom for baby KJ. Now, when you go towards commercial, if you want to launch a product commercially, you need to go through the entire PPQ process validation. And the question becomes, from a risk-benefit approach for commercial launch, what aspects of that platform need to undergo the full armamentarium of process validation? And can we take a risk-benefit perspective on the individual components? Second concept relates to the platform designation. So the developers have to be incentivized to develop these technologies in a way that's cost-effective and sustainable. And if we limit the platform designation to only those pharmaceutical developers that have already a commercial product, then you're losing out on the individual platform components that enable the overall platform, like the LNP, like the guide RNA, like the mRNA. And I think that regulatory ambiguity leads to uncertainty in terms of investment and leads to that long-term challenge that was identified in terms of patients needing to fundraise for very charitable ways. So we need to figure out a mechanism from bench to bedside to billing. And I think none of these concepts are fully aligned. And I think commercial manufacturing within the context of N of 1, we can't retrofit it and try to fit it into the current paradigm that exists for large-scale manufactured drug products. It's different. We need to recognize that, and there needs to be clarity from the with line of sight towards commercial enablement. And I'm agnostic on the commercial path. I mean, I'll note one more thing. You'll notice that of the 10 partners that worked with Dr. Rebecca Arns-Nicholas and Dr. Kiran Musunuru, none of them was a traditionally vertically integrated pharma or biotech company. All of them were solutions providers or academic institutions. So I think when we think of the guidance and in terms of the reimbursement, we also need to think very creatively that these drugs will likely not be taken to market by a pharmaceutical company or by a biotech company, but likely by different routes that don't exist in the ecosystem today. So we need to designate and design these regulatory pathways, not thinking that the ultimate sponsor is going to be a big pharma or a biotech, because they've had the opportunity for many years, and many of them have shelved these products because of the lack of billing uncertainty and regulatory. We need to be more creative in the way that we regulate these things for risk-benefit commercial manufacturing that will enable sustainable treatment of many other patients.
Thank you for that. And I want to turn to Mark now, because you are the one voice we have not actually had a discussion with today, and that is a little bit of that payer perspective aspects and the evidence that we need to think about generating.
You're going to make me play the mean guy.
No, I'm not. I mean, it's usually me sometimes, so I'll take a few minutes off the seat. But what are your thoughts as we as we think about what we exist today? And where are some opportunities from the perspective of what you see, and how do we think about this? And this will actually lead into a broader, hopefully, next question where we get a little more into the aspect of reimbursement, which is something that hasn't been mentioned, but it is something that's in that needs to be thought about, because the one thing I have been hearing regularly across today is the cost aspect to be able to do the work. Not even to get to us, right? To have the conversation is how do we do that piece? So, what are your thoughts with what we have right now and the things we need?
Thanks very much for including me. Great to be here. Just to clarify my role and position, I'm here from Tufts Medical Center. We run a program called NewDigs, which runs multi-stakeholder consortium and has for about 15 years. Some of you may remember us from long ago when we did adaptive licensing. Others of you in the room may recognize us the last 5-10 years. We've been really emphasizing cell and gene therapy, payment innovation, and reimbursement processes. So we do systems change through multi-stakeholder collaborations. One of the key collaborators always at the table has been payers. And we've worked with dozens of them. So I have all different kinds: Medicaid, Medicare, private, self-insured employers, et cetera, for those of you who know that space. All right. So I'll be channeling my payer participants as we talk about this today, amongst others. They live in great fear of providing care to patients that is snake oil. So any of you who come from Southern California know there are large numbers of Mexican cell therapy shops that patients routinely go over the border south of San Diego and do a variety of cell therapies, right? Their protection from doing those unregulated, not knowing if they're any good therapies, is FDA approval, right? In the absence of an FDA certification of some kind, whether it's the new mechanism approach, accelerated approval, or a full approval, payers won't touch any of these therapies at all for two reasons. One, they honestly don't know if they're going to work, right? And two, if they say yes to these without an FDA approval, they have a backlog of several hundred other therapies that people are saying, gee, you ought to reimburse us for this. And they're not going to open up that floodgate for these kinds of therapies. So they're in a bit of a box, right, as to setting precedent. They need to know that these therapies are going to work. And from a commercial standpoint, well, what does that mean, right? So what you're really talking about is not commercialization. You're talking about money, right? I'm a health economist, so I was wondering when we were finally going to get back to money today, right? And what you're saying is, well, NIH isn't funding this, right? The philanthropy has limited pockets today, right? We need another pocket. Let's commercialize. Okay? And until you can honestly tell both patients who have not been involved in the process at all, right? Don't think of your patients who are coming to you saying, gee, do something bold. I want to be courageous for whatever reason. I want to participate in this. Think of the Medicaid patient who knows very little about any of this, right? And you're now going to tell them, we have a therapy, it's going to work for you, right? And it's been FDA approved, right? Or not, right? And we want Medicaid to pay for it, who, by the way, won't pay for anything that isn't Medicaid approved. It's part of their rules and set in their legislation. So how are you going to approach those patients to assure them that it's going to work? And for those Medicaid directors in each one of the states, right? How are they going to know that, well, you've never done it in this disease for this patient, right? Because your platform got approved, not this indication, because there's only 2 patients who might ever have this in the next 10 years, right? So what's going to be our metric to know that this worked for this patient in this condition? Because I have taxpayer monies I'm paying from the state of Texas, right? State of Oklahoma. How am I going to tell my legislators that I've spent this money well for them? And for other big things, I say, well, on average, it kind of works out. I don't have to worry about this. But this is very particular. And you're probably going to want to charge me $1 million, right? Which that's what you charge me for rare diseases that have 100 patients, right? It's been every order of magnitude. The price goes up an order of magnitude. So are you thinking this is a $10 million product, right? Well, anything that hits over $500,000, $100,000, they're going to want to have some way to know it made a life-changing impact on that patient, right? So they look, as you've talked about today, they want clinically meaningful patient improvement endpoints. And are you willing to say, pay us nothing if we don't reach, I think Janet and some of our other friends were saying, 50% improvement on a standardized scale of morbidity, right? 50% improvement. You willing to stand behind that claim? Pay you nothing if you don't hit 50%, pay you $1 million if you do. Then you're going to start having conversations with people, right? But without FDA approval and without standing behind, is it really going to work? I think you're going to have a very hard time getting to tap that type of revenue, right? That pocket of money, right? To put it in context, as I was sitting here thinking, I was involved in the Human Genome Project. Sorry to go long, just be really short. I helped run one of the 3 or 4 largest genome sequencing centers in the world back around 2000, right? I just looked up today how much that whole program cost. It was $3 billion. I'm an economist, so I adjusted it to today's money. So that's about $6 to $7 billion. And I worked on it from the private industry side. And I know that we spent just as much as the governments did. So we spent in today's money about $12 billion on the Human Genome Project, right? I heard today a benchmark of $1 million per kid, right? I heard that. That was without you guys scaling. $1 million, 10,000 patients gives me $10 billion. That's $2 billion less than the whole Human Genome Project cost, right? Why aren't we gathering together and selling this as not a one by one, all the crabs fighting for a little bit of something in the barrel? And this is a huge, large program of $10 billion to treat 10,000 kids over 4 years. It's the clinical payoff from the Human Genome Project investment from 10 years ago. Let's have a similar program now. You should be able to sell that. Right? And industry, once they see that money going in, they'll double it. You'll have 20 billion before you know. So that's how I think you approach it.
Well, thank you for that. So Dan, I want to turn to you with this more of an international perspective of thoughts about the regulatory approaches there. And, you know, if you want to highlight anything that's currently going on in the UK with some of the efforts, because as we know, rare disease drug development is not unique to the US, it is global. And there's other health authorities around the world that are facing the same challenges and are asking similar questions to what the FDA is. So Dan, I was just wondering if you could provide us with some thoughts about leveraging current regulatory frameworks and where are those opportunities for these individualized therapies?
Yeah, thanks, Michelle. Really, really good to be there today. Sorry I couldn't be in person. So I'm Dan O'Connor. I work at ABPI, which is a industry trade association. It's really good to hear some very good alignment across the panel. Actually, I think there's good alignment in terms of the problem statement. I didn't hear all of the discussions today, but it sounds very much based on what Julia said, that actually there's a really encouraging momentum in terms of actually we've got to change the paradigm. And I think that's really, really key from an international perspective, because I think we can absolutely learn from each other. I think one of the consistent messages that comes out is that the current regulatory and payer system really needs to be amended. It was designed in a very different time when we've already heard that it was based around population regulation. And now we're really moving in an incredibly exciting time of 21st century technology, personalized medicine, and the opportunity to really transform patient lives. But we won't do that with the current system that we have. And I think there's a real momentum now to move from the theoretical discussions to actually some practical work. And that's what we're trying to do in the UK through an initiative that Julia is also involved with, and some of the other colleagues that are in the hall today, called the Rare Therapies Launchpad. I think a commonality from that initiative is that not one organization or one part of the discipline in terms of creating a new pathway can do it on their own. So although the regulatory system is absolutely crucial and the regulatory system absolutely adds value, there are other parts of the end-to-end pathway, the diagnostic part, the approach to ensuring that you match the mutation, the actionable mutation, to a therapeutic modality within the therapeutic window, and that you design a system where you have good regulatory oversight so that you can actually follow that patient through that process and learn from each patient. So what we're trying to do in the RTLP is really, we've got it termed a switch from research to treatment with iterative learning, because as long as the modality is still considered to be research, then unfortunately the payer system is not going to consider that something that they can support. It's going to come down to philanthropy or research funding. So I think that's a really crucial component as part of the pivot that we want to create within the UK. And we're not starting from scratch. I think there's many really great precedents from a regulatory economic modeling perspective that we can sort of build on and really build out the opportunity for responsible innovation, really trying to help all of these patients. So through the RTLP, I think there are a number of core areas that we want to tackle. I think the first is that, as we've already heard, the switch from, and we've just seen some movement also from the MHRA in this direction, a sort of switch from thinking about authorizing individual medicines, but actually thinking more of a process approval type approach. I think another pillar is learning from data, and I heard the last session around the importance of data and the ability to share and learn from that data. A key part to the model that we want to test in the pilot is really thinking about centers of excellence to deliver this medicine, where you could utilize the regulatory system in a more arm's length type approach with more do and tell type processes once a process approval has been put into place. I think that's a really key aspect around that delivery into expert centers. But that must involve collecting data, and that iterative learning is a key component to that, alongside really a bit of a reset in terms of what are the roles and responsibilities of each of the individual players that are in the field: industry, patients, the hospitals, the clinicians, the regulator, and the payer. How do we all work together and ensure that actually those roles and responsibilities are really clear? So in summary, I think what we're trying to achieve is to create a new end-to-end pathway, and that link between where actually increasingly we'll be able to find the patient, so that diagnostic odyssey, which has been so problematic for the rare disease community for so long, is actually not necessarily now the big problem. Actually, what is the big problem is the gap now in between once you found the patient and linking that patient into a modality that can really make a difference to them in a therapeutic window that is going to change their lives. So a lot of excitement, I think, in terms of where we can go to, and I think there's a lot of commonality in terms of the pillars that we need to pull out from theory into reality through a practical pilot.
Julia, you wanted to reflect on that?
Yeah, I just wanted to add something. Thanks, Dan, for explaining the Rare Therapies Launchpad. I think, I didn't have a lot of hope in the US that this was going to scale until recently, and now I do. I went to the UK and met Dan at the time, who was running the Innovation Accelerator at the MHRA, the regulator in the UK, who immediately, within the first call, said, there's no way that this new era of individualized medicines is going to fit into the current system. Clinical trials will never work, and we need to think about this differently. All the way up to June Rain, who was the CEO of the MHRA at the time, also agreed. And that's really what kicked off 2 years ago, the Rare Therapies Launchpad. And we brought together sort of representatives of the different stakeholders, not everyone, but we had a few hospitals. We have Everyone Medicines, the company I started, so that parents will have to stop selling cookies and lemonade and selling raising millions of dollars, and all got together on the table in order to demonstrate, in some ways on a small scale, what the new regulatory and reimbursement path might look like. We realized that we had to do that by treating patients. And so it was literally knocking on the door of the MHRA and going there with the first patient. And what's very exciting is that in the last 1 month, within the RTLP context, our representative sponsor, which is Everyone Medicines, because it's the only company that's crazy enough to exist right now because there's no pathway to reimbursement, but we believe there will be, they brought patients and they got a green light from the MHRA to have a master protocol, which is really exciting. It's a master protocol across different diseases, different mutations, all children, dying of different neurological conditions, all intrathecal delivery, all ASOs of similar chemistries. And that's not the ultimate fix, but it's a great step to get to the ultimate fix, which is the process approval that Scott was talking about earlier. We got to start somewhere. So after we treat, 10 kids in the context of the RTLP, the idea is to demonstrate that you can keep the same process each time and that you can collect that data, which is critical. And Klaus, really, that to me is like, without that public database of this system doesn't work. We need to have the data that enables, or let's say that guides the sort of the new standards. Once a sponsor is process approved, they're not just like free to keep going. There's still regulatory oversight. It's still a application that's owned by the FDA. Or the MHRA, but it's of the process, and there's data that's coming in that's actually backing up those standards and updating them. So I think the MHRA has done, in the context of the RTLP, has done a really good job at showing what could be done now here, and I think there could be really good communication between the regulators. Like, we both want to make this happen, and different systems, but very similar principles. So,
thank you for that. Thinking about what we've heard today. We've identified, I think people may have been surprised by some of the things we've heard today, including, hey, we don't hear about all of these things, the successes and failures sometimes, because we're not publishing them or we're not data sharing, right? And I'll be honest, I go to plenty of workshops and meetings like this, and I learn more about what's happening in people's programs than I do when I see you come sit across the table from me sometimes, right? So I think in the spirit of how do we collaborate together with what we've identified, right, in these areas of data sharing, what can we do a priori specifications? How can we standardize and develop rigorous approaches? What is that evidence needs to be generated for reimbursement? What is that going to look like? Because we need to start thinking about that prospectively now, right? And this is true in all the rare diseases, right? Everyone's excited. I know Pat Furlong was here earlier today, right? And we should talk about people are all excited for the first approvals, and then oh my gosh, now it's the reimbursement piece. We didn't think about that, right? Uh-oh. We have to think about it all together, right? Because in the end, we want to get therapies to the market, and we want to make sure we have access to the largest amount of patients possible with these approved therapies, right? So thinking about this as we begin to start closing out some of our thoughts, what are these areas of collaborations from the seat that you sit at this table or this, our long stage and virtually today? that we should be thinking about with our audience here and to help formulate these beginning conversations that are going to need to happen. It's not when they happen, they're going to happen, to think about, because it's going to be some different conversations and everyone's going to come together, but we do need to start thinking about what that looks like with all players in this space. So Scott, what do you think?
So there's a number of things we've talked about today that are actually not N of one individualized custom therapy problems. They're rare disease problems. This issue isn't actually a rare disease problem. This is a platform technology problem. We've never before had a situation where the treatment of one drug or therapeutic could have such a large impact on other therapeutic programs as we do today, whether we're talking about gene editing, gene therapy replacement, ASOs, all of them. We are not set up to be able to transmit that knowledge across programs effectively. There is mechanisms for that to end up in the FDA's hands.
And we can do that once it gets to us.
But how we get it out from the FDA to everyone else who might benefit from knowing about that is really challenging. And I think there's a real public good argument around necessitating, whether by law or other mechanisms, that sharing is mandatory within this space. And if we're thinking about alternative pathways and expedited approvals, maybe now's the time to negotiate some of the trade-offs associated with that as well as we move in a direction and a pathway towards commercialization. We must have ongoing learning and the ability to integrate those data so that we can understand the impact and ripple effect of learning on one program that has impacts across the platform. That's a new problem for us that we didn't have to deal with in small molecules. I really love the RAC idea, or analogous, sort of a committee. We don't have to wholesale give all the data away so anybody can look at it, but you could start with, is there a committee you can bring together of various stakeholders where you could review data and responsibly decide how and what you can share in aggregate ways that protect certain elements for companies if they're in the space, for privacy of individuals who are part of these treatment programs so that it's not identifiable, but allows us to share the scientific knowledge in a responsible way. That might be a starting point to starting to kind of move the bar a little bit on that. And of course, working with CPATH or other groups that can help to aggregate and create common platforms that allow us to more easily be able to share data is going to be critical, and it has to cross preclinical and clinical domains.
How do we de-risk some of this to comfort levels for that data sharing? Because we all collectively have to learn. Like, we have to learn too. I mean, we heard today in one of our earlier sessions with something that said, and we said, please send us that information. We would love to learn more about why you think this is possible. We need that evidence, because perhaps, if we don't see it, because no one else is too afraid to tell us, we won't be able to respond. We give the best advice when we get the most details. So I think that's a really highlighting point of how do we work together in that way to feel comfortable.
And I think as any one individual developing something, we've never before had such a strong opportunity for ROI on sharing. If I share my couple of things that ASOs I'm trying to develop, but I get access to hundreds of ASO developments, my ROI on sharing is incredibly valuable, right? So I get a lot more back than I had to give in order to do that. And it really allows the ecosystem to learn together and all members to benefit from that and have more drugs that are likely to be safe and efficacious than if we're all holding it close to the chest.
Thank you for that. So I'm just going to let my panelists know the order I'm going to go in as I'm watching the time go down. I want to manage us correctly. So I'm going to go to Sadiq next, and then to Michael, and then to Mark, and then Dan, and then Julia. So just so you guys know the order of this and with your thoughts. So Sadiq, what are your thoughts?
Just building on the concept of the process being the thing that gets approved ultimately, or the platform. I think with the example of baby KJ, the recommendation would be to widen the aperture of what constitutes the process, because each of these elements were not owned by a single entity. Although the charge obviously was bravely and fearlessly led by Dr. Rebecca Arns-Nicholas and Dr. Kiran Masanuru, there were individual technology and platform components that came from different players in the ecosystem. And it happened once for KJ, but how do we incentivize each of those players to now contribute in a cohesive and sustainable way to make this platform process actually something that they can repeat, rinse, and repeat, wash and rinse repeatedly for other patients like KJ. So the point here is that the technology, the sharing spans all aspects, not just clinical development, but I think manufacturing, non-clinical, and then recognizing and incentivizing the players to cooperate with each other. So it's not trying to build mini fiefdoms that try to recreate some aspect of what the other has so that you can have it all vertically integrated. I think what the KJ example shows is that if you can align people around a mission critical purpose and incentivize it in the proper regulatory and reimbursement pathway, people will work together. But if we sort of use this current path where people are incentivized to just build their own thing and their own little mini fiefdom, we're just going to be having this conversation again and again and again, 5 years, 10 years, 15 years later. So it's really being generous about the concept of platforming, who can drive a platform technology, and what does it mean to have an approved platform process rather than an approved new molecular entity or new biological entity.
Michael, your thoughts?
Yeah, actually, Sadek and I have talked about this a lot, and I think what you just heard there is you can incentivize a for-profit company to have an interest in this, and it's of course, I know that Sadek is doing this for altruistic reasons, and he's really a believer in it. But I know also that Danaher believes they can do this and not give everything away for free. So you have to share data and information, but in a way that doesn't disincentivize the key parties that are involved. One, I'll give you an example of something we did, a small first step, but one that we think was important. We held a meeting in November of last year. We call it the Scientific Exchange. So we convened with companies like Danaher. Danaher was a supporter. Thank you, Sadek. We convened gene editing companies, about half a dozen of them, you know their names. And also academic KOLs who are working on this problem from IGI. Thank you, Fyodor. And Kiran Masanuru participated as well. And the task was that people, we didn't come in under confidentiality. It was a small meeting, but we live cast it to the entire world. We had over 1,000 people watching. And the task was bring a case study using these platform principles and give us an estimate of how quickly you think, or how to what extent this would streamline development time-wise and cost-wise. And there were a number of the developers were very forthcoming in this, and some of the results of this have been published in a paper in Cytotherapy that I'm happy to refer anybody to if you're interested in reading more about it. But we estimated that you can, and this was actually Kieran's case study, you could develop a product like this in as little as 6 months. And we knew that they were actually working that case study at the time, but I didn't know how far along they were. So I remember when he first said that, I thought, are you really going to stand up and say that in front of everyone? But he did, and they did it. So that shows you that these principles can be used to speed up development substantially. We also believe you can cut down on cost by a factor of 5 or more, perhaps an order of magnitude. That part still needs to be proven, but I think it's possible if you give the right incentives. The incentive for a developer is if you can see over time how you can use these principles to extend into larger indications, right? Then that is the route to sustainability for a for-profit company. And that's something that I think we should keep in mind as we're going through this process.
Thank you for that. Mark, what are your continued thoughts as you highlighted some already about what you have seen in your experience with working with a diverse payer population within the US, because it is a diverse population themselves, and bringing that voice to the table early, probably earlier than people think most of the time is my guess. But how do we start thinking about that when there is uncertainty of success, right?
And the payers know that there's going to be uncertainty. No drug works perfectly, right? They just expect that. But this field this year has, I think, now passed a tipping point where payers will be concerned, right? Enough to come to the table to talk to you about it. Clinical success, pathway to approval being talked about, where before, if they have success, it's experimental, I don't have to deal with it. Oh no, FDA might actually approve these things, right? And a pipeline of hundreds. Oh my gosh, this is a big deal. Because if it's one or two, it's like, one or two, lightning strikes, I'll take my chances, right? But now you got a troika where they have to start thinking about it. You're now the management consulting theme for payers for 2027, right? 2026, you sell it. 27, they'll all be at the table, right? Even next year, the leading ones will be. The best payers always think 3, 4, 5 years ahead as to what's coming through the pipeline, what they have to be ready for, et cetera, whether it was gene therapies, obesity drugs, et cetera. You're at the point where the advanced payers are going to be at the table now to talk to you about what this means, and that they're going to be need some time to think about, this isn't an indication approval, it's a platform approval across many things, and being truthful with them that it's not going to work every time, that there's going to be some failure. It's going to work 3 out of 4 times, right? 1 out of 2. But at least then they have some sense. All right, well, we have to think about reimbursement in that success rate context. It's a great time to be creative with them and hear them. And patients, you'll be surprised that the payers sound a whole lot like you guys at this time of the conversation, because they will emphasize, we don't care about biomarkers. Don't tell me about molecular this. Did it change the life of the patient in a way that they cared about? What's your endpoint that the patients are going to value, which means that the members in my plan are going to value it, which means that the employers are going to value it because their parents are going to say, my kid needs this to help them in their life. So both the payers and the patients, when it's at this junction, are completely aligned with telling the research scientists and the FDA, molecular's molecular. Tell me what the endpoint is that's going to really be helpful to my patient, and that's what I'll pay you for. It's going to be great conversations, and then they'll be ready to pay for them because you're going to convince them through all this that this is transforming kids' lives and older patients' lives, et cetera, right? But kids are always an easy story. Sorry I went there.
That's all right. I'm trying to also read the room to see if anyone's going to want to ask a question as I build in the remaining of my moderation time. Okay, I see some potential. I see Annie. All right, so we're going to get to Dan and Julia, and then I will allow, let's get the mics. Dan, what are your thoughts on areas and opportunities of collaboration when it comes to these? And I would probably just add the international aspect, because I'm sure if something's happening here in the US, it's going to come to MHRA, it's going to go to EMA, Health Canada for similar type discussions.
Yeah, thanks, Michelle. I think there's definitely the UK national opportunity for collaboration, and then there's the international spot. So I think from a UK perspective, I think the cross health system collaboration is really, really critical. As I said earlier, I don't think there's not going to be one stakeholder that's going to solve this issue on its own. And I think certainly in our model, when we think about one of the elements of the Rare Therapies Launchpad, when we think about the accredited center, I think that offers the right amount of guardrails, and if that can be linked to reimbursement, so bringing all of that collaboration together, the accepted additional uncertainty, but in areas of higher medical need, then I think that would generate the scalability, but also the sustainability in terms of ensuring that there's good industry buy-in. But that sharing of data and the trust in sharing that data, and that data sort of being able to be audit trail in terms of the data is collected and that you do learn from that, I think is really crucial. So that does require, I think, quite a sophisticated framework to be able to collect that data and to be able to use that data in a national approach to collaboration. I think certainly if you look at the last couple of years and you think of all of the different initiatives and all the different interests, the US, the UK, the Netherlands, Australia, a number of other kind of countries all working on this similar problem, there's got to be opportunity. There was a meeting last year in Lausanne from ICMA, the International Coalition of Medicines Regulatory Authorities, that looked at the approach to individualized medicines, and some really strong opportunities, I think, coming out of that platform and other platforms for collaboration. So hopefully today, will be a really pivotal moment for the field, building on the FDA paper, the MHRA paper, and really learning together around how can we work to treat these patients as soon as we possibly can with the tools that we now have.
Thank you for that. Okay, we'll lay out the last 10 minutes to the audience. We got 10 minutes. Julia's going to speak next. And I know I've identified at least 3 people with questions. So to my questioners, here's your thing. Quick if you can be. We also encourage our panelists, if they're willing to stay afterwards, if there's a more specific in-depth conversation you want to have, please do that as well, because I do want to make sure I give time for my panelists to wrap up with their final 30 seconds. So just laying out the next couple minutes of how I'm hoping we're going to go. But Julia, what are your thoughts as we've had this conversation about those areas of opportunities?
Well, a bunch of people touched on some really important points. So I'd reiterate, first and foremost, the data is crucial. So if we're going to move from product to process approval, and we know that it needs to be approved in order to be reimbursed, which is fundamental, then we need a database, and that means CPATH is already throwing out an idea. It's just important that we make that mandated. I agree with what Scott said. If you don't want to share your data, like go have fun and have a clinical trial. That's fine. This is a different pathway, and it requires there to be giving something up in order to be able to do this. I don't think it needs to be such a huge amount of data that it's going to be that there cannot be companies, because the second collaboration I would say would be companies. I feel very strongly there need to be biotechs in this, not only because they bring funds, which by the way, no one has right now. They also bring drug development experience. But I think the most important thing is the shared risk, in the sense that this all falls on Tim and Kieran and Becca and others. And I will say my personal opinion is those are exceptions to the norm. Tim is amazing. So is Kieran and Becca. So are numerous other people in here that have already said that they're treating people with individualized ASOs and CRISPR. That's great. But at scale, doing this thousands or tens of thousands of times in an academic center, I don't think is fair to say that that should all fall in that. So I see biotechs working with academic centers, call them interventional genetic centers. That's what I think they probably will be. That's not my idea. That's many people talking about this over the years. I think that we need to make sure that we are creating a system that allows for there to be, hopefully not just my company, Everyone Medicines, but many others, because there's tens of thousands of medicines that we need to produce. So I hope there's lots of interventional genetic centers and lots of biotech. So I think they need to be brought in, obviously together with the payers, right? The third and last, and I think this is a good place to end here too, is patients. Okay, so right now, parents like me are not being given any chance at all, unless you're lucky. Mila was the luckiest unlucky kid in the world. And if there were a conference next door on the other side of this wall that were, oncology, pediatric oncology, I have spent a lot of time talking to these parents. I have friends who have lost their children, same age as Mila, and the conversations they're having in that room next door are completely different. So the risk tolerance that's already been established in children with end-stage cancer it's a thing, and there are kids wearing 120 beads around their neck. Each one of those is an attempt on life. My daughter had 1 bead, and she was like the luckiest kid in the world to have 1 bead. Almost no one has 1 bead, okay? These kids having their ribs broken, they're radiated and put in metal boxes, they're being resuscitated numerous times, they're having horrendous treatments, okay, that are experimental, where they don't know what the risks are, and they're being given that chance. And I feel like there's just a massive divide. So if we don't bring parents into this and assume that they can consent, and if they don't consent, that's totally fine. We're not coercing anyone, because the line is going to be around the world 10 times. If we actually say your child is amenable to a treatment, to a therapeutic modality today, we have created a way for you to rapidly access that, and we consent them properly, like Tim did to me and gave me the real understanding of Mila's neurons. Some of them have died. Some of them are dying and they might be able to be stopped. Some of them are dying, they might not be able to stop. That is what I saw afterwards. He consented me correctly, and Mila's dad and I both agreed to do that. So give parents who have children that are going to inevitably die and are amenable to technologies. And by the way, there's tens of thousands of them today we could treat. So why are we not doing that? Bring them to the table, explain this to them, and give them a chance to have beads on their necklace. Otherwise, we're aware that there's a discrepancy, and it's the anger is rising right now, because now people know that their kids are amenable to these things, and we are impeding, not enabling access to the technologies that exist today. So bring them to the table.
No fun. Let's stop right there.
So Annie, you have the microphone, right?
I'm Annie Kennedy with the Overlay Foundation for Rare Diseases, and this has been an incredible meeting. I just want to tie something that happened in the first conversation in the first panel with the conversation here, and maybe a comment that a meeting just like this or a forum just like this focused less on regulatory and more on reimbursement and coverage would need to run parallel alongside this. Terry Jo did a beautiful job talking about the lived experience, and there are real cost implications for that lived experience. And as a community, we've done a lot of work to collect that data around what the health economic impact of that lived experience is. Mark is talking about payers do a good job of doing that forecasting and looking 3 or 4 years out. But I would submit to you that what we're doing here is not trying to make people cheaper. We're trying to improve their lives and trying to make people healthier. And one of the reasons that that's difficult when we meet with payers, whether we're talking about a common rare disease or a rare disease, is that we don't insure people in their health system, in their family system. We insure people and we look at them individually. And when that, to me, is the social contract that should shift. If we made an investment in Terry Jo's son and looked at the impact that would have on her and her husband and her family, because that health insurer insures them as a unit, and we collected data around that, we would be able to have a very different conversation than we have today when we talk about investing in therapies and cures and treatments and clinical trials. So I would challenge us that as we're having this conversation in a regulatory context, that we also have that one.
Thank you, Annie, for that comment. I think it resonates to everyone in this room that those are parallel tracks. I know someone else wanted to ask a question. Can you make it really quick, like really a couple seconds?
That's quite a challenge.
I know.
Max Bronstein, I run a health advocacy firm called Viva Strategies, worked on ultra rare for almost a decade now. So to the point around scale, I think this is really critical. One of the ways we can achieve scale is through incentives. So wanted to let folks know that I'm helping to craft legislation to actually create new incentives to drive, to really turbocharge investment into ultra rare. I want to go to access really quickly too, because this is equally important, I would say, because you can have the best therapies in the world, but if patients can't get access to them, we're not moving the ball forward. We spent a lot of time, Mark and I, talking about sort of value-based payments, reinsurance, other mechanisms to help sort of de-risk coverage for exciting therapies, whether they're cell and gene, ultra-rare, N-of-1 precision medicines. I haven't heard you talk about them today, so wanted to give you a chance to talk about that, and maybe Michael can speak about it too, because I know ARM has worked on this issue a lot.
Perhaps in your 30-second wrap-ups, we can tie that in.
Sounds like a great idea, given the timing.
Well, I want to, think what we're hearing, and I'm going to give our panelists a couple seconds. They know it has to be a couple seconds of final thoughts. Like, what is their number one thing they want to make sure this audience hears as we begin these conversations? I think what we've heard today throughout the day is we still need rigor. We have to have standardization. Where can we think about things a priori as much as possible? And that can happen. Data sharing is critical. We heard early and often, right? We really do mean those words when we say come early and often and talk to us. There is a reason to that. And I would say I heard a lot of things in this conversation about a fear of, I don't have something validated, biomarker, technology, a COA or whatever. That should not be your fear, right? That should not preclude you to have the conversations about, this is what I do have, and how can we leverage that, or what else may I need as part of your conversations when you do talk to us or a health authority, because you're asking those same questions. So, Scott, in a couple quick seconds, what is your one thing you want to get out to the audience?
We wandered into rare disease a bunch today, which makes sense. I think we're actually getting better. There is momentum in rare disease to think about rationally grouping cohorts of rare diseases to be able to scale natural history, to improve rigor, to validate outcome measures in groups rather than individuals. That is moving in the direction of ultra rare, from rare to ultra rare. This is where clinical trial design of N-of-1s actually informs going the other direction. Patients as their own controls is a better model for most rare disease than a frequentist model for a clinical trial. So you have those two things, our base knowledge and our clinical trial design and experience, meeting towards this space of rare disease, one coming from the ultra rare and the other from the rare disease. And I think that as you start to merge those is really where the game totally changes.
Thank you. Sadiq, what is your final thought?
I think we have a really great framework for getting into a first-in-human trial, at least from a manufacturing perspective, but lots of ambiguity and lack of clarity on the concept of approving a platform from a manufacturing perspective and risk-benefit based approaches from a commercial manufacturing perspective. And I think if we have clarity on those fronts, it'll unlock investment and reimbursement. And so that's really my main takeaway here. Risk benefit for commercial manufacturing.
Thank you. Dan, what is your final thought?
Thanks, Michelle. Thanks for doing a great job chairing. I think we should be bold with responsible innovation, and I think we really need to pivot in terms of the view around protecting public health, although important from a safety perspective, also has to include timely access to new methodologies and new medicines.
Thank you. All right, Mark, your final
Well,
thought.
you've all inspired me that we're evolving from a serendipitous system where only the lucky patients get access to these things to a adaptive, replicable process that offers these therapies to all patients who need them. That's a phenomenal pivot. As part of that, it brings up reimbursement. And as I was prompted by Max, there's wonderful tools for contracting approaches that are outcomes-based and value-based processes to new kinds of insurance pools to help cover these, private or publicly funded. And if you want to see more of that, lots of resources, but one is our website that's called Paying for Cures. So just all one word, Paying for Cures, put in Tufts Medical Center, be the top Google hit for you. You'll see lots of details.
Thank you very much. Michael?
I'll keep being the platforms drum. We need regulatory principles and frameworks that can allow these principles to be employed for all rare diseases, not just in a few cases. And then rare disease is just the tip of the spear, in our opinion. And there's no reason why this can't ultimately extend to more common conditions as well.
Thank you. Now, Julia, I mean, you brought us here all these years ago. I remember that first meeting. I might have been at the table. What is your final word? And I hope you do still feel the optimism from today's conversations as we go forward. So what is your final word you want to share with us as I turn it back over to Garrett for final remarks?
Yes, I do feel a lot of optimism. So yeah, I do. And I don't normally hear, so I'm excited about that. I would say the FDA has an incredible opportunity right now to announce or roll out process approval. It's going to take a year or two for the first academic nonprofit or for-profit to get process approved. And in the lead up, it's going to look like an umbrella trial. So nothing changes and no legislation's needed. That can be done today. So I hope that I'm not here again next year at this meeting and we're still talking about it. And I hope that it's actually already been rolled out. And we know that it's possible because we're already hearing from you guys that, this is something you're interested in, and plausible mechanism, don't like the name, but I love the concept. And I think that, it's happening. So let's do it.
Great. Well, thank you. Thank you, Garrett, who will provide a wrap up and final remarks for this.
Thank you. Thanks.
This will be the most brief portion of the day. Thank you, Michelle, and to all our participants on the final session. Indeed, throughout the day, it's hard at the end to not jump up and regurgitate the 12 or 15 or 17 things that you were most excited about, but I will avoid that because that's what that previous session was for. I would implore folks in the same way that I did to start the day, though. These events are really wonderful. They're special to be a part of. They're an enormous amount of work to put together. There will be another one on March 30th, as mentioned. We talked, though, at the start about the importance of kind of like the 8-hour, 30 people in a room workshops on single topics, clinical, non-clinical, manufacturing, payer-related issues, platforms, data sharing. So hopefully between now and March 30th, far beyond just the work of Margolis, lots of that will happen. Because if not, the in-between period is kind of when all the work gets done. And it takes a village. So Margolis, CPATH, ARMS, N of One Collaborative, NLORM, industry, listed as a bit more broadly types of companies, to Sadek's point: clinicians, academics, regulators, patient advocates, payers, legal and policy experts, kind of the composite of the 120 folks in the room today and the 2,000 that registered online. We do have a survey. Oh, am I doing this the right way? There should be a link for a survey, and in case folks could take the time to give us some additional feedback, guide our future work, tell us how we did today and how we can do better, we would be grateful. Last part, in some ways, to me, the most important. Obviously, we thank all of our speakers, panelists, moderators today, and critically on my end, thanking our colleagues on the FDA planning team, Amy Comstock, Rick, Pippa Friedman, Kathleen Davies, Nancy Glom, Sandy Retzi. Those of you that are in this type of business know how much work goes into a day like today. So thank you for the partnership there. And then my favorite part is always how we end. On our end at Margolis, beyond folks that participated on the days today, behind the scenes, Luke Durashore and Hannah, I'm going to say Villaretta, Vidiello, on our comms and events team. You'd never see them. None of this could happen without them. The two people, though, that are responsible for this day happening more than anybody else in the world, they're in the back: Kristen Potziad and Mia Williams. Endless hours, months. And then, of course, everybody that attended here and online. We only probably got to 15% of the online questions that were submitted. I think there were about 35, and we were able to work in 5 or 6. All of the others are forwarded to our FDA colleagues. Hope to see you all again 4 months, 5 months from almost to the day. Take care.